Greetings and welcome to this satellite symposium sponsored by Chromadex, your partner in scientific discovery. Today, we will be discussing nicotinamide adenine dinucleotide in human health and disease, the state of the science on nutrition interventions. I am your host, Dr. Yasmin Nkrumah Elie, the Director of External Research at Chromadex. I will be joined by my co-chair and co-host, Dr. Ron Tian from the University of Washington, Seattle. My disclosures is that I am an employee of Chromadex, and I'm also a consultant for health and wealth with Dr. Yaz. Today, we have an absolutely wonderful panel of investigators from across the world that are going to be discussing NAD with us. First, we will have Dr. Tien, who will be framing the issue, why should we care about NAD? Following Dr. Tien's presentation, Dr. Charles Brenner, this year's ASN Mary Schwartz Rose Senior Investigator Award recipient, will be discussing a summary of the recent findings as it relates to NAD, human health, and disease. Following, Dr. Canto from the Nestle Institute and the Federal Institute of Technology outside of Switzerland will be comparing and contrasting NAD precursors and looking at those mechanisms. We will then have a discussion by Dr. Mackey of Indiana University evaluating the current state of clinical research. And finally, we will have a panel discussion with all of our panelists, hopefully answering some of your questions. In addition, if you have more questions, please feel free to reach out to our panelists, and they will hopefully get back to you quickly with answers to your questions. Now get ready for a very exciting panel.
Good morning. I also want to join Jasmine to welcome you to this symposium. My name is Ron Tien. I'm from University of Washington, Seattle. And I want to start by giving a very brief introduction of the issue about NAD metabolism in human health and disease. It's mostly to bring up the question, why should we care? For disclosure, I serve on the Scientific Advisory Board of Cytokinetics. Don't have any other conflict of interest. So this is the molecule we are going to talk a lot about today, NAD, nicotinamide adenine dinucleotide. And the structure is shown here. And it is exists in the cell in two forms, oxidized form NAD+, and reduced form NAD, NADH. By looking at this, you realize it's an electron carrier. It's heavily involved in oxidation reduction reactions. And interestingly, the oxidized form, NAD+, is also a substrate for a number of very important proteins, and that is involved in protein modifications, DNA repair, and signaling mechanisms. So this is a very critical molecule for our health. The interest in NAD metabolism started a long time ago, but it really surged when there are observations more than a decade ago showing that NAD levels showed age-dependent changes. So here's an example of the findings in old rats. When there is an age-dependent in almost all the tissues, a decline in NAD levels. And this is a major decline in NAD+. At the same time, there's an increase in NADH levels. So the NADH redox has also changed in an age-dependent manner. And probably more relevant and more interesting to us is the change in humans that here shown in the bottom. It's in the human brain. And this is a non-invasive measurement of NAD+, NADH, and the total NAD level over a wide range of age from 20 years old to 70 years old. And you can see there's age-dependent changes. And the main change is a slight decline in the overall pool, but there is a drastic change in NAD, NADH redox. And we always wondered what that means for aging. Theoretically, if you look at the role of NAD in the cell, NAD can be a core enzyme. It serves the role of carrying the electrons that's generated from substrate metabolism, and then bring the electron to mitochondria for oxidative phosphorylation, shown here on the left. So this means NAD metabolism is critical for energy metabolism. But if you look at the right side, the NAD is also a co-substrate for a number of very important proteins in the body, such as PARP and the probably most well-known sirtuins. And so the NAD as a co-substrate for these proteins plays a very important role in cell signaling. So how do we, if we want to target NAD in the cell for therapeutic purposes or just for health purposes, where do we start? So these slides gave you an overview of NAD metabolism. There's a number of pathways that NAD can be synthesized. The de novo synthesis here is shown in brown that starts with tryptophan. And there's also another pathway called Price-Handler pathway on the upper left corner, and that starts with nicotinic acid. And those pathways are very important developmentally, and they are slower pathways in mature cells. Probably the most active pathway in mature cells is the one showing on the right side, called Savage pathway in green. So in this pathway, the consumption of NAD turns it into nicotinamide. And so those are the major NAD consumers here, sirtuins, PARPs, and CD38. And then the nicotinamide will go back to NAD through the NMN pathway. And there's a number of places where this pathway can be targeted. For example, we can give precursors of NAD either at the level of nicotinamide mononucleotide NMN or at the nicotinamide riboside NR that we will hear a lot about. And there's also approach that to supply cells with nicotinic acid that can go through the pH pathway to replenish the NAD pool. And recently, there's also strategies to inhibit NAD consumers. In that way, the NAD level can be built up in the cell. So we were going to hear a lot of analysis on this pathway and the clinical consequences of targeting those pathways in the next few talks. And here, I just want to take a minute to show you what we found of NAD as a therapeutic target for heart failure. And so we first used a mouse model of heart failure by imposing an afterload on the heart by constrict the transverse aorta. In this way, the heart will gradually develop dysfunction by fighting against this high pressure. And as you see here shown in red, and the heart function gradually decline over time. If we supplement animals with NMN, we can mitigate the dysfunction of the heart. And in a slightly different model, we also induce pressure overload to induce dysfunction in a mouse model, but we didn't treat until the dysfunction appears. There about 4 weeks after the constriction of the aorta. You can see that in the red symbol, if you don't treat the animals, over the next 2 months, the function will decline significantly. But if that time we initiate a treatment with nicotinamide riboside, and we can prevent that dysfunction and can sustain the heart function. We also looked at the mechanisms. Those studies are already published. And one of the interesting mechanisms is manipulate NAD metabolism will protect the mitochondria, make it reduce its sensitivity to calcium-induced permeability transition pore opening, here showing on the right upper corner. And since then, we have initiated a number of clinical studies and look at the pharmacokinetics of nicotinamide in healthy individuals, and also examine the safety and tolerability of NR in heart failure patients. Those studies have been completed, and you can see the information on the clinicaltrial.gov website. And so with this data and the many more you're going to hear today, we ask question, is NAD a therapy for human disease? If you look in the literature, there's lots of enthusiasm for this. And here I just gave you some shot of the recent papers that published the title. You can see there's a losing enthusiasm for going for the therapy. And so I just want to give you a quick overview where we are to bring this to clinical research. And this is a courtesy of ChromaDex that they did a thorough search and listed how many clinical trials right now are registered. So there's nearly 900 studies on this topic in clinical trials. And there's about 500 listed NAD precursor as an intervention. And there's a breakdown, as you can see on the left side, which precursors being used. And on the right side, there are the top 10 clinical conditions that's been tried by manipulating NAD metabolism. Some of those we're going to hear from Dr. Markey later. So at this point, you might want to ask, if it's so good, it's so helpful for so many things, should we just put it in the water? Are we all set for this question? Actually not. There's many unsolved questions we need to address, and I think some of them will be discussed in today's presentation, and there might be more that we could address in a question and answer sessions. Here are just a list of few, such as what are doses we should use, and if we take it as a health supplement, what we should use versus if we take it for treatment. And we still need to understand better about pharmacokinetics and metabolism, NAD, its precursors, whether it's the same in healthy versus the diseased human. And we want to know, if we boost NAD levels, does it affect the cell at every subcellular level? NAD are compartmentalized in the cell. And also, as I mentioned at the very beginning, NAD exists in both oxidized and reduced form. If we increase NAD level, it comes in as oxidized form. Would it affect the redox in the cell? And what is really the mechanism that will promote health or treat disease if we increase NAD level? And we have a lot to learn from there. And a very important question in terms of supplement and treatment aspect, what is the long-term safety? I hope some of these questions will be discussed in the next few talks we're going to hear. We are here to listen to the words as experts' opinions. I hope you enjoy today's session. Thank you.
Good morning, everyone. I'm Dr. Carlos Cantó from Nestlé Research in Lausanne. As disclosures for my presentation, I'm an employee for Nestlé Research, and I also consult for Nestlé Health Sciences, which has commercial interests in NAD boosters. So during the presentation that I will be giving in the next minutes, I will try to discuss the relevance of different NAD precursors. So in the previous talks from Professor Brenner and Professor Tian, we've seen that multiple molecules can lead to NAD synthesis. These molecules classically have been tryptophan, nicotinic acid, nicotinamide, and since 2004, Professor Brenner also described nicotinamide riboside as a conserved NAD precursor from yeast to mammals. So the question that comes out when looking at this scheme is why so many molecules can lead to NAD, and why have we evolved to synthesize NAD from so many different entities? So the story gets more complex, because if we look at different tissues, apparently they have a different taste for all these molecules. So in a study published 10 years ago, they examined the levels of NAMPT, the critical enzyme to synthesize NAD from nicotinamide, and NAPRT, the critical enzyme to synthesize NAD from nicotinic acid. So what they've seen in this study is that some tissues have a really high expression of NAMPT, which means a really high availability to synthesize NAD from nicotinamide, while some other tissues prefer nicotinic acid based on NAPRT expression. This goes in line with some genetic models. For example, if we delete NAMPT in skeletal muscle or heart, these mice have really a lot of troubles to survive. While if we delete it in liver, the only thing we promote is a relatively mild decrease in NAD levels, which comes to say that different precursors have different relevances in different tissues. When we look at a more novel molecule like nicotinamide riboside, we know that the first and initial and essential step for nicotinamide riboside to synthesize NAD is the phosphorylation by a protein kinase called nicotinamide riboside kinase, or NRK1. When we look at the expression in different tissues, what we see is that NRK1 is really highly expressed in kidney and liver, and while it's there, it's not as highly expressed in other tissues. Again, pointing towards the fact that different precursors might have different affinities for different tissues as a main source for NAD synthesis. This said, the question that we'll be tackling today mostly will be, are all these NAD precursors redundant? This means, can you live on one precursor if you don't have another one? And to test that hypothesis, what we did is ask ourselves the question, can we live without nicotinamide riboside? What if we cannot use nicotinamide riboside as an NAD precursor? To answer this question, what we generated was knockout mice for NRK1, which is the essential enzyme to synthesize NAD from nicotinamide riboside. These mice do not express NRK1 at all, as you can see here on the Western blot. This lack of NRK1 is not compensated by any means by any other enzyme of NAD biosynthesis for other pathways, which means these mice can perfectly synthesize NAD from nicotinamide, tryptophan, nicotinic acid. And that's proven by experiments in primary hepatocytes, where when we treat with nicotinamide riboside or any other precursor, what we see is that specifically nicotinamide riboside synthesis is impaired in our NRK1 knockout mice. Nicotinamide or nicotinic acid NAD synthesis is not impaired, and nicotinamide mononucleotide, when given extracellularly, we know it needs to be dephosphorylated to nicotinamide riboside, hence why it's also impaired. So what do we know from these mice? What we know is that on regular diets, and at least until mid-ages, like 9 months of age, these mice look pretty much normal. So nothing really comes up as a problem in not using NR until this age. What we aim right now here, and the work I will be showing you, is what happens if we tweak with these components. What if we shift them from a regular diet to a high-calorie diet to mimic human obesity? And what if instead of taking them at relatively young ages, why if we let them age and see what occurs if we cannot use NR? So what happens when we take NRK1 knockout mice and we give them a high-fat diet? So what we see is that the first thing we examined was glucose tolerance, and what we see is that in low-fat diet, these mice look perfectly normal, perfectly fine. But when we challenge them with a high-fat diet, what we see is that these mice become glucose intolerant, heavily glucose intolerant, and insulin resistant. So not only that, when we examined why these mice were insulin resistant, what we could see is that whenever we injected insulin, insulin signaling was heavily affected based on AKT phosphorylation. We could also recapitulate that if we simply had primary hepatocytes from these mice, we incubate them with lipid, or we have them on a lipid overload, and we test insulin signaling, we see that lipid overloading is enough to completely destroy the ability of knockout hepatocytes to respond to insulin. When we looked at the livers from NRK1 knockout mice, what we saw is that, again, on low-fat diet, we didn't see any significant difference, yet when we pushed them on a high-fat diet, these livers became heavily steatotic, so really filled with fat inside, as can be seen here on histology, and also when measuring triglycerides through biochemical methods. This increase in lipid accumulation, we could associate that even by many results, even if I'm just showing here one, to the fact that these mice or these livers have a really impaired ability to oxidize fat, to use fat as an energy source. Here's a simple assay of palmitate oxidation. What we can see is that NRK1 knockout mice have problems to use fat as an energy source, hence leading to lipid accumulation. So also in a nutshell, and I will just review here in one slide many other findings, what we saw is that NRK1 knockout mice, when pushed with a high-fat diet, they have increased circulating markers of liver damage. They also show increased fibrosis, which is really rare to see on wild-type mice. So this is really a unique feature, showing up that really these livers are not doing well. We also see a huge amount of apoptosis and compensating proliferation, which is also a mark of really severe liver damage. And then when looking more closely at histology cuts and more molecular profiles, we also saw high levels of DNA damage, much more higher than in normal mice. So the question that we ask next is, what happens to NAD in these mice? Again, as we thought, on low-fat diet, NAD levels between wild-type mice and NRK1 knockout mice are completely comparable. Again, when pushed with a high-fat diet, we see that NAD levels are being pushed down in our knockout mice. They cannot sustain NAD levels. Again, we could recapitulate that in primary hepatocytes, just overloading them with lipids. What we see is that NAD levels in these hepatocytes overloaded with lipids are much lower whenever you don't have NRK1 in the cells. So looking at the other metabolites, and this is an interesting feature to discuss. Of course, one of the predictions, if we eliminate NRK1, as you can see here in the graph, is that NR should accumulate inside the tissue, which is exactly what we see. It's not processed, so it's accumulated. NMN levels, which is the immediate downstream molecule of NRK1 activity, is lower, as predicted from the decrease of NRK1, yet nicotinamide levels are completely perfect, which means that, again, that there's a specific impairment in NR, leading to NAD synthesis, as predicted from the NRK1 knockout model, yet they could perfectly synthesize NAD from nicotinamide, yet then they don't manage to do it, for some reason that I will discuss later. So again, pushing out to the possibility that NR deficiency leads to specific effects that cannot be compensated by other NAD precursors. And when analyzing the downstream consequences of losing NAD levels, in this case, in the NRK1 knockout mice, what we saw, and making a really long story short, we did not see really a huge influence on SIRT activities, which is one of the main paths by which NAD levels are supposed to modulate cellular function. What actually we saw was that the main path affected was poly-ADP ribosylation. We saw really low levels of poly-ADP ribosylation in our knockout mice compared to wild-type mice, despite the fact that they have much higher NAD levels, which comes to say that these mice have high levels of DNA damage, but simply they cannot sustain PARP activity to really recover from this DNA damage. In a nutshell, from what I explained so far, we know that hepatic NRK1 deficiency sensitizes to diet-induced metabolic damage. Also, the NRK1 mice that don't express NRK1 in the liver cannot sustain PARP activity upon lipotoxic-induced DNA damage. And finally, that NRK1 deficiency cannot be compensated by other precursors. These mice, and I want to stress that, can perfectly use any other NAD precursor. The diet that they eat contains nicotinamide. We've even supplemented these mice with nicotinamide, yet the phenotype of the NRK1 deficient mice cannot be recovered by other precursors. So this is a really genuine phenotype of lacking nicotinamide riboside. So the second point that I would like to discuss briefly is the response to aging. So how do these mice, what happens to these mice that cannot use NR when you simply let them age? And here we also saw some surprising findings. Just as a disclosure, here what we did was just to have wild-type mice, NRK1 knockout mice. We let them live until 22 months old, which is pretty much long for mice. They generally live between 24 and 30 months of life. What we saw is that the NRK1 knockout mice are much leaner than wild-type mice at advanced ages, not at normal ages, not until 1 year old, but since then they simply plateau or even lose a little bit of weight. That this decrease in body weight, it's really related to a decrease in fat mass. Yet one thing that was striking to us is that some tissues, even if they were leaner, some tissues were much bigger. That's the case, for example, for kidney or for pancreas, where the size was much bigger despite being smaller mice. So when we analyzed NAD levels in different tissues, what we could see is that some tissues, liver and kidney, had really significant decreases in NAD levels. This is completely in line with the fact that NRK1 expression is quite high in liver and kidney, yet not in many other tissues. And when we looked specifically at liver or kidney, what we saw is a really unique feature, and I would say common feature between both tissues, which is here's, for example, the case of liver. What we can see is that a number of circulating hormones like FGF21 or markers like ATF4 expression in the liver, they're really increased as a sign of mitochondrial dysfunction and mitochondrial stress. And we see also fibrotic signatures being highly expressed in these livers. And we can see in the histology next to it that age-wild-type mice have mild perivascular fibrosis, yet age-NRK1 knockout mice had really marked fibrosis on those livers, which probably makes them unfunctional. Also, when jumping to kidney, we saw exactly the same kind of profiles, increased RNA profiles of fibrosis, and also when we looked at the glomeruli in the kidney specifically, we can see really high fibrotic profiles. So, in a nutshell, and what I would take, or the message that I would like to transmit through this talk, is that on a healthy state, at least in mice, probably the NAD needs of our body can probably be met for dietary nicotinamide and nicotinic acid. And in line with what Professor Brenner has explained before, whenever we face challenges and we increase the NAD turnover of tissues, then the possibility of need for other NAD precursors, such as nicotinamide riboside, becomes critical. And if this apport of nicotinamide riboside is not over there, like in the case of the NRK1 knockout mice, this might condemn the organism to overt disease. So just in the last 3 minutes of my presentation, one thing I would like to show you is that probably we've only seen the tip of the iceberg when it comes to the different ways of synthesizing NAD. I come back to this slide that I showed at the beginning with 4 classical precursors: nicotinic acid, tryptophan, nicotinamide riboside. Yet we found out lately that probably there's one addition to this family. And this addition is the hydronicotinamide riboside, or NRH. So this molecule is just a reduced form of nicotinamide riboside, and as you can see, it's just one less in saturation in the nicotinamide ring. And the lab of Anthony Sauvé, as well as ourselves, we have demonstrated that this is a really potent NAD precursor. As you can see here in a dose response or a time course experiments, NRH in culture cells is an extremely potent NAD precursor. It can increase up to between 6 and 10-fold the levels of NAD in the cell, which is much, much higher than any NAD precursor known to this date. And also the actions are really fast. Within 5 minutes, we have really significant increases. One of the most striking findings is that despite both molecules, NR and NRH, have a strong similarity between them, it's just one reduction of the molecule, they use different paths to synthesize NAD. So NRH generally constitutes an independent NAD precursor. What we know is that NR, as I described previously, depends on NRP1 to synthesize NAD. What we know, and we described last year, and this has been confirmed lately by the Anthony Sauvé lab, is that NRH, it has to be phosphorylated by adenosine kinase and then transformed to NADH, and then this is the way it leads to NAD+. So as a confirmation of this experiment, when we put adenosine kinase inhibitors in cells, what you can see is that NRH enters the cell, yet it doesn't manage to synthesize NMNH, NADH, or NAD. So definitely NRH leads to NAD synthesis through adenosine kinase, and it constitutes a totally independent path from the classical 4 NAD precursors that I described previously. And then also as an interesting situation, when it comes to therapeutics, NRH also proves to be orally bioavailable. Here it's a really quick experiment in mice orally gavaged with NR or NRH. What you can see is that in line with the results in cells, NRH has a more potent effect on NAD. And in contrast with other NAD precursors, NRH can be found in plasma really nicely after oral administration at levels around the 10 micromolar range, which are, based on culture cell experiments, already aimed for efficacy. So with this, and the conclusions of my presentation are that NAD precursors are not always exchangeable. We find circumstances where only one of them, for example, nicotinamide riboside, will be effective for an indication. We also know that the preference for particular NAD precursors is tissue-dependent and stress-specific. Then we know that, based on our studies, that NR is required to sustain NAD levels and protect hepatic and renal functions upon high-fat diet and aging in mice. And then finally, that NRH constitutes a novel orally bioactive NAD precursor, and why not? This might be just the beginning of finding a new constellation of potential NAD precursors. So with that, I would just like to acknowledge the people that contributed to this work, especially the 5 ladies on top of this slide, and thank you very much for your attention.
Hi, I'm Kevin Mackey from the Indiana University Department of Applied Health Science in the School of Public Health in Bloomington, Indiana, and also I'm the Chief Scientist for Midwest Biomedical Research in Addison, Illinois. And today I'm going to be talking about emerging effects of NAD precursors in humans, the current state of clinical research. These are my disclosures. I do consulting and industry-funded research for a number of companies. And as we've heard in some of the previous talks, there are various ways to increase NAD in cells. You can supplement with nicotinamide, nicotinamide mononucleotide, nicotinamide riboside, or niacin. And so we're in the early days of clinical research in humans to understand the effects of supplementation in humans, but it's an area that is generating a lot of interest and in which I think there's a lot of potential to show benefits. As we've heard in some of the earlier talks, NAD levels decrease with age, and there are various conditions associated with aging where you consistently see low NAD levels with visual and hearing loss, hepatic fat accumulation, neurodegenerative and muscular degenerative diseases, skin disorders, cardiometabolic, renal diseases, infertility, and osteoarthritis are all conditions that have been associated with reduced levels of NAD. And we're just in the early days of exploring how NAD increasing through supplementation may help to prevent or manage some of these conditions. Now, there are other folks on the panel here who are much better than I at explaining some of the biochemistry, but essentially when NAD levels are low, you have potential impairments in energy metabolism, DNA repair, gene expression, and cellular stress responses. And these may contribute to the development of various cardiometabolic conditions. Furthermore, those cardiometabolic conditions may feed back and impair these cellular functions, and so NAD may help to prevent some of the adverse effects on cellular functions of the progression of those cardiometabolic diseases. So the first question is, are we able to increase NAD levels with supplementation? And there are various studies that have addressed this. I'm showing one here by Kanzi and colleagues, where nicotinamide riboside was given at doses of 100, 300, or 1,000 milligrams per day for an 8-week period. And compared to placebo, there was a clear dose response effect to increase whole blood levels of NAD, which we believe is a good indicator for tissue levels of NAD. And in that study, there was no indication of any safety signals, so it was well tolerated, and no changes in clinical laboratory and other parameters that would suggest safety concerns. Now, there have been just a couple of small studies done in people with cardiometabolic diseases. So in this slide, I'm showing a summary of results from a study of 40 healthy sedentary men, 40 to 70 years of age, all with body mass index above 30, so they all had obesity. And they were given 2,000 milligrams per day of nicotinic riboside for 12 weeks or placebo in a parallel arm trial. And what we see is that most things did not change. So glucose, glycated hemoglobin, and most lipids didn't change. There was an increase in triglyceride level from about 133 milligrams per deciliter to 159 milligrams per deciliter. That was statistically significant compared to placebo. But there was no change in ALT, a liver enzyme. And so for these parameters, the only one that changed was triglycerides, and they changed in a direction that was probably opposite of what we might have expected. They used in this study euglycemic clamp to evaluate insulin sensitivity, and that's the gold standard methodology, and there was no effect of supplementation on insulin sensitivity in this study. They looked at body composition, and they found no effects on either percent body fat or body fat distribution as indicated by subcutaneous adipose tissue and visceral adipose tissue. So no changes in body composition or body fat distribution in this study. They also looked at hepatic fat, and while there was no significant effect, there was a trend toward reduced hepatic fat. And looking at the individual responses, it's clear that the trend was really driven by people that had higher levels of hepatic fat at baseline. And so this suggests that additional studies really should be done in people with NAFLD, non-alcoholic fatty liver disease, who have higher levels of baseline hepatic fat, because it looks like there's at least the potential to see larger effects in people with higher hepatic fat at baseline. Although I'll emphasize that this is hypothesis generating at this point, and we really need more studies. This trial by Remy and colleagues is just out in e-publication form. This was a small study of 13 healthy, sedentary men and women, 45 to 65 years of age, all of whom were overweight or obese with body mass index 27 to 35. And they were given 6 weeks of nicotinamide riboside at 1,000 milligrams per day or placebo. And then after a 4 to 7 week washout period, they had the opposite treatment. So this was a crossover study, and they evaluated many metabolic parameters. They looked at intrahepatic lipid and intramyocellular lipid using magnetic resonance spectroscopy. They looked at sleeping metabolic rate, various measures of cardiac energy status and cardiac function, substrate oxidation, ambulatory blood pressure, and body composition. So even though it was a very small study, they had a wide range of measurements. And there was no change during supplementation for body weight, but there were shifts in this study in body composition. So there was a decline in fat mass and an increase in fat-free mass. And that was accompanied by an increase in sleeping metabolic rate. And that's what you expect to see when fat-free mass goes up, because there's a very strong correlation between fat-free mass and resting or sleeping metabolic rate. Like the other study I mentioned, they found no effect on insulin sensitivity or mitochondrial function, no effects on hepatic and intramyocellular lipid accumulation, no effects on cardiac energy metabolism or ejection fraction, and no changes in ambulatory blood pressures, plasma lipids, or markers of inflammation. So I'll emphasize, though, that this was a small study with a relatively short treatment period. Moving on to another study by Martins and colleagues. They looked at 24 healthy men and women, 55 to 79 years of age, average body mass index 24. So these people were generally in the normal weight range. And they looked at 6 weeks of 1,000 milligrams per day of nicotinamide riboside as compared to placebo using a crossover design. And they saw some modest but statistically significant reductions in both systolic and diastolic blood pressure. And then they also saw a reduction in pulse wave velocity, which is a measure of arterial stiffness. And so there was an indication that arterial stiffness was reduced, and it also appeared that the reduction was more pronounced in people who had higher levels to start with. And so again, this is really hypothesis generating, and it appears likely that studies are needed in people with probably higher blood pressure and possibly higher levels of pulse wave velocity or arterial stiffness. And so, more work is needed to put these results into perspective. Now, as mentioned earlier, there's biologic plausibility of supplementation of NAD precursors for protection against various types of stress, including damage done due to UV exposure. And so I won't get into the mechanisms other than to say that when you increase NAD levels, that may help with DNA repair, that may be useful in stress conditions such as sunburn. And then also there may be usefulness for reducing inflammatory responses to various stressors, including UV radiation. And that could have relevance with regard to chemoprevention of skin cancer. And this has been studied in a phase 3 double-blind randomized placebo-controlled trial, and the results were published in New England Journal of Medicine. In this study, 386 men and women with at least 2 non-melanoma skin cancers in the previous 5 years were studied, and they were given 1,000 milligrams a day of nicotinamide or placebo for 12 months. And the primary endpoint was the number of new non-melanoma skin cancers, and that would include basal cell carcinomas and squamous cell carcinomas that developed over that 12-month follow-up period. The results are shown here. The primary outcome variable showed a statistically significant reduction in the group that was given nicotinamide, 23%. And then there was a trend for reduction in each of the components. Basal cell carcinomas were reduced by 20% with a p-value of 0.12, and squamous cell carcinomas were reduced by 30% with a p-value right on the edge of significance of 0.05. So for the primary outcome variable, there was a significant reduction, and there were trends in each of the component variables. Also, the investigators looked at actinic keratoses. These are non-cancerous lesions that can be precancerous. So a small percentage of them do turn into basal cell or squamous cell carcinomas. And what was observed over the 12-month follow-up period was a significant reduction in the nicotinamide group that was statistically significant at each of the time points, 3, 6, 9, and 12 months. Now, there have been some additional trials that have been done to examine the effects of NAD precursors on cognitive function. Some of these are underway, and some of them are evaluating interventions that are classified as pharmaceuticals. One in particular, a phase 2 trial of nicotinamide as a treatment for early Alzheimer's disease, is underway with expected completion very shortly here in June of 2020. And in addition, there's a women with mild cognitive impairment to examine the effects of increasing NAD levels on brain function and cognition, and that has an estimated completion in December of 2020. And so we should stay tuned for more results with regard to effects on cognitive function and the progression of cognitive decline in people who have cognitive impairment at baseline. So to summarize, clinical studies of NAD precursors, mainly nicotinamide riboside, have confirmed persistent NAD elevation as well as safety and tolerability. And to date, results from clinical trials examining the cardiometabolic effects of nicotinamide riboside have had very small sample sizes. And so far, we've had some mixed results, with 2 studies showing no effect on insulin sensitivity, 1 study showing reduced fat mass, increased fat-free mass, and increased sleeping metabolic rate, but the other study showing no effect on body composition or body fat distribution. Also, studies on hepatic fat have been small and to date have shown no statistically significant effect, but some indication that there may be an effect in people with higher levels at baseline of hepatic fat, so more studies are needed there. For blood lipids, most of the studies have shown no effects, adverse or favorable, but 1 study did show a triglyceride elevation, and so follow-up is needed on that to further evaluate effects on the lipid profile of supplementation. And then with regard to blood pressure, the results have been mixed. In 1 study, there was no effect on ambulatory or resting blood pressures, and in another, there were reductions in systolic and diastolic blood pressure and a reduction in pulse wave velocity. And so more studies are needed, and in particular, more studies are needed in people with elevated blood pressure or elevated arterial stiffness at baseline. And then finally, promising effects have been shown for nicotinamide for skin cancer prevention in a phase 3 trial with 1,000 milligrams per day over 12 months. And there are potential effects on cognitive decline and performance. That's an active area of research, including nicotinamide being studied as a pharmaceutical for intervention to prevent the decline in cognitive function in people with prior mild cognitive impairment. And as Dr. Tian mentioned earlier, there are many ongoing studies of NAD precursors that are evaluating various aspects of human health. So we're in the early days of assessing the effects of supplementation as a preventive and also as a means of managing various diseases and conditions. And so stay tuned for more research, which should be available soon. So thanks for your attention, and I look forward to questions during the panel discussion.
So thank you, Drs. Tien. Thank you, Dr. Brenner. Thank you, Dr. Canto, and thank you, Dr. Mackey, for absolutely wonderful presentations, delving pretty deep into NAD, many of the benefits that we're just starting to see, some of the information about different precursors. And so we want to open up for questions, and we have a list of questions that we are prepared to ask, but also panelists, if you want to extend the discussion, we can do that. We have about 15 minutes, so we do want to be mindful for time. So our first question, and anyone can address this, is how should NAD status be measured? What is considered low status versus adequate versus really optimal? And how are these tied to health outcomes? And then the last part of that question is, given that we're still learning about all of this, what research gaps need to be filled in order to provide more definitive answers?
Can I be heard?
Yes. Yes, Dr. Brenner.
This is Charles Brenner, University of Iowa. We've, as you know, have been measuring the blood NAD metabolome for a number of years now, and we consider blood to be essentially a biomarker of people taking precursors, but not necessarily indicative of tissue NAD. Recently, we've completed a study on the liver NAD of people that came to a clinic in the UK for alcoholic liver disease or other liver conditions, including liver cancer. In the case of liver cancer, we were able to collect adjacent normal tissue, and we were able to show that human alcoholics have a depressed NAD metabolome. In the future, we think that some of the non-invasive techniques that are being developed with imaging are going to be able to report on NAD status in particular tissues, and that we'll also be able to find circulating biomarkers that report on tissue NAD.
Thank you. Does anyone else want to comment?
Yes, if I can add a few words to what Professor Brenner just mentioned. One has to keep in mind that NAD is a dynamic molecule, so it's all the time synthesized and consumed. And sometimes we don't see necessarily a decrease in NAD, which doesn't mean there's not a need for an additional supply of NAD precursors, for example. This is the case, for example, of our results, for example, in obese mice, where we can see that obesity does not necessarily lead to a decrease in NAD, yet it needs to, or it leads to an increased demand for NAD. So I think that in addition to NAD, one could think about maybe finding out some, or one could think about other potential metabolites that might not only indicate NAD levels, but also NAD turnover levels at this point. And this could be methylated forms of NAD degradation products. This can be other types of metabolites, and Professor Brenner has described that depending on how you supplement, you might find some bizarre metabolites in blood like NAD, which could be useful for diagnosis. So I think there's also a path of evolution on this direction.
And this is Kevin Mackey. I'll add to that the ideal study design is to have a biomarker that you can use to identify people with low NAD status, and then have additional features such as insulin resistance or elevated blood pressure or dyslipidemia, what have you, and then look at correcting that low NAD status and the effects on your outcome variables of interest. And to date, we've had somewhat of a challenge in that without good validated biomarkers of status that, it's hard to know what percentage of the people that you're giving supplementation to, have a higher potential to benefit from that.
I want to jump in here. So this is Ron Tian. I think Kevin and Carlos brought up a really important point. It looks like the basic research has demonstrated a very remarkable benefit, and we're struggling in translation. And so far, the clinical research has small numbers and very mixed results. I think a good question to ask here is how can we do better in clinical research? I think biomarker is clearly an important thing we should go after, and probably whether there is a way to identify the subpopulations that most likely will benefit for this type of therapy. So I would like the panels to comment more and specifically on what we should do to promote the clinical research.
Thanks for coming to my talk. Charles Brenner from University of Iowa, and it's my pleasure to present to you my work on nicotinamide riboside and its relationship to conditions of metabolic stress. You can find me on email, you can follow me on Twitter at Charles M. Brenner, and my disclosures are that some of the work that I'll be discussing involves my intellectual property that was developed by Chromadex Corporation, for which I serve as Chief Scientific Advisor. I'm also a stockholder, and I've received research grants from Chromadex. I'm on the Scientific Advisory Board of Cytokinetics, and recording this talk on May 21st, 2020. I'm also showing you funding agencies. So NAD coenzymes, and there are 4 NAD coenzymes, are the central catalysts of metabolism. Metabolism is not just converting what we eat into the biological energy of ATP. Metabolism is converting everything that we eat into everything that we do and everything that we are. And depicted here on the left is nicotinamide adenine dinucleotide. As Dr. Tian pointed out, it's an electron acceptor. The reduced form is NADH, shown on the right. This process of NAD reduction linked to fuel oxidation is the way that we burn our fuels. NADH is largely reoxidized at the intermitochondrial membrane in order to make ATP, but in addition, NADH is reoxidized to NAD in order to maintain blood glucose. So in gluconeogenesis, NADH is reoxidized to NAD+, and in generating ketone bodies, NADH is also reoxidized to NAD. When you put a phosphate or a NAD kinase puts a phosphate on the 2-oxygen of the adenine part of this dinucleotide, you get an NADP, and then there's yet another redox couple, NADP, NADPH, which have absolutely essential roles in metabolic processes. NADP is the primary electron acceptor for the pentose phosphate pathway. That's how we make RNA and DNA precursors, among other things NADP is used for. And NADPH is the electron donor for free radical detoxification, as well as for biosynthetic processes, including making all the lipids that we need for every cell membrane and making things like cholesterol and steroids. So these are really fundamentally crucial coenzymes. As Dr. Chen pointed out, there are also NAD consumption pathways. So now you're seeing NAD at the top, and you're always seeing in A, B, and C some type of enzymology that is breaking the nicotinamide moiety. You see it being kicked off from the rest of the NAD molecule, which is called ADP ribose. Today, I want you to pay particular attention to path A on the left, which has ARTs and PARPs, ADP ribose transferases and poly ADP ribose transferases. So these are enzymes that will transfer an ADP ribose moiety to a protein side chain or to ADP ribose itself in order to do some sort of a signaling function. Turns out a defense function in many cases. And then cyclic ADP ribose synthetases and sirtuins are other enzymes with similar enzymology. They're using NAD as a substrate, and they're consuming NAD in the process. We think that in part, these NAD consuming enzymes explain a 100-year-old problem called pellagra that presented itself in the American South with really what looked like a pandemic in 1920. There were 1 million people in the United States that presented with diarrhea, dermatitis, dementia, and ultimately many of them suffered death. And they presented, as this individual does, with rosacea, with glove and stocking lesions, and this led to the issuance of the very first grant and the creation of the Public Health Service, the forerunner of the United States National Institutes of Health, in which Joseph Goldberger discovered that this was not an infectious disease. It was actually a disease of poor diet. And Goldberger found that fresh food, and ultimately it was shown that tryptophan, nicotinic acid, and nicotinamide are pellagra preventative compounds. And so this, of course, led to the discovery of the 2 classical forms of vitamin B3, nicotinic acid and nicotinamide. Anybody that has been paying attention has noticed is NAD made. As Dr. Chen pointed out, there is a pathway that I depict from the left-hand side here of tryptophan, which is the de novo pathway that takes 8 biosynthetic steps. There's the pathway coming from the lower left, driven by nicotinic acid. These are yeast gene names, by the way. And then there's this pathway that involves production and then salvage of nicotinamide. And this is all the received wisdom that was available to me in the early 2000s when our group started working on NAD synthesis. We questioned the completion of the NAD gene set, and I won't go into all of the details, but in the course of doing this work, we discovered nicotinamide riboside, NR, as an unanticipated vitamin precursor of NAD that bypassed all of the known routes to NAD. That's Pavel Bieganowski, the first author of the 2004 paper identifying the NR kinase pathway. And then subsequent work identified additional NAD precursors, including nicotinic acid riboside, additional biosynthetic pathways, genes, transporters. And now if you fast forward to 2020, we can say a number of additional things about the NAD gene set. One is that a gene in vertebrates from nicotinamide to NMN called NAMPT is not steady in its expression. So it declines in aging and it declines in many inflammatory conditions. The nicotinamide riboside kinase pathway that we identified is induced by many types of damage and metabolic stress, and I'll show you two examples of that in this 20-minute period, one in heart failure and the other in coronavirus infection. That the nicotinamide to nicotinic acid transformations occur largely in the gut through the microbiome. We and others have discovered that the genes involved in NMN to NAD synthesis have a relationship to neuroprotection, and that one or more of the genes that converts NAD to nicotinamide, and in that case to cyclic ADP ribose, is required for many types of neurodegeneration. So you can see just from the gene designations and the relationships of these genes to known diseases and conditions that NAD is quite important in health and disease. So we realized that there was an essential tool that we were going to need to develop to quantify the NAD metabolome. I'm showing you a picture of Sam Trammell, who developed our LC tandem mass spec method. We won't go into the details, but this allows us to determine whether the NAD metabolome is stable in a condition or whether it's actually under attack. We also, and so to make the 15 years of work go by in a few minutes, I'm going to give you a summary of the conditions of metabolic stress that challenge the NAD metabolome, and some of this is our work, and some of this is others. But in alcohol metabolism, NAD is disturbed. We have a paper coming out in Hepatology Communications that shows that with human samples. In mice, we've shown that overnutrition leads to a depressed hepatic NAD pH. Anthony Saube has shown that insufficient noise to induce hearing loss, there's a loss of cochlear NAD. It's well known that DNA damage caused by sun and reactive oxygen will activate PARPs and dysregulate the NAD system. And then I'm going to kind of skip ahead and point towards heart failure, pregnancy and postpartum, and infection as other conditions of metabolic stress that disturb the NAD system. So here is one such system. This is a genetic model of dilated cardiomyopathy and heart failure, in which Matthias Marixey and Nicolas Duguay's favorite gene, which is called serum response factor, is knocked out in the heart. And before 2 months of age, when these mice start getting a dilated cardiomyopathy, Duguay and Marixey and I discovered that the level of cardiac NAD was down, and the gene expression of one of our favorite genes, nicotinamide riboside kinase 2, was highly induced. The NAMPT gene that is required for nicotinamide salvage expression is going down. The NMRK2 gene is going up. And as you can see in the middle, the same thing is found in human heart failure. NRK2 expression is going up, NAMPT expression is going down. We showed that by 8 different criteria, that including the left ventricular ejection fraction, the fraction shortening, and 6 other molecular cardiology parameters, that serum response factor deletion mice have depressed cardiac activity, and that oral administration of nicotinamide riboside can protect all of these parameters, all of these phenotypes. And we further showed that nicotinamide can't rescue these phenotypes, nor can it rescue the attacked NAD in the failing heart. And so we think that it's particularly important that there are different gene encoded biosynthetic routes to NAD, and that the nicotinamide riboside kinase pathway is favored in conditions of metabolic stress. If you read this paper, you'll see that under conditions of heart failure, AMP kinase is activated. That's Rong Tien's favorite, one of her favorite pathways. And it turns out that AMP kinase turns on the nicotinamide riboside kinase 2 promoter that is allowing the failing heart to restore its NAD levels without costing a lot of ATP to do that. And so I think that that's a fascinating theme in metabolic stress. Now, the last little vignette I'm going to tell you today is highly topical. We're not meeting in Seattle this year for Nutrition 2020 because of the coronavirus pandemic. And what I want to tell you is that the first stage of the innate immune response to coronaviruses and all viruses that go through double-stranded RNA intermediates is something that is termed an interferon response. And I'm going to show you that PARPs, and they're actually mono ADP ribose transferases, but they're called PARPs in the literature, are among these interferon activated genes. And I don't have time to show you all of the data, but many of these genes have been shown to have a critical role in antiviral defense mechanisms. So the interferon activated genes are trying to shut down viral protein synthesis so that the initial cell that is infected by a coronavirus doesn't allow viral replication. Innate immunity is something that a naive, non-professional cell, not a professional immune cell, but a naive cell can do to shut down replication before antibodies are even made. And so here are some of the data. And if you're interested in this, you can look at Charles Brenner on the bioRxiv and you'll see our preprint. This work is under peer review right now. And so this shows the gene dysregulation of, we looked for 75 genes that control the NAD metabolome. And you can see that genes that are to the right of zero are upregulated, those that are below zero, going left of the zero on the x-axis are downregulated, and you can see there's a whole bunch of genes called PARP that are upregulated by coronavirus infection. We've looked at this in cell lines, we've looked at this in infected ferrets, we've looked at this in the lung of a person that died of COVID-19. You'll also see the nicotinamide riboside kinase gene is strongly induced by coronavirus infection. So what does this mean, and what are the consequences? We've further shown, and this is with a murine virus that causes a hepatitis disease in mice, that coronavirus infection knocks down cellular NAD. It does this in cell lines, it does this in the target tissue, which are macrophages in the case of the murine HV virus, murine hepatitis virus, knocks down NAD, knocks down NADP, and interestingly, knocks down ADPR as well. When we look at one of the genes or one of the enzymes, PARP10, that is induced by coronaviruses, we can monitor PARP10 activity by Western blotting for this ADPR modification. And these data show that PARP10 has limited activity when you overexpress it, and that you can actually boost PARP10 activity with agents that boost NAD. And so these data and the fact that the nicotinamide riboside kinase pathway are transcriptionally induced by coronavirus infection suggest that we test whether nicotinamide riboside boosts the innate immune defense, and we're doing those experiments in cell lines, in rodents, and if justified, we will move that work into human clinical trials. So to summarize, when the NAD system at the metabolite level and at the gene level is disturbed, and the nicotinamide riboside kinase genes go up and NAD goes down, nicotinamide riboside is very likely to help. We've seen that in more than 10 different rodent systems. It is a common signature of metabolic stress, from noise-induced hearing loss to heart failure and coronavirus infection, that NAD is going down, whereas nicotinamide riboside kinase genes are transcriptionally induced. You'll see data further in this symposium. The nicotinamide riboside has been safety tested to 2 grams per day. There are placebo-controlled trials, very short duration trials and small number N trials that show anti-inflammatory effects in as little as 3 weeks. There's a newly published paper in the American Journal of Clinical Nutrition showing body compositional improvements in 6 weeks. And it also looks like we're moving the needle on liver fat mobilization in just 12 weeks' time. There are many clinical trials in progress, and you can follow me on Twitter for more updates and the latest news when papers go out on the bioRxiv and when they are peer-reviewed and available in literature. Thank you so much for your attention.
So thank you, Drs. Tien. Thank you, Dr. Brenner. Thank you, Dr. Kanto, and thank you, Dr. Mackey, for absolutely wonderful presentations, delving pretty deep into NAD, many of the benefits that we're just starting to see, some of the information about different precursors. And so we want to open up for questions, and we have a list of questions that we are prepared to ask, but also panelists, if you want to extend the discussion, we can do that. We have about 15 minutes, so we do want to be mindful for time. So our first question, and anyone can address this, is how should NAD status be measured? What is considered low status versus adequate versus really optimal? And how are these tied to health outcomes? And then the last part of that question is, given that we're still learning about all of this, what research gaps need to be filled in order to provide more definitive answers?
Can I be heard?
Yes. Yes, Dr. Brenner.
This is Charles Brenner, University of Iowa. We've, as you know, have been measuring the blood NAD metabolome for a number of years now, and we consider blood to be essentially a biomarker of people taking precursors, but not necessarily indicative of tissue NAD. Recently, we've completed a study on the liver NAD of people that came to a clinic in the UK for alcoholic liver disease or other liver conditions, including liver cancer. In the case of liver cancer, we were able to collect adjacent normal tissue, and we were able to show that human alcoholics have a depressed NAD metabolome. In the future, we think that some of the non-invasive techniques that are being developed with imaging are going to be able to report on NAD status in particular tissues, and that we'll also be able to find circulating biomarkers that report on tissue NAD.
Thank you. Does anyone else want to comment?
Yes, if I can add a few words to what Professor Brenner just mentioned. One has to keep in mind that the NAD is a dynamic molecule, so it's all the time synthesized and consumed. And sometimes we don't see necessarily a decrease in NAD, which doesn't mean there's not a need for an additional supply of NAD precursors, for example. This is the case, for example, of our results, for example, in obese mice, where we can see that obesity does not necessarily lead to a decrease in NAD, yet it leads to an increased demand for NAD. So I think that in addition to NAD, one could think about maybe finding out some, or one could think about other potential metabolites that might not only indicate NAD levels, but also NAD turnover levels at this point. And this could be methylated forms of NAD degradation products. This can be other types of metabolites, and Professor Brenner has described that depending on how you supplement, you might find some bizarre metabolites in blood like NAD, which could be useful for diagnosis. So I think there's also a path of evolution on this direction.
And this is Kevin Mackey. I'll add to that the ideal study design is to have a biomarker that you can use to identify people with low NAD status, and then have additional features such as insulin resistance or elevated blood pressure or dyslipidemia, what have you, and then look at correcting that low NAD status and the effects on your outcome variables of interest. And to date, we've had somewhat of a challenge in that without good validated biomarkers of status, it's hard to know what percentage of the people that you're giving supplementation to have a higher potential to benefit from that.
I want to jump in here. So this is Ron Tian. I think Kevin and Carlos brought up a really important point. It looks like the basic research has demonstrated a very remarkable benefit, and we're struggling in translation. And so far, the clinical research has small numbers and very mixed results. I think a good question to ask here is how can we do better in clinical research? I think biomarker is clearly an important thing we should go after, and probably whether there is a way to identify the subpopulations that most likely will benefit for this type of therapy. So I would like the panels to comment more and specifically on what we should do to promote the clinical research.
I think I can address that. This is Dr. Brenner again. I'd like to point people to a newly published editorial in the American Journal for Clinical Nutrition. It's Flew-Hardy and Brenner, 2020. And we pointed out that there are some things that people ignored in the preclinical research that really are quite important. And then that created some assumptions that led to design of clinical trials that were sort of overly optimistic, too short, and not mechanistic enough. So the first point is that in the mouse obesity studies, it's actually not NAD plus that is the key NAD metabolite that declines in the liver of an overfed mouse. There's some decline in NAD plus, but there's a much greater decline in NADP, NADPH, and that's, I think, because in this particular condition of metabolic stress, there's a storm of reactive oxygen species that is churning NADPH and really challenging the NADPH system. So you have to sort of look at all of the available compounds in the NAD metabolome to really see what's going on. Second, because a lot of clinical researchers like to measure insulin sensitivity, and because we found in our first mouse and our paper with type 2 diabetes and obesity that there was better glycemic control, it was assumed by clinical trialists that nicotinamide riboside was directly addressing insulin sensitivity. But as we pointed out in this brand new editorial, there's a lot of different parameters that could be the driver parameters responding to NR, and others would be sort of going along for the ride. So, For example, if NR promoted a malabsorptive mechanism or NR promoted hyperactivity, then one would have spared mice from weight gain, and they would have, not had such an excess of calories, and then they would have started regulating their things. And it doesn't, the fact that their blood glucose was better does not mean that NR directly was involved in regulating insulin sensitivity. And if you look carefully at the data in Dollerup, which Dr. Mackey summarized, and in the data from Remy, it really looks like it's fat mobilization that is the direct target of reboosting the NAD. That as Dr. Mackey pointed out, in the Danish obesity study, it was lipid fat that was being mobilized, and they couldn't detect, it was actually I was on the paper, we couldn't detect a P less than 0.05, but that's because the people were not randomized for their lipid fat. They were randomized for their insulin sensitivity. Similarly, in Remy et al., there was an improvement in body composition. So they're mobilizing their fat. They're not losing weight yet because it was only done for 6 weeks. So I would suggest that people be mobilized for their body composition, randomized for their body composition, randomized for their hepatic lipids, and for the trials to be done of a sufficient duration, probably 6 months, and that the standard of care be increased physical activity. So if everyone is exercised and then you have a placebo control, NR versus placebo, I think you're going to see people responding by mobilizing their fat, and that's going to be the primary driver effect of boosting their NAD, and then other metabolic parameters are going to follow.
And incidentally, just a comment about that, increased fat mobilization could be a potential explanation for the increase in triglyceride level that was observed in one study, in that increasing free fatty acid exposure of hepatic tissue will tend to increase production of VLDL particles and triglyceride enrichment of those particles.
Absolutely. And so we think that if you combine the potential lipid mobilizing effect of nicotinamide riboside with exercise, and you do the study for sufficient amount of time, you're going to see real fat clearance and substantive changes in people's body composition and other cardiometabolic parameters.
So from my side, I would also like to add a couple of things in the direction a bit different from what Professor Brenner and Mackey have shown, which I agree completely with them, which is that, of course, the expectations on nicotinamide riboside were extremely high based on model organisms, especially mouse models. But one has to remember that mice have a completely different metabolic rate than humans, so much higher in this case, which probably is more prone to create NAD deficit situations than what we have in humans. So I think we might need to scratch a little bit more on the clinic to find these situations of NAD deficiency in humans. And I think we're reaching this direction. There's the recent paper published by the Anu Vartirala's lab showing that in some mitochondrial myopathy patients, there's a clear decrease in NAD levels. Charlie has commented on his work with alcoholic patients where he sees a decrease in hepatic NAD levels. So I think we're reaching this direction, but I think that was a key element that was missing to this date. Many of the studies that have been done to this date are in healthy humans, maybe at risk of disease, but still rather healthy humans, where NAD levels must still be relatively protected or contained. And we might need simply to look for these conditions where NAD levels are really suffering. And I think this is coming out recently.
So I have another question for the panelists. So there's a lot of people interested in taking NAD precursors or boosting the NAD levels in one way or the other for just for health, not treat particular disease. So this is more like a health supplement approach. So what would the panelists recommend for targeting the NAD levels just for the nutritional purposes.
Well, since no one is jumping in, I will jump in. This is Kevin Mackey. I think that there is a lot of potential, and from animal studies and mechanistic studies, we have good reason to believe that declines in NAD over time with aging and in specific conditions may produce health effects that are adverse. And so for people who are interested in maintaining, general health and, maintaining something closer to an optimal level of NAD, it's hard at this point to say you should take this supplement at this dose for this length of time and you'll observe these benefits. But I think it's equally likely that we can say that if you supplement, and we've mentioned various ways to supplement to increase NAD, that it's likely to reduce the degree to which NAD levels in tissues are suboptimal. And to date, we can't give strong recommendations, I don't think, on what dose and for how long. But there is a lot of research that's ongoing that should help clarify that in the not too distant future.
One of the interesting things that we're learning from the COVID-19 pandemic is that there are many pre-existing conditions of metabolic stress, including smoking, obesity, hypertension, that are associated with worse SARS-CoV-2, COVID-19 outcomes. And it will not escape your notice that these are many of the conditions of metabolic stress, including age, that disturb the NAD system. Many of the types of stresses that disturb the NAD system are virtually inevitable. So we need sun and oxygen. We enjoy food and drink. We are exposed to noise. When we're traveling again, we're going to be exposed to time zone disruption. And for sure, we are exposed to viral and bacterial pathogens that activate NAD-dependent defenses. So I think that the use case for healthy people taking something like Niagen as a safe, orally available, and GRAS and NDI certified nutritional supplement is a reasonable one, and we understand the safety, and as there are more clinical trials done, I think that we're going to see some measurable benefits for people. You can clearly see the anabolic effects of taking nicotinamide riboside. Anybody that has taken it for a month notices that their hair and fingernails grow faster, and there's often noticeable effects on skin, sleep, and other parameters. None of those things have been established in randomized placebo-controlled trials, but the safety is pretty clear, and the upside of having improved NAD status for immune health is pretty clear at this point.
I would further add to your comment, Charlie, that even your work has somehow elucidated a few ways in which we can enhance the ability to increase the use of these precursors. So it's quite well known that exercise can trigger the expression of enzymes aimed to increase NAD. Work from Mary's study with you, Professor Brenner, have demonstrated also that PPARs might also impinge on NAD-related paths. So, and these are paths that probably can tackle through some way through nutrition. So I think there's really good avenues for research in these directions.
So if we have a couple more minutes for the Q&A, I want to bring up this question that we so far haven't touched today. That is, the NAD metabolism is clearly involved in cell growth. And there are studies by Dr. Brenner and also reports from Switzerland that NAD metabolism is important for postnatal growth, for stem cell proliferation, and actually in the cancer field, NAD metabolism is a target to kill the cancer cells. So I would like to invite the panelists to comment on while we're trying to boost NAD metabolism for health. Is there anything we should take into consideration of cancer risk? I really enjoyed Dr. Markey's talk about NAD to prevent skin cancer. And how about other types of cancer and how we consider the issue?
Well, this is Kevin Mackey. I'll start by saying that my main focus is cardiometabolic health. And so, you know, I'm less familiar with the theoretical issues and certainly not familiar with the clinical research that may have been done in the area of cancer other than with the prevention of skin cancer, which I think is very exciting. So I'll just say that, you know, the data from the skin cancer prevention was impressive with nicotinamide, and I'd really like to see more studies, but I'd also like to see studies that are longer term that evaluate potential effects on other types of cancer.
Consistent with what Dr. Kanto was talking about in terms of liver function and loss of DNA repair with loss of the nicotinamide riboside kinase pathway, it's been shown in a mouse model of hepatocellular carcinoma that supplementation with nicotinamide riboside is protective against mutagenesis as well as carcinogenesis. So there are a number of indications from preclinical studies that suggest that NR and essentially all of the NAD precursors will be safe for people to take. We've, as you know, done a lot of work on NR and nicotinic acid riboside as protective agents against neuropathy, and there's an ongoing clinical trial at the University of Iowa that's testing nicotinamide riboside in breast cancer patients in order to protect against development of neuropathy. It's early days in terms of the clinical experience with NR, certainly in cancer, but there's nothing that has come up that gives us cause for concern.
I would even add more in here. I mean, not only your research has shown previously that NR can be protective against neuropathy or pain feeling after injury. I would say that when it comes to cancer, I mean, of course, we cannot generalize. I mean, each cancer can have different cancers can have extremely different metabolic properties. So I think we have to go a bit beyond that in the sense of thinking that NR will be a magic bullet for any kind of cancer. Yet we know that the NR can be used somehow in combination with cancer treatments as a way to relieve pain, potentially. Also, research in which I collaborated in the past by the lab of Olayana Beiras has shown that it can also recover to some degree hematopoietic lineages after chemotoxic treatments. So I think that here is probably the avenues that we would need to focus a bit more, rather than the metabolic profiles of cancer, which can be extremely varied.
So thank you to all of our panelists for such a great discussion, for absolutely wonderful presentations. There was actually a recently published paper by Hamady out of the University of Iowa and Dr. Hammond's lab. So definitely want to refer this to all of you. And the title is Nicotinamide Riboside Relieves Paclitaxel Induced Peripheral Neuropathy and Enhances Suppression of Tumor Growth in Tumor-Bearing Rats. So still not clinical research, but definitely more to add to the conversation. So thank you again to our speakers. We recognize that there are a significant number of research gaps, and there is a need for more NAD research to answer these questions as we're looking at, and as we have discussed, in the areas of biomarkers, effective methods for NAD detection, and still even answering some of the cancer questions and more. So if you are a nutrition researcher and you're interested in getting into NAD work, or even if you're outside of the nutrition field, we want to introduce you to our CERT program, and we provide preclinical and clinical materials for NAD research, complementary for great proposals. And we also are able to help you develop your proposals and support your grants. So if this is something you're interested in, please reach out to us for our external research program. There's no cost for anyone to do this and to reach out to us, and we want to be able to support and advance NAD research as much as possible. So again, thank you to all of our panelists. Thank you to my co-chair, Dr. Tien, for such a wonderful discussion. We hope to see you at future nutrition meetings, and thank you again.
Well, good morning, afternoon, or evening. I'm Catherine Price, Director of Strategic Initiatives and the lead for Nutrition Innovation at PATH, as well as a Secretariat member of the Bridge Collaborative, which aims to find sustainable solutions for both people and the planet. I'm joined today with my co-moderator, Madhu Mboya, from the Global Alliance for Improved Nutrition, and three very distinguished panelists working across the environment, food systems, and nutrition sectors. The session today is entitled Nutrition in a Pandemic Environment, and I'd like to take a minute to introduce you to our three panelists. The first is Dr. Christian Walser. He'll be presenting on the link between zoonotic diseases and the food system. He is the Director of Health at the Wildlife Conservation Society and a professor at the University of Veterinary Medicine in Vienna, Austria. Dr. Walser is an internationally recognized diverse expert in working with wildlife, especially megavertebrates, carnivores, and primates gained from combined years of leadership and research in Europe, Asia, and Africa. He acts as a consultant in wildlife matters for various organizations such as UNDP and the World Bank, and has authored numerous peer-reviewed research and publications, as well as book chapters. Dr. Walser is the recipient of several research and service awards and is a board-certified wildlife veterinarian. He will be followed by Dr. Marie Ruel, who will be presenting more specifically on the impact of COVID-19 on global food systems. Dr. Ruel is the director of the International Food Policy Research Institute's Poverty, Health, and Nutrition Division, and has led the multi-country program on challenges to urban food and nutrition and the Global Regional Project on Diet Quality and Diet Changes of the Poor for over 20 years. She has also worked at the Institute of Nutrition of Central America and Panama and the Pan American Health Organization, and her current research focuses on the evaluation and strengthening of a wide range of integrated multi-sectoral development programs in the areas of agriculture, social protection, and health, and at building the evidence on their role in reducing maternal child malnutrition globally. She has published extensively and has served on numerous international expert committees, such as the National Academy of Sciences and the International Zinc in Nutrition Consultative Group, as well as the Micronutrient Forum. Dr. Ruel holds a PhD in international nutrition and a master's in health sciences. Finally, our third presenter will be Dr. Megan Parker, and she will be presenting on next generation tools to track malnutrition and improve resilience. Dr. Parker is a nutrition epidemiologist with PATH, working on the nutrition innovation team, and she has over 15 years of experience in global public health, working on child malnutrition interventions across Southeast Asia and Sub-Saharan Africa. While at PATH, she has focused on research, launch, scale-up, and policy work related to a number of nutrition interventions, including fortified rice, edible insects, as well as a number of diagnostic tools and validation studies. She holds a PhD in nutrition interventions and epidemiology. In terms of the format of our program, we'll spend the first hour listening to 3 pre-recorded presentations, and then our panelists will join us live for a 30-minute question and answer period. You can submit your questions through the live stream. Thank you all for your interest in this timely and relevant set of presentations. We're delighted to have such experts on our panel, and we're looking forward to a very lively discussion following our presentations. Thank you.
So good morning from the Bronx. I hope you're all well in your respective locations throughout the world, as far as I'm aware. I'm going to be speaking to you today about spillover events, zoonotic diseases, and how it relates to food consumption, and basically on how we possibly or definitely need to rethink our relationship with wildlife and wild places. So this is coming to you from the Bronx, from the Wildlife Conservation Society. We're a large global conservation NGO with quite a robust and strong health program working in some 60 countries around the world. I'm sure all of you have been obviously following the ongoing pandemic, have read all the books that are out there, and but I still want to take you through some of the background of how this zoonotic spillover event happened, what we know, and also a lot about the things we don't know at this time, and how that possibly relates to our use of food, and especially how we use livestock and wildlife for food. So what do we actually know? So we do know that, zoonotic diseases are diseases that move between animals and humans. They go both ways. They go, in this case, of course, we're particularly worried about disease spillover from animals into humans. But in my field of work, of course, we're also particularly worried about diseases that spill over from livestock, for example, into wildlife populations and cause massive devastation. Point out right at the beginning that there are 3 ongoing pandemics at the moment. There's obviously the COVID-19 that we're all acutely aware of, but there's also African swine fever and the peste de Pitti ruminant, disease of goat and sheep, which is having devastating effects on obviously on livestock, but also in our area of wildlife. So emerging infectious diseases are really dominated. So these are the new infectious diseases. They're dominated by zoonotic diseases. And from these zoonotic emerging diseases, about 3/4 of them have their origin in wildlife. So wildlife does have a really, really important role when it comes to these emerging infectious diseases. And the emerging infectious diseases are the ones that cause us the most concern at the moment. They are obviously in relationship to other diseases that afflict humans. They are quite small if you compare it to the effects of cardiovascular disease or anything. Infectious diseases are minimal, though they cause, as in this pandemic, the economic impacts are massive as well. The frequency of emerging infectious disease has been increasing steadily and markedly over the last 5 decades. Some of the zoonotic diseases I'm sure you're aware of is HIV/AIDS, Ebola, the avian influenzas, then SARS, of course, the original SARS, Nipah virus, Hendra virus. Those are all quite well-known emerging zoonotic diseases, and that have impacted humans on varying scales. Now, when we go back to the ongoing coronavirus outbreak, what do we know? Well, there's a lot we don't know. But the disease was basically noticed and noticed. So sometime in mid-December, there was more and more reports of a specific pneumonia and seemed to have infectious nature. And the cases were coming out of a city in China, out of Wuhan, as everyone knows, of course. And originally, from the first 400 or so cases, more than half of the patients had direct links to this large, what's called a wet market. So it's a large market that sells a variety of goods. And in this early transmission, there's a slight bias there as well, because the early case description had to necessarily include the market. So there is a selection of cases towards that market. As I'm sure you're aware of, this is becoming more dubious, this link. One thing, though, is very clear that this market did provide some kind of amplification, and it was at this time when, at the end of December, on the 30th of December, when WHO was notified of this ongoing outbreak, there was this very, very strong link to the market. The origin is unclear, though. The virus was certainly circulating previously, but the links to the market have become much, much more murky. That doesn't mean they don't exist, it's just there is no data. Now, what do we know about these markets? So these markets, wet markets generally are absolutely unproblematic. Wet markets sell produce, they sell seafood, hence the name. Wet market is mostly related to the ice melting, and they're always wet and then they're being washed out, that's why they're wet. So they're very important function in providing food. Throughout Southeast Asia, throughout the world, you have markets, so-called wet markets in various kinds. But what's quite special in Southeast Asia is that on numerous wet markets, you have a wildlife trading section, and these can be huge. So we're talking about markets where, first of all, the animals are alive. This is quite unique. So we have lots of live species of wildlife that are being traded there. Sometimes 50 to 100 species of wildlife. This is a mix of legal and illegal trade. They come together. The big concern with wildlife, live animals, is that they are able to shed viruses, of course. Once you're dead, you can only shed in a very limited capacity. But if we have 50 species of wildlife stacked on top of each other, their holding conditions are obviously very, very poor. The animals are stressed. They're immune compromised through the situation. The shedding of viruses is massively enhanced. And then you add into the mix domestic livestock, like a lot of pig and poultry, and then, of course, 10,000s of consumers which visit these markets to buy products. Additionally, the wildlife is often slaughtered on site, so you also have exposure of inner organs, gastrointestinal tract, and of course the blood. And so this is pretty unique. You don't have markets anywhere else in the world on this scale, and most markets that deal in wildlife actually deal in dead product, and often the product is actually smoked and, in other words, also conserved, which greatly reduces its risk for transmitting disease. Now there's a signal. Unfortunately, the market was cleared out very, very quickly. So by the 1st or 2nd of January, that market had been completely cleared out. Animals were not sampled, at least to our knowledge, and there's no clear record of which species were there, where they were in the market exactly. There's no mapping. But after the market was cleared out, some 500 plus environmental samples were taken, and 33 of those samples were positive. And of the 33 positive samples, 31 of them originated from the western part of the market where the wildlife was housed. So there's a very strong signal to link this outbreak with the wildlife trade. It doesn't link it to the animals specifically, but it does link it to the cohort of people and wildlife in that part of the market. Unfortunately, as I said, at the moment, we don't know exactly the dynamics and the spillover, how this happened. One of the things that we do know, though, thankfully, based on previous research, is that the ancestral or evolutionary host of this virus comes from a horseshoe bat. And the reason we know that is because these animals and wildlife generally are being sampled through large international projects over the past 2-3 decades. And because the molecular techniques are so much better, very, very quickly we're able to get the full sequence. By before mid-January, already it was known that it was a coronavirus, and the full sequence was made available. And that one matched a sequence in a horseshoe bat species. And this is absolutely not surprising. This is something that had been expected and warned of repeatedly in the last decade. Just to put it in perspective, we've been working together with many partners through the past 2 decades, sampling wildlife across the world, and bats have been identified as hosting numerous viruses. The reason they do that is mostly because there are just a lot of different bat species. It's been shown recently that it's not so much the species per se or its immune system that is important, it's really just the abundance and diversity of these taxa. So there's a lot of bats, there's a lot of rodent species, and that's why those two harbor the most viruses. And to put it in perspective of how many viruses we're talking about here, if you look at the 25 high-risk viral families in birds and in mammals, it is estimated that there are about 1.7 million unknown viruses to date. And of those 1.7 million unknown viruses, it's estimated that about 700,000 of them have the potential to infect humans. So these are models, these are modeling exercises, and we all know that there's a lot of noise in there. But even if you got it wrong by an order of magnitude, we're talking about a lot of viruses, certainly more than you could address by proactively creating vaccines or something. So we need to deal with these spillover events. Basically, for each coronavirus that we know, there's thousands that we don't know, which are circulating in wildlife at the moment. And this is a completely natural effect. We, similarly to bats, we humans harbor viruses. They don't all cause any kind of disease, and we're able to transmit these viruses in spillover events as well. So as I said, one of the things we just don't know is how the spillover event happened. You see here on the right-hand side, you'll see some bats here. You'll see this one bat. This represents the MERS virus, the Middle East Respiratory Syndrome. It uses the dromedary camel as an intermediary host to infect humans, also a respiratory disease. This here represents SARS-1, the original SARS outbreak in 2002-2003. The origin is thought to have been in a bat. The bat has not been identified, but there's strong consensus that everyone agrees it comes from a bat, from the SARS coronavirus clade in horseshoe bats. In this case, a civet was the intermediary and amplification host, and at market interfaces infected humans. The civets are a species that is a wildlife species that is widely farmed in large numbers across China and Southeast Asia. And then this last bat up here is our horseshoe bat with the SARS coronavirus 2. And what we don't know is how it came from the bat to the humans. Certainly we know it's not in a bat soup, it's not in snakes, it didn't come from space. There's a lot of information out there which is, borders on conspiracy theories and so on. One species that's got a lot of traction recently is the pangolin, as you can see here on the left, but also the evidence is very weak at the moment. There is no good evidence to link the pangolin clearly link the pangolin to this outbreak. There are some similarities in viruses recovered in healthy and also in sick pangolins, but nothing at the moment. There's no evidence at the moment, so we just don't know, honestly. And so we need to assume, and I think the most parsimonious explanation at the moment is that we know it came from a bat, and there's two ways. It could have gone directly into humans, and we had a cryptic phase of evolution in humans no one noticed, and the virus acquired the necessary traits to transmit from human to human, or else an intermediary host at these market interfaces was used. And that's something most researchers believe is the most likely route at the moment. So the ongoing search. One of the things I do want to point out is that with this coronavirus outbreak, there's also been what's been termed as an infodemic. There are just absolute millions and millions of people talking about coronavirus constantly. The amount of misinformation out there is extraordinary, and the speed at which it can be shared really has made our work very, very difficult. And obviously also the politicization of this whole pandemic doesn't make it easier for scientists to inform the public. So basically what I want to get at, it's not about bat soup, it's not about civets or pangolins, but it's all about interfaces. It's about these interfaces that allow for spillover events to occur. And while there are a lot of interfaces, an interface where you bring together 50 to 100 wildlife species with domestic livestock and poultry, and you house them together under problematic conditions, and they're able to shed viruses. What we would really call this a cauldron of infection, basically. You really couldn't do it much better if you wanted new viruses to emerge, and then they also have the opportunity to spill over. So this is obviously one interface that we need to address. This seems to be is a completely unacceptable risk to move forwards. And as you know, the governments of China, And Vietnam is following suit, and several other governments are also calling for a permanent ban on the use of wildlife for consumption. Our own research has also shown that as the animals move along the food value chain, as the wildlife moves along the food value chain, and here this is a field rat from Southeast Asia, as it moves along the food value chain from the field where it's captured to the market interface and then finally to the restaurant or kitchen, it increases its positivity to coronaviruses in this case. So there's more and more coronaviruses as the animal moves. Basically, as you can see on the chart here, once it gets to the restaurant or kitchen, every second rat is positive for some coronavirus or other. So that's obviously concerning. For the end consumer that eats the rat in the cooked, as you can see in the bottom right, in the cooked stage, there's absolutely no risk because you've heated it and there's no risk. But for the staff that have handled this rat, processing it, there is obviously a significant risk. The other thing that needs to be found, there's a lot of discussion out there about halting the illegal wildlife trade. And it's really important to understand it's not at all about if it's legal or illegal, sustainable, non-sustainable, if it has a conservation impact or not. It's really about using wildlife. We have very robust monitoring systems in place for livestock and the consumption of livestock. We have spent thousands of years co-evolving with livestock and poultry and have these systems in place to monitor diseases. We have acquired diseases from livestock during the domestication process, and spillover events happen repeatedly. One of the most famous, probably, disease we've acquired from cattle is measles. Several hundred years ago, we made measles from a shared disease with cattle to a uniquely human disease. These are normal processes of co-evolving with species. With wildlife, we do not have these systems, monitoring systems in place. So if the porcupine is legal or illegal makes no difference. We know very little about the viruses that porcupines harbor. So this is why we need to really address this whole trade in wildlife, especially in live animals and in urban centers. So as I said, it's about interfaces, and you'll see this, you'll see a road here. Depending what field of work you're in, you'll see a road. We probably will all agree, probably it's some kind of logging road in some forest. And for many people, this road will go from the bottom of the image to the top, so from A to B. Now, looking at it from an epidemiology point of view, that road is a long, long interface. Right and left of that road is a forest. There are people using that forest that are hunting, trapping, and eating wildlife. And while they did that in the past, before the road came within their community, and probably had co-evolved with the species that they were living with, suddenly this road provides a trade route and exposure to urban centers and to movement of animals out of that forest into other areas. And remember, each of these pieces, each carcass that you're moving out in Africa, for example, you'll always have carcasses. No one's going to try to trade a live duiker in this animal. These animals will be, they're basically a suitcase full of pathogens as well. So as we are as well, it's completely natural. But with each carcass that you're moving, it's not only the meat and its nutritional value, you're also moving the pathogens with it. And I really want to make clear that the spillover to happen, so the switching of virus from one species to the other, is actually very, very difficult. It doesn't happen very often, and even less so does it have any kind of consequences. So as you can see, here's on the right hand side, you can see this is an image, a figure that Rainer Plowright put together once, which really clearly sort of highlights the fact that many, many barriers need to align for a virus to actually manage to spill over. So the point is, it's really a numbers game. The more opportunities you give a virus to spill over, the greater the chance is that at one time all those barriers are going to align and you will actually have a spillover event. Remember, a spillover into humans happens repeatedly. Very rarely does the virus cause disease, and much less likely will the virus be able to transmit from human to humans, and then particularly transmit very well, as in this case with the respiratory route. So thankfully that's a very rare event, but as I said, it's a numbers game. The more opportunities you get, the more often this is going to happen. And these interfaces are particularly important at these, what we call these edges of destruction. It's where we as humans are encroaching on the habitat, where we're displacing bats, where we're changing the ecosystems in which the pathogens are housed. In these habitats where we go collect wood, the first 1,000 yards, 2,000 yards in the forest, collect wood, return back into the villages, return to the road with some wildlife. Those are the main areas where we get spillover events. What's really important also to fully grasp is that nothing is remote anymore. If you sell a Gambian rat on the road in Northern Republic of Congo, you stand on the road and you sell it. It's a dead rat, but it's still dripping in blood. It's very fresh. You put it in your car. 12 hours later, you can be in Brazzaville. You can be in Kinshasa. Multi-millions people live in these cities. And if it's unlucky, you can take that rat, illegally of course, but you can be in Paris within another 12 hours. So the movement and the interconnectedness of the populations nowadays, and these remote communities that maybe have co-evolved with these pathogens, and urban centers is very, very close nowadays. So that's something we need to really be aware of, and this is how things have changed. So this idea that the diseases are occurring in some heart of darkness somewhere in the deep and have no effect on us, as we, in the 70s, Ebola was a disease which happened somewhere in Africa. That's it, But now it's potentially, it could, of course, travel very fast, as we've seen with the coronavirus, very fast to the rest of the world. So talking about all these, the markets and these impacts of the food system have, it's very important to point out as well that these urban markets in Southeast Asia and in China really are meeting the needs of the new middle class. The products, buying wildlife is expensive. It's a luxury item. Eating a pangolin is, first of all, it's completely illegal. It's been illegal for a long time. And it's a status symbol. It does not meet any nutritional needs. There is also very little cultural needs to eat a pangolin. This is quite a new occurrence, but it is definitely a status symbol. And that is in contrast to the millions and millions of people mostly in Central Africa, but also in part in Latin America, which are absolutely dependent on wildlife to meet their nutritional needs, to cover their protein needs and macronutrient needs. So it's very important that we balance our response, whereas on the one side, we may want to stop these markets, which are just from their sheer size and the fact that they're alive. But at the same time, we want to make sure that the rights and the needs of indigenous peoples and local communities that are at the moment dependent on wildlife are met. Basically, these big markets in Southeast Asia are draining the resources from the forests and draining resources from the local communities that actually need them. If you have a market for pangolins where the increase in price is 300 to 400% from the forest where it's captured, you're actually moving out of the forest these pangolins as one of the species, moving the pangolins across into China, for example, where they're then sold. So you have this big drain, and that really impacts the welfare and the nutrition needs of the indigenous people and local communities. So we've been working for a long time in the Northern Republic of Congo and other countries, of course, where we work with the communities. Mostly it's focused around Ebola virus and to make, there's an educational pillar to it where we talk about obviously not eating dead. If you find dead carcasses find carcasses in the forest, it's probably not the best source of food. There's certain dangers associated with that. And we now work with communities where basically we value the local knowledge a lot and pair that with modern molecular techniques to have this sort of a frontline diagnostic of potential Ebola outbreak or other viral outbreaks. The hunters will notify us. Everyone has cell phones nowadays, will notify us, and we will go to these front lines, sample the carcasses, and we can now work with very small smartphone-driven diagnostic machines, and we can, on carcass side, within 3/4 of an hour, actually make a diagnosis, yes, no, Ebola. And so this is a sort of frontline diagnostic system, early detection system, and it also supports the community because we can warn them if there is something there that's problematic. That's of course paired with the education campaigns and interacting with the WHO, Red Cross, and so on. Nowadays, just want to make that clear, nowadays if you have an Ebola outbreak in the region, there is no reason, and it's absolute standard, to keep eating bushmeat. There's no point. This spillover event is a singular event normally, and subsequent transmission is human to human, similar to this coronavirus, a singular or a clustered spillover event, and then subsequent human to human transmission. And in these areas where you have a spillover of Ebola virus, if you tried to ban the food, then you would bushmeat, you would then compound the effects of the virus by also nutritional stressful situations, because there is no alternatives in many of these areas. So there are obviously a lot of work going on providing alternatives in the larger urban centers, and paired with making sure that the needs are met for those communities that still rely on bushmeat. Just want to finish off one of the ways, just bring this to your attention, this sort of a One Health concept. One of the ways to address, and one of the necessary ways to address these kind of outbreaks as we're going through at the moment, being aware of the nutritional needs, the socio-cultural, socio-economic constraints, is a One Health approach. It's a holistic approach to health, tries to break down the silos, just clearly recognizes that the essential links between humans, livestock, wildlife, plants, and the ecosystem all are important and need to be addressed together, and taking note of the various drivers. Climate change, for example, is one that drives many of these processes, changes a lot of the dynamics of disease emergence. And so one of these things to be aware of is how we move forward from this outbreak and start to look at health and emergence of diseases in a much more holistic approach, sort of a One Health approach. Now, just as to finish off with the last slides, I use a very famous, one of the earliest maps, probably the earliest epidemiology maps from cholera outbreak in about 1850s in London. And this is a very famous map by John Snow, who was the first to determine by mapping the cases and saying, okay, we had this cluster of outbreaks and it was centered around this one water pump. We're going to close that water pump. And this was the first time that someone did a real epidemiological study and determined, okay, we'll close this pump and that's going to stop the outbreak. And in this case, they did. They famously removed the handle of the pump, and the cholera outbreak subsided in that area. And so what do we need to do in this case as it relates to this outbreak and this zoonotic disease? So WCS, Wildlife Conservation Society, has been pushing for a permanent ban on the commercial trade, and it's really centered on commercial trade in wildlife for consumption. So this is something we're pushing very hard. The Chinese government is in the process of doing exactly that by enshrining it in legislation. We're waiting for that, actually probably today or tomorrow, hopefully. And that needs to be paired with, obviously, with efforts to combat the trafficking of wild animals, because of, bringing in viruses from Africa into China and so on. That is all illegal already. So that needs to be strengthened, and recognizing these different species and enforcing the existing legislation. And then the third pillar is, of course, working on behavioral change to change these consumption behaviors. And this is really focused a lot on consumption in urban centers, and especially in large cities where there is no necessity. So thanks very much for your attention, and I'm happy to answer any questions as we move forward. Thank you.
Good morning, good afternoon, good evening, everyone. It's a pleasure to be here today to talk about the impacts on nutrition. As many have noted, the COVID-19 pandemic and related economic crisis and food system disturbances have all of the makings of a perfect storm for nutrition. We expect impacts on all forms of nutrition, from child wasting to stunting to micronutrient deficiencies, and to overweight and obesity in some contexts. This is likely to put us seriously off on our recent progress in improving nutrition. We are really concerned about going backwards and losing track of our very important progress. The next slide, please. The reason why this is happening is that the current crisis is affecting all of the systems that we are relying on for improving nutrition. We rely, of course, on the health system. The health system provides or delivers many of our essential nutrition actions. We rely on social protection systems that tackle poverty and are important for improving nutrition. And we also, of course, rely on the food system, which provides food, and on water and sanitation, which helps us protect from infectious diseases. So all of these systems are important. I will focus on 3 of them. I will not talk about water and sanitation just because of lack of time. Next slide, please. So in terms of the health systems, we all know by now that the disruptions are tremendous. There is a reallocation of staff in all of the countries to the issues related to the virus. And that also has led to the containing measures that have been taken by the government to reduce mobility and to prevent gathering. And so all of those factors have resulted in tremendous drops in coverage, at least for a short period of time, and for some for more extended periods of time, drops in the coverage of our essential nutrition actions. So this includes antenatal care, childbirth, neonatal care, immunization, micronutrient supplementation, nutrition counseling. All of those are affected, as well as the intervention to prevent and treat infectious and parasitic diseases, malaria, for example, a main killer for children, as well as severe acute malnutrition. In addition to that, we have informal evidence of stockouts of essential drugs and micronutrient supplements. And those, again, are very important. Iron, folate, multiple micronutrients, vitamin A supplements, all of those, including also the ready-to-use therapeutic foods that are necessary for the treatment of severe acute malnutrition. We also have important drops in demand for services. This is what we're hearing from the countries, that people are just scared to go to the health centers, and they avoid going even for childbirth. So we have an increase in births taking place at home, which we know has severe implications potentially for the mother and the child. Next slide, please. The economic crisis, many of you, if you attend IFPRI seminars, might have seen the modeling results from David Laborde's team, which show that the economic downturn is likely to lead to 48 million additional poor, an increase in 20% in 2020. And this is also likely to continue beyond this year. The other aspect that they have documented is the fact that it also is likely to reduce the consumption of nutrient-rich foods. You see on the right panel that the red bars are for drops in fruit and vegetable consumption and drops in meat consumption as well as dairy. And you see the yellow bars are increases in consumption of crops, of staple crops and non-perishable foods. So this is to be expected because in times of economic crises, we expect people to try to improve try to maintain their intake of calories, and they go for cheap sources of calories and at the expense of more expensive nutrient-rich foods. The next slide, please. Other and for the treatment of severe acute malnutrition. We also have important drops in demand for services. This is what we're hearing from the countries, that people are just scared to go to the health centers and they avoid going even for childbirth. So we have an increase in births taking place at home, which we know has severe implications potentially for the mother and the child. Next slide, please. The economic crisis, many of you, if you attend IFPRI seminars, might have seen the modeling results from David Laborde's team, which show that the economic downturn is likely to lead to 48 million additional poor, an increase in 20% in 2020. And this is also likely to continue beyond this year. The other aspect that they have documented is the fact that it also is likely to reduce the consumption of nutrient-rich foods. You see on the right panel that the red bars are for drops in fruit and vegetable consumption and drops in meat consumption as well as dairy. And you see the yellow bars are increases in consumption of crops, of staple crops and non-perishable foods. So this is to be expected because in times of economic crises, we expect people to try to improve try to maintain their intake of calories, and they go for cheap sources of calories and at the expense of more expensive nutrient-rich foods. The next slide, please. Other information, very, very little so far, real information from the field that comes from some of the phone surveys that have been conducted. The first one is in April, the Bangladesh Institute for Governance and Development reported a few data on how people are reacting to the economic crisis or are suffering from the economic crisis. They report a 70% drop in income, which is larger in urban slums compared to rural areas, for example, and about 25% drop in food expenditure at the time in April already, 28% in urban slums, slightly higher, and a dangerous increase in coping strategies such as reducing the number of meals per day and reducing food consumption altogether. We also have some early evidence also from phone surveys from our team, IFPRI team in Ethiopia, that showed disruptions in food supply chains and affecting both demand and supply, or resulting from both demand and supply. So in Addis, for example, the interviewees reported, 50% of them reported avoiding dairy, meat, and poultry, and 22% report limiting vegetables. This is purely due to perceptions, misinformation. People believe that animal source foods and fresh vegetables are contaminated and are a risk of acquiring the virus. And so they avoid these foods. There's also information about the vegetable value chain in Ethiopia that is affected both, again, in terms of reduced demand from individuals who are scared of contamination, but also from the restaurants that are closed. And there are several supply side limitations, scarce labor, inputs, shortages, high prices, and waste in these kinds of products. So we have evidence of reduced consumption of nutritious food, and we have evidence, very preliminary evidence of deterioration of diet quality. And so we are very concerned about impacts on micronutrients. The next slide, please. In terms of social protection systems, social protection programs exist in many, many countries. And Ugo Gentilini from the World Bank has been tracking what has happened to these programs during the crisis. And we have 170 countries that have planned, introduced, or adapted social protection programs, mostly cash and in-kind transfers. We also know that those are very unlikely to be able to meet all of the demand, which is very large and will continue to increase probably. School feeding programs are part of social protection systems, and we have seen with the school closures that up to 370 million children are without school meals. 45 countries have implemented take-home school meals. 10 countries have been implemented cash-based transfers, and 15 have other various modalities. But here the concern is again about the quality of meals. What can be delivered safely in a take-home school meal? And we are concerned that nutrient-rich foods will be scarce, and that this will affect diversity and the micronutrient content of those meals. And again, a concern for micronutrient malnutrition. The next slide, please. In conclusion, I wanted to put up a series of recommendations, which are really, I would call them big buckets of recommendations targeted to all of those systems that we rely on. So first, of course, keep the agro-food systems functioning. We've heard that, we hear that consistently from economists. We need to keep the food systems functioning, including the informal sector. We need to help protect the informal sector, provide them water and sanitation and education on how to do their trade in safer ways, facilitate food systems innovations. Another area is to support the local production, especially of fruit and vegetables and eggs, even homestead food production where possible, so that we improve access to those nutrient-rich foods. Find innovative ways to simulate demand for nutrient-rich foods. Make sure we address misinformation. I was very, very surprised to see all of this misinformation about animal source foods and vegetables in Ethiopia. Use new techniques and tools to reach people with messaging, with phone messaging and other radio, etc. Use social safety net programs to improve diet quality, not just quantity. Invest in water and sanitation. I have not, as I said, mentioned much about this sector, but we know that water and sanitation are incredibly important for stopping the transmission of this disease. And we also know that in low and middle income countries, the situation in water and sanitation in urban areas are tremendously bad. And so that will have a lot of consequences if we don't address it. And finally, we need to address vulnerabilities and inequalities. The crisis is exacerbating all of the inequalities in age, gender, for refugees, for poor, non-educated people. And if we do not address these inequities, the whole crisis will be even worse for the vulnerable groups. Thank you.
Morning from Seattle. My name is Megan Parker. I am a Senior Nutrition Research Officer on PATH's Nutrition Innovation Team. And I'm going to talk to you today about the work of PATH and some of our collaborators. specifically next generation tools to track malnutrition and improve resilience in the context of the coronavirus and climate change. At PATH, we've had a long history of innovating and working with the private sector to solve health problems using diagnostics, drugs, vaccines, and health system service innovations. Now, in the context of COVID-19 and climate change, I want to talk about a few innovations that have relevance as we think about solutions in a multi-sectoral way. Right now, many families are struggling with reduced incomes and limited mobility. Food supply chains have been disrupted and access to markets and fresh foods has decreased. Schools have closed, so many children don't have access to their normal social safety net programs. And this is troubling, especially because some of the significant reductions we've made in the last 2 decades could be lost. Additionally, during this pandemic, utilization of essential health services could be decreased. A recent article in The Lancet estimated over 250,000 additional child deaths could be expected from only a 15% reduction of routine services in 6 months. It's our goal to strengthen people-centered health systems and help communities be resilient. This crisis has produced an opportunity to stop and plan for more resilient futures instead of reinstating the status quo. Over the last 5 years, PATH has been working in the nutrition and climate change space and collaborating with innovative technology groups, one of which developed a practical solution to evaluate child nutritional status at a distance. We need to be able to safely track malnutrition during pandemics when rates will likely rise, even though communities do not want to be in close contact with data collectors and hospital workers. We need this information so we can better target nutrition interventions. So I want to highlight some of these practical technologies and solutions for a changing world, but this is not an exhaustive list. So first there's the AutoAnthro technology, and it was developed by Body Surface Translation. And this tool was developed to automate infant and child anthropometry measurements. It captures 3D scans of children and then develops a model from which a variety of measurements can be extracted, like height and length measurements, middle upper arm circumference, and also head circumference for kids under 5. It uses regular smartphones and 3D cameras. It captures data in short scan bursts, and it's able to minimize movement artifacts caused by squirming children. It operates on both iOS and Android platforms. It was originally evaluated on an iOS platform by Emory University within a US population, and that data has been published. And more recently by the CDC in China, Guatemala, and Kenya. Further Android validations are set to begin in 2020. The results that were published by Emory demonstrate that the AutoAnthro tool has the potential to be less invasive, easier to use, more accurate, consistent, and more efficient for large-scale data collection than current manual approaches. Once it's fully validated, it could allow better monitoring for child growth in national nutrition surveys and enhance local growth monitoring and promotion efforts within national nutrition programs. It could easily be used by frontline health workers or nutrition survey teams. So you can see here how the tool works. It takes a raw scan, it estimates the posture, and then it generates a wireframe model from which child measurements are derived. Right now, there are 4 primary challenges to traditional anthropometry, which AutoAnthro is able to address. So first, you have inaccuracy due to poor training and fatigue during data collection, but this tool offers a consistent computer process for heightened accuracy. Next, sometimes you have missing or incomplete digital record capture, but this tool, it immediately digitizes the results, so there's no data entry. Third, you can have uncooperative children, but this tool is twice as fast, and it doesn't require pinning kids down to a length board. So on the Android platform, it only requires 5 scans now. Fourth, it has, like, normally we use heavy, bulky equipment, but this is a simple tablet and a camera rather than a large, heavy shoreboard to cart around. So now, in the time of COVID, it's even more relevant because this process can happen at a respectable social distance of 1 to 2 meters apart. In 2017, UNICEF developed a target product profile for digital health length measurement devices, and this tool meets that criteria. On average, the AutoAnthro tool takes half the time of manual anthropometry and only a few days to train. the data collectors. If anyone has any questions about this tool, we have asked Peter Miller and Jean Alexander from BST to be on the line to respond to your technical questions. Next, we have food fortification. Now, globally, micronutrient deficiencies present a widespread public health concern, particularly among vulnerable populations in low and middle income countries. Deficiencies of essential micronutrients like iron, zinc, vitamin A, and B vitamins can lead to impaired cognition, immune status, and growth and development, with significant and lasting effects for the socioeconomic development of both individuals and nations. As the world's population continues to grow, we need to be smart about what we're growing, choosing both micronutrient-dense crops and resilient breeds, because current food systems are increasingly stressed by climatic extremes. So first, there's biofortification by Harvest Plus. Biofortification of staple crops is an effective option to reach rural communities across Africa and South Asia. Harvest Plus was able to use conventional crop breeding techniques and local seed banks to increase the iron, zinc, and vitamin A content of certain staple foods grown and consumed by smallholder farmers. Today, we have over 340 varieties of 11 biofortified crops, including iron beans, vitamin A cassava, and zinc maize, and they've been officially released in over 40 countries with funding from Harvest Plus and the International Potato Center. And thousands of varietal lines are currently in testing. This is a localized nutrition solution that's less vulnerable to value chain disruptions. The smallholder farmer owns the solution because it's growing in their fields, and they only have to buy the seeds one time as a single investment. Now, biofortified crops piggyback on crop varieties that are bred for more desirable attributes, which include resistance to climate change effects such as tolerance to heat, drought, and flooding. Over 50 million people are already growing and eating these biofortified crops. Clinical studies have shown that significant improvements in child diarrhea, pneumonia, iron deficiency, and night blindness. With climate change comes rising CO2 levels, and higher levels of CO2 cause plants to lose their nutritional value, but biofortified crops have the ability to mitigate this risk. Also, with the COVID pandemic, we see that people are spending less on food, and they have less access to micronutrient-dense perishable foods, but biofortified crops are a way to deliver micronutrients through staple foods. Similar to biofortified crops, we have another strategy to fortify staple foods. This is done post-harvest. PATH has been working on fortified rice for over 30 years, and this technology was developed here in Washington State. Now, today, we work with governments to develop fortification guidelines. We license local businesses to produce fortified kernels through a tech transfer process, and we ensure a sustainable business model that can then feed into the social safety net programs or be sold in the private sector. One of the benefits of fortified rice is that it repurposes broken grains, which might otherwise be discarded, and it grinds them down into a flour. And then you add micronutrients to this flour and you push it through an extruder or a pasta machine. And then those fortified kernels are blended back into regular rice at a 1% or 2% ratio. In India, we've already licensed 10 kernel producers in 7 states, and we have another 8 kernel producers that will be licensed soon. We've conducted multiple clinical trials in Southeast Asia and Africa to examine efficacy and effectiveness, and we set a precedent for school feeding programs in Gujarat, Karnataka, and Odisha by conducting clinical trials and setting up the centralized kitchen infrastructure that then is needed for governments to scale up mass fortification across the state. In Karnataka, we started by feeding 2,600 schools, and then the government scaled up to the rest of the state. Whatever your setting is, the micronutrient formula can be tailored to your population and their health needs. Typically, we target anemia with iron, vitamin A, and B vitamins, sometimes zinc. We have worked to improve the formulation with Abbott Laboratories and Isotope Lab, the Isotope Lab at ETH Zurich. The multiple micronutrient formulation, it significantly increases hemoglobin, reduces anemia, and improves cognitive test scores. The World Food Program now provides fortified rice through their school feeding programs across Southeast Asia. Micronutrient fortification of staple foods is an affordable and targeted tool that can be programmed. Okay, alternative proteins. Over the past year, we've conducted 2 alternative protein landscape analyses for a donor that's interested in future food systems. And now, in the time of COVID-19, with food system disruptions and meat production system disruptions, requests have been made to Congress to support alternative protein research to ensure future food systems are more resilient. So first, we have cultured proteins, and you've likely heard of cultured milk and eggs, maybe meatless meat or finless fish. These are environmentally friendly vegan products. They're animal source food proteins made without any animals. The cultured protein market is at a pivotal phase with products about to launch next year. And the manufacturing companies have only just formed within the last 5 years. Several attributes make cultured proteins suitable for low and middle income countries, like longer shelf life. These products are more like protein fortificants, and they can be added as ingredients to new foods or food rations or micronutrient powders. They provide an animal source protein without the environmental burden of traditional livestock. We think of them as an also, but not a replacement, as there's many bioactive factors in milk and eggs. And these products are only now just rolling out across the US and the EU, where policy regulatory environments are stronger. This is not a short-term fix, and there's still some issues to work out. But we think that these products will grow the market. In the short term, there will be little to no effect until the policy regulatory environment is figured out. Then next, we have plant protein isolates. We recently reviewed 26 plant food crops that were all very well researched and had near complete data. We examined their amino acid profiles, their digestibility, and their PDCAAS scores. We identified soybean, quinoa, chickpea, duckweed, spirulina, and potato as having adequate concentrations of essential amino acids per gram of protein with respect to infant and young child requirements. We examined all the 9 essential amino acids, but at minimum, these plants had to provide an adequate amount of lysine and methionine. We've never considered duckweed or potato as being valuable protein sources, so this market landscape really brought new contenders to the table. We had a longer list, but after examining sustainability, organoleptic properties, ingredient versatility, and efficacy research, we narrowed down our pre-selection of 9 to just those 6. Then third, we have edible insects. Edible insects are mini livestock, and they can be easily microfarmed by landless farmers or their young children. They produce less greenhouse gas emissions, they require no land, and they require minimal amounts of water, feed, and energy. They are environmentally friendly in comparison to traditional livestock. Pat has been working on edible insects for over 5 years with Fire Food Group, Heifer International, University of Ghana, and Tech University in Kumasi. We conducted focused ethnographic research with Gretel Pelto to understand mothers' value systems when choosing infant and young child foods and how to go about developing new foods. We studied the adequacy of insect proteins, the ability to increase iron and zinc content by testing new feeds and pairing with the beer industry to test peto mash, the food waste products. We worked with local food technologists who then developed insect-fortified foods. And we equipped 50 young mothers to microfarm this mini livestock in the Ashanti region as an income generation project. This was a really fun project, and we hope to expand our edible insect work to new geographies with the Wildlife Conservation Society soon. So I hope we've been able to introduce you to some new concepts to think about going forward that have relevance both during the time of COVID-19 and also climate change. Thank you for listening, and we're happy to answer any questions you might have. Thank you.
Well, hello everyone. I think we have our panel now up, and I'm seeing my colleagues Marie and Madhu and Dr. Walzer and now Dr. Megan Parker. So this is the part of the presentation where we go through your questions that came in during the presentation. So first, let me introduce you to my co-facilitator, Madhu Maboya, who is going to be helping us with getting some of the questions in. And then, of course, our 3 panelists are here as well. So thank you all for those great presentations. I think it's a really interesting way to look at food systems, both from a nutrition perspective and a health perspective, but also increasingly this link between emerging infectious diseases and then some of the new or mostly new technologies that might help us deal with the implications as we move forward post-COVID. So, Madhu, do you want to start with the first question?
Certainly, Catherine. Thank you. Just a quick introduction of myself. Hello, everybody. My name is Mdudu Zimbuya. I'm a senior technical specialist with the Global Alliance for Improved Nutrition. I'm assisting Catherine in co-moderating this session, and would like to take this opportunity as well to thank you all for logging on and thank the presenters for these splendid presentations. So the first question that that we have is for Chris, and the viewer asks, what efforts are being made to reduce the demand for wild meat while preserving the needs of rural communities? So could you maybe comment on this, Chris?
Yeah, thanks very much for the question. That's a very good question. And it's very context specific. It really depends on which areas we're speaking on. So for example, in Southeast Asia and in China at the moment, a lot of work is being put into new legislation, as the countries have already announced that they're going to be banning the use of wild meat and wildlife for consumption. So we're supporting them a lot on drafting new legislation, putting new regulations in place, and so on. Quite in the contrast, in an Central African context, of course, it's really much more focused on securing the needs of indigenous peoples and local communities, making sure they have access to high quality bushmeat and products from the forest. So most of the work is focused on education and changing consumption in the ever increasing urban centers where the bushmeat is not necessary. So, you know, the cities that had 3,000 inhabitants now have 30,000, and they're still eating, 85% of people are still eating bushmeat. So it's very context specific. I hope that answers the question.
Great, thank you. We have a next question, and this one is for Marie. Marie, you're a world expert on nutrition. To what extent do you think the nutrition community is aware of the issues around zoonotics and emerging infectious diseases as part of their thinking about food systems and nutrition? And what do you think the implications are post-COVID-19? And Marie, I think you're on mute.
Yes, I am. I'm just trying to understand the different aspects of the question. First of all, most nutritionists, if I may say so, are concerned about the health crisis right now. This is the main channel, as I mentioned in the presentation, for us to deliver our nutrition interventions. And we have preliminary assessments of the impact of the closures on the preventive health services, for example, even for a short period of time, the shutdown, as well as the reaction of people after to not attend the services are really likely to lead to a lot more deaths, especially for mothers related to delivery and to the newborn child as well. And there's also a possible really large problem related to wasting in the short term and probably stunting in the longer term, which is there due to a combination of both the health services not being available to prevent these manifestations of malnutrition, but more importantly, the economic breakdown. The economic downturn is there. It will get worse probably, and it will last for a really long time. So we are really worried about and in many countries in Africa, for example, people are all saying, we will not die of COVID, we will die of malnutrition and hunger. And that's a real concern for us because, as I mentioned briefly in the presentation, the social protection programs that are put in place and the efforts to distribute food or cash are just not going to cut it. The demand is so great with 70% of the people having lost their income and having very little to eat and avoiding animal source foods, we really need to make sure that this demand goes up again, that people realize that this is not a source of transmission and they need those products and the vegetables as well for a healthy diet. So one thing that people can actually do for food systems is to keep the demand up. I mean, the individuals, the households need to keep the demand up so that it also helps the supply side. As I mentioned, the early supply side effects were much more a reaction related to the reaction of the individuals in terms of demand than to the actual crisis itself. So it's a complex system, but wherever we look, we see that it will take quite a bit of education and correction of all the misinformation to push that demand up. And there are, of course, all the issues related to international trade as well that limit the inputs from the supply side, and that results in changes in prices. So this is a really complex crisis, and we have no practice on how to approach it and what to do. And I think we're still struggling, even sitting here in our offices at home, we're really struggling with how to support the developing countries where we work and support the people that are partners there. So it's really a very loaded question and a difficult answer.
Great, thank you. Dr. Walser, would you like to add anything to that?
No, not particularly. I was sorry. That was a very complete answer, and I was listening. And thank you.
Great.
Great, thank you, Marie, for that. The next question is for Megan. Could you talk a little bit about the technical process for producing cultured proteins? So what does it entail, and what's the process through which these proteins might be added to food rations, and what evidence is needed also to inform the introduction of these products? Over. Hello, Megan, I think you're on mute.
Am I still, I'm off mute?
Yes, I can hear you, thank you.
Okay. So the process by which cultured proteins are made, it's a proprietary, it's a fairly secret process at the moment. They do use a fermentation process that then incorporates certain ingredients like yeast, sugars, dairy flora, bacterial catalysts, and I think I had listed in the presentation the variety of different products that will be available. Perfect Day has just produced a milk protein that came onto the market. They just received GRAS status. Typically, you have to first obtain GRAS status to show it is safe, and then these products should be going through efficacy effectiveness studies to show the impact of the benefits, and then we would have to do shelf life studies, and then, sorry. There are strict regulatory environments that these, proteins would have to go through, and if they are made in large enough quantities, then they could be affordable for low and middle income countries. Yeah, and then you'd also have to go through USDA and have, after safety and efficacy is proven, then you could apply to have them put on a food commodity list. Maybe this could happen by 2035 or 2050.
Great, thank you. So Dr. Walser, this next question is for you, and it's a very long and detailed question, so I'm going to sort of paraphrase and summarize it. The question really at its core is there any reason to believe that simply the possibility of accidental release from a research lab could not be considered? And this person clearly has a great deal of knowledge about the biology of it, but indicates that the NIH has been collaborating with one of the labs in Wuhan to explore the gain of function research in viruses, and where gain of function is understood to be an attempt to understand how a virus can become more infectious or deadly. Given that this type of research took place in the Wuhan area, and the nucleotide sequence is unlikely to have arisen by mutation process commonly observed in cirruses, and given that the accidents of the contaminator or release have been observed at virology labs around the world, is it possible that it could have been released?
Yeah, unfortunately, this is a question I obviously get a lot. And one of the things we have to be very clear at the moment, we just don't know. Yeah, but I want to be absolutely clear. There is no evidence at this point in time to show that this virus was released from a lab. That would be one of the more really surprising routes for a virus to have spilled over. What's been shown very clearly, there's a paper came out on last Friday, from Science that did a very nice collaboration between a large group of scientists and they showed very nicely how the most likely way that this virus came about was through recombinations between various existing and previously documented coronaviruses. Obviously, this is based on the ancestral bat, and then some of the cleavage sites and receptor binding domains which have been seen in pangolins, for example, but possibly also in other species. They also bring up that possibly a third species or another one was involved as well. The problem is we just don't have the samples, and the market was cleaned out very quickly, and we neither know which animals were on the market on those days. We don't know anything about the status of these individual animals. They obviously weren't sampled or not adequately sampled. And all we have are the so-called environmental samples. But even that, we know very little about the type of environmental samples that were taken. We know that the virus was circulating already previously in the human populations. We know that. So it's very difficult to find out where was the actual spillover event, when did it happen in time, and what was involved. One cannot exclude the lab theory, but there's absolutely no evidence. It seems highly unlikely as well in view of the fact how these labs actually work. This is a misconception that nowadays people work with live viruses in these labs. It is possible that a pathogen could have happened in a lab, but basically now you work mostly with computer simulations because it's just too much effort to work with a live virus. Over.
Thank you, Chris. The next question is for Marie, and I'll combine the two questions that were asked. Is there movement towards a more plant-based diet in populations who now eat primarily meat or wild meat in some instances? And also, is there any movement towards encouraging vegetarian diets with pulses among these communities instead of depending on mostly animals for protein? Over.
I'm not sure it's a question for me. Are you sure? Because I don't think this comes from, I mean, the populations I'm talking about are populations that really need a little bit of meat in their food supply, just because they have very little access to fortified foods. And so we're certainly not going for recommending vegetarian diets. They already know how to do that. They already eat pulses and cereals, and not enough nuts probably, but so they do have a way to get even complete protein from the right combinations. But what we're worried about is the fact that because of the scare, they really do not eat any of the animal source foods, including dairy. I think I didn't mention dairy, but even dairy is especially raw milk, is considered dangerous. And so they're not getting the highly bioavailable micronutrients that are needed. So there's no intent to make these populations think of a vegetarian diet. On the contrary, they have to continue to be, the promotion has to be for small amounts, and then eggs are important for young children, as we know, or a good way for young children to get some of the bioavailable micronutrients. So I think that maybe the questions were related to, well, given all of the transmission that is happening between animals and humans, should we find ways of going for vegetarian diets, or what should we recommend vegetarian diets for those kinds of populations?
Thanks for that, Marie. I think context is really important, and I think the question was probably triggered by the balance in risks and benefits. Over, Catherine.
Thank you. Yeah, our next question, we received a number of questions related to the autoanthro technology. So maybe I'll throw out a few of them here, Megan, if you can maybe answer some of them. One is around, is it currently available for commercial use? And if so, how would someone be able to find it or buy it? And then also, are there any estimates regarding costs in comparison to current methods for anthropometric measurement? And then another question around its ability to use the tool in adults. And also, is it able to take a weight estimate? So why don't we start with those?
Sure. Okay, thanks. So the tool is available now on a limited basis for validation and testing. And it will be commercially available by 2021. The cost of the device, well, it's dependent on the customer and the application, but it does need to cover the cost of the phone and the 3D scanner. So for the iOS platform, that is an Apple, that's a tablet. And for the Android platform, that is a Samsung 8 Galaxy 8. And then there's also the 3D scanner. I would encourage you to reach out to BST to ask questions, and there was an email on the slides. Regarding adults, it can be used with adults in the future, but BSTs would still have to conduct validation studies before it could be officially incorporated into the final tool. Regarding weight, there's been quite a bit of research on this. It's not yet able to capture weight, but this is in the process, and it is on their development timeline, so it should be potential in the future.
Great, thank you. Over to you.
Thank you. There are several questions for Megan on the edible insect aspect of the presentation, so I'll follow up with on that, Megan, while you have the mic. So first, thank you for the presentation. So what species of edible insects did you study? Africa has a wide range of edible insects, and these may also serve as disease vectors. So have you considered the issue of detoxification before use? Over.
Right, okay, thanks. So we primarily worked with Aspire Food Group in Ghana, and they specialize there in the palm weevil larva. We were not worried about the pest system. These are larvae that really, can move. And we farm them in a system, just using wood. They burrow into the pith to lay the eggs, cocoon, and become a larva. We put it in there. You put a screen over it. It's it's easily secured. So yeah, we're not worried about it becoming a pest. We mostly just worked with the palm weevil larva there in Ghana, though we had gone to Thailand to study cricket farming and the methods, and there also they use the screens over to, with elastic bands to make sure that nothing gets loose. But, Christian might have something more to say on potential of, disease being carried through insects. Similar to that.
Thank you, Megan. I think there was a question, so I might as well just read it out for you, Chris, that should we have the same concern while promoting insect protein or other novel proteins?
Yeah, no, I think as Megan would point out, this is something we really, I'd never say never, some things, also insects and invertebrates have a lot of viruses, but they're so taxonomically so far distance from us humans, that the entry into a human cell would be really extraordinary. So this is not a concern at all. And actually, similarly, WCS is investigating using invertebrate protein source to raise chickens, to produce the eggs, and so on and so forth. So I think this is really an interesting way forward. And while we do need to be concerned about spillover events from other animals, invertebrates seem like a very good solution moving forward. That being said, diseases of invertebrates that kill invertebrates are very, very important to be on top of when you're raising invertebrates. That's something we need to really consider.
Thank you both for that. Catherine?
Great. So this question is for Marie, and Marie, it's again sort of a series of questions, but they're sort of asking, what are the data needs at this time, and what are the opportunities for obtaining such data? Are programs pivoting to meet the downstream consequences of the pandemic, perhaps deviating from original program design?
I missed the last part of the question. There was some breakup in the line.
Sure.
Programs or something?
Yeah, so they're asking, are programs pivoting to meet the downstream consequences of the pandemic, perhaps deviating from original program design?
Mm-hmm. Okay, yeah. First of all, in terms of data needs, this is really a good question for my team and the kind of work we do. We had to close 25 research projects in the field because of the lockdown and et cetera, and the inability to travel. We're all avoiding, unfortunately, presence in the field, and we are concerned that this may last for a long time. So our research is directly targeting programs and understanding what works and what doesn't work and how it works. And so this is devastating to not even see the light about when we're going to be able to go back to the field. Some of our partners on the data collection side are eager to start again, but we are not. We are very concerned that even local partners are saying, well, you know, the WHO declares, for example, that this country is at level 4 of contamination, therefore we should really not do any field work, but people feel that inside the country is reopened and we should just go ahead and do this research. We are doing some research through phone surveys. I mentioned some of the results from that, and this is really not providing the same quality of data. So it is a concern. If we're going to have to stick to phone surveys for a long time, we will never have the level of quality that we've had before, in the sense that we can last for maybe 15, 20 minutes on phone surveys. We can't entertain a 2-hour interview with people. We can't do, therefore, full dietary recalls or full consumption modules to understand the impact on people's food expenditure, but also budgets overall, or dietary needs, gaps. You know, this is really a very difficult time for that. In terms of programming, I think at this point we work, the nutrition community works with the UN institutions to try to have a call for action in terms of protecting nutrition in these very hard times. We have WHO, WFP, FAO, and WHO that we've been able to bring together and try to see how they can maybe modify the way they do deliver services. In India, for example, they have distributed a large number of cell phones to the health workers so that the health workers actually can do some of their work. The community health worker can actually do some of their work through phone, even including behavior change counseling to the mothers in the communities, or sending reminders for mothers to attend their antenatal services, etc. So there is, that's hard to manage a phone call at the minute. So anyway, all that to say that what we are trying to get at is that people start to think of innovation in how they deliver services that respect as much as possible some of the social distancing that is necessary, and that also makes people feel safe to go and be part of the services that are actually offered.
Great. And with that, I think we are going to wrap up this session. I'd like to thank all of our panelists for really interesting presentations, very diverse set, but I think very a great way to sort of think about what we're doing now, how we think about food systems in a different way, and then how we're going to start to collect data that might be easier for people, and maybe in the context of having a more distance kind of data collection methodology. So with that, thank you so much. And do you want to say any last words?
No, just to echo the word of gratitude to the panelists for their great presentations and responses to the questions from participants. And thank you all for listening in or tuning in from wherever you are in the world. Thank you.
Thank you. Bye-bye.
Thanks, everyone. Bye-bye.
Hi there, this is Dr. Dave Heber, and I'm very happy to welcome you to the Herbalife Nutrition Institute Satellite Symposium of ASN 2020. The title of today's symposium is The Global Nutrition Transition, Protein as an Anti-Aging Strategy. My disclosures are here. I receive honoraria and travel reimbursement from the Herbalife Nutrition Institute and Herbalife International. My title at the university is for identification purposes only, as the University of California does not endorse specific products or services as a matter of policy. The mission of the Herbalife Nutrition Institute is to encourage and support research and education on the relationship between good health, balanced nutrition, and a healthy, active lifestyle. This is the 9th of the HNI-sponsored satellite symposium at the annual meetings of the American Society for Nutrition. As you can see, they started in 2011. What is the global nutrition transition, and why do we use that title? In the same countries where only malnutrition was once found, there is now obesity and its associated disease conditions. The appearance of obesity in these locales globally has been called the nutrition transition. What underlies this transition are changes in diet resulting from the introduction of Western foods in place of traditional diets. There has been a shift in dietary proteins, carbohydrates, and fats as a result, and these changes, along with sedentary lifestyle, have been linked to obesity and age-related chronic diseases. An example of the global nutrition transition is shown in this slide from Peru that was a image from 2010. The yellow areas for most of the country are stunted children and obese adults, since children who become adults with a lower lean body mass are more susceptible to obesity. The green transitional area with stunted children and few obese adults, and the red areas are the more high socioeconomic status areas of Peru where you have both obese children and obese adults. And this pattern repeats itself through many countries around the world, and we can clearly see that obesity is a global epidemic. Today's talk is going to be on protein as an anti-aging strategy, and we're going to spend a lot of time looking at the variable aging of muscle throughout adult life. As you can see in the upper left-hand corner of the slide, in early life, we're increasing our muscle mass. In adult life, the decrease in muscle mass depends on the level of physical activity. And in older life, there's an intrinsic aging of muscle mass related to some changes that have been described in the mitochondria. On the lower part of the slide, you can see healthy aged muscle versus unhealthy aged muscle with an accumulation of damaged mitochondria leaking oxygen radicals. This is believed to be an intrinsic method of aging, which may be reversed by some of the methods that we're going to talk about today. So aging processes can be classified as intrinsic aging or extrinsic aging. The rate of aging varies among individuals, and inflammaging affected by overweight, obesity, and diet has been identified as a potential modifier of the rate of aging. Protein impacts protein synthesis in muscle cells, and protein supplementation with exercise may be able to reduce the rate of muscle loss and the development of sarcopenia-associated metabolic diseases. New methods for assessing body composition, skeletal muscle mass, and protein quality can point the way to improved muscle, protein nutrition, and exercise to reduce the rate of muscle aging. Today's agenda, following this introductory talk, will start off with Dr. Stephen Heimsfield, professor at the Pennington Biomedical Research Center, Louisiana State University, who's going to talk to us about methods for evaluating body composition and the application of new technologies such as 3D optical imaging, fMRI, and PET to the study of human metabolism. Thank you.
Good morning. This is Steve Heimsfield. I'm a professor at Pennington Biomedical Research Center, and I'm going to give an overview of methods of evaluating body composition for studying human energy metabolism and protein metabolism. First, I have two disclosures. I'm on the Tanita Corporation and Medifast medical advisory boards. Now, if we think about body composition, we can begin with the most rudimentary form, that is body size and shape. That's a function of height, adiposity, age, race, genes, diet, activity, hormones, drugs, and many other factors. That's just our fundamental shape. And if we go from there, we can establish that shape is unique to each individual. It's just like a fingerprint or a retinal photograph. I'm going to come back to that in a minute. And in turn, our shape, that unique shape each one of us has, is related to our composition in terms of muscle, bone, and other components like that. And finally, our body composition then relates to our health risks, our athletic performance, energy expenditure, and metabolism. And this is the paradigm we're going to focus on. We're going to go through some of the ways of quantifying each of these components. Now, if we think about body composition as a whole, we can view it as 5 levels ranging from the most basic atoms all the way up to the whole body, and in between molecular, cellular, and tissue organ components of the body. This is the so-called 5-level model of body composition. And by far, the most common way in the clinical and research setting that people quantify it is the simplest model, the so-called 2-compartment model, in which weight is a function of body fat and fat-free mass. Nevertheless, that 2-compartment model relates to components at the atomic level all the way up to the whole body level. And I'll review some of the methods we then use to quantify these different components. At the most basic level, as Dr. Heber mentioned, protein literature, of course, is very interesting and important, both clinical and research settings. And Stanton Cohn was the first, among others, to develop a technique called neutron activation analysis, in which the body elements can be quantified, carbon, nitrogen, hydrogen, oxygen, and minerals, using a technique in which subjects are exposed to a radioactive source, and then the decay in key elements are then quantified using a whole body counter. And from that, Cohn and others were able to develop a model of the body in which they quantified protein, both muscle and non-muscle protein, bone, water, and fat. This is a key method that was the foundation of many other methods that followed, and is the gold standard for quantifying total body protein. It was not too after Cohn's work that Richard Mazes developed a technique known as dual photon absorptiometry, or DPA, in which the body is quantified using a gadolinium radioactive source, and it was mainly for bone. And this illustration shows Mazes' actual DPA scan back 40, 50 years ago, in which he was able to divide the body into fat and fat-free mass, and the fat-free mass part consisting of lean soft tissues and bone minerals. This was a benchmark technique, and I'll come back to it in a minute. Around the same time, Godfrey Hounsfield first introduced the technique of computerized axial tomography, now known mainly as CT, computerized tomography. And with CT, he was able to quantify not just the molecular level of body composition, but the tissues, adipose tissue, skeletal muscle, and bone. And with the CT scan, then you can separate out these different compartments. This illustration just showing muscle and adipose tissue, but you can also quantify visceral adipose tissue, ectopic fat deposits, and you can quantify, as shown in this little panel here, the attenuation of the liver measured in Hounsfield units now can be used to quantify liver fat. So CT was a revolutionary method for the study of body composition and metabolism. Well, not too long ago, then Mazes's dual photon absorptiometry technique gave way to dual energy X-ray absorptiometry, now known as DEXA, in which the radioactive source was replaced by an X-ray tube, and the X-ray then provided two different energy levels, dual energies. And as you can see, a modern DEXA scanner can give quite beautiful images of bone, lean soft tissue, and fat. And also now DEXA scanner software can provide estimates of not only total body fat, but an estimate of subcutaneous and visceral fat, as shown in this illustration over here. And much more recently, because there's so much obesity, now we can have very large DEXA scanners. This one, a supersized one that can quantify people who have morbid obesity. Otherwise, it's quite difficult with DEXA to get someone who weighs several hundred kilograms onto the platform. Now, one of the features of DEXA is that it allows you to measure total body lean soft tissue, the bone-free component of fat-free mass. And then if you take the appendages, they're mainly muscle in that lean component. And with that, you can quantify what's called appendicular lean soft tissue, ALST. And with that, you can predict total body muscle mass, as validated in this plot over here, showing total body muscle measured by CT scan against appendicular lean soft tissue. And I know Bill Evans will speak later about the creatine dilution method, but this is a widely used approach for quantifying muscle mass. Now, one of the limitations of all of these approaches is that they mainly quantify the size of a structure or its volume, but they don't address composition. And this just shows what happens in protein calorie or protein energy malnutrition when muscle atrophies, or as it does with aging. What happens is the proportion of that muscle as connective tissue and other non-functional elements increases with either muscle atrophy due to disease, malnutrition, or aging. And so when you quantify muscle with a technique like DEXA, you're not really learning about its composition, just its size. Now, the introduction of DEXA allowed us to develop what's called a four-compartment model for body fat. This is the most accepted reference method for measuring total body fat mass, in which you measure total body water with isotope dilution. You measure bone mineral with DEXA. It was a great advance to allow us to do that. And you measure body volume with air displacement plethysmography. And by combining these techniques, body water, bone mineral with DEXA, and body volume with BODPOT, as it's called, you can quantify these four body compartments. And it also allows you to get a pretty good estimate of total body protein. Well, one of the really major advances was whole body magnetic resonance imaging, first introduced around 1973, and then for body composition around 1992. And we can do a whole body scan. We can slice from head to toe, whereas CT provided a lot of radiation. MRI is, of course, radiation free. And as a result, you can do even pregnant women with special permission, or you can scan up through the entire lifespan. You divide the body into multiple cross-sectional slices, and then you can read those slices for each of the major body compartments, subcutaneous adipose tissue, visceral adipose tissue, muscle, and so on. And you can reconstruct the whole body then in these body compartments. And if you have enough patients, you can do 1 millimeter slices, and you can get beautiful images like this of the whole body, both transverse and cross-sectional. Now, one of the limitations of MRI is it takes a lot of work to analyze those images. They're usually done by hand. And this just shows a 3D reconstruction, a volumetric reconstruction, showing subcutaneous adipose tissue, skeletal muscle, and the intermuscular adipose tissue compartment. And this shows the intermuscular adipose tissue linked with diabetes and other diseases, but particularly with aging. All these compartments can be quantified, but it's quite tedious. The real revolution here came with what's called the Dixon method or water fat imaging, in which special scan can be done. It can be done, it's about a 10-minute scan, and you get what's called a water fat image. And that water fat image then allows you to separate out water images, mainly lean like muscle and adipose tissue separately. And now we have automated analysis methods. There's several companies that do these automated analyses. There's a cost to it, but nevertheless, it's much faster, and you can quantify individual muscle compartments. You can also measure liver fat with MRI using water fat imaging or spectroscopy. And this is now widely done. You can do these very easily with whole body or regional MRI scans. This little figure just gives you an idea of whole body MRI. It's going to be used to look at changes in muscle mass with aging. This is cross-sectional data, but you can see you get nice distribution of muscle across the age span, both in men and women, and across different race groups. So this technique's widely available today. Just shows sarcopenia as you might expect it to change. And a feature, as I mentioned, with most of these imaging methods, you get size of a structure, not its function. And what you see as people get older, the loss in muscle strength is much greater than the loss in muscle volume or muscle mass. And you can separate out these two doing both volumetric and functional measurements. MRI and MR spectroscopy also allows you to get compartments such as intramyocellular lipid, which permits you then to quantify both the lipid intracellular and extracellular space, and that component's related to metabolic features like insulin resistance. You can also quantify the resilience of muscle technique using MR elastography, again, widely available method. And the muscle becomes stiffer as it develops more connective tissue and is less good quality muscle. And this can easily be done now with special techniques available in many research settings, looking at the elasticity of muscle. Another important technique is ultrasound, been around for many, many years, but nevertheless, ultrasound now allows us to look at muscle composition. This just shows a young muscle and an older muscle, and you can see the whiteness, as it's called, of the muscle increases with age. You can get handheld devices that provide that information, and you can also do ultrasound of the liver for fat content, and this is a technique called FibroScan, and this technique allows you to quantify both the fat and the connective tissue component of liver tissue. Now, finally, in the last few minutes, one of the most recent advances is digital anthropometry. This technique was introduced in the 1980s for quantifying body size for the clothing industry, automated clothing fitting. And then it gave way more recently to laser scanners. This is a whole body laser scanner, which you can then generate an avatar of the human body. And the introduction of lower cost devices is a 3D optical imaging device. The device on the left, the laser scanners, roughly $100,000 US dollars. The device on the right is about $10,000 or $15,000. And you can get even lower cost devices than that today. This is one in which the platform rotates, the person stands on it, you get a 3D optical image, and you can even get cell phone apps that do digital anthropometry. If you're really interested enough in it, you can buy this one for home. You can do it every day if you want, if you're interested, if you're into a fitness program, and so on. And the way these work is you get an image, it generates an avatar shown on this cell phone app over here. And that avatar then can be quantified. You can measure 300 or 400 different body dimensions. These are just a few examples. And you can use those body dimensions for predicting body fat, muscle, bone, and other components. And you can also model them. For example, this particular figure shows a normal weight man, and you can then ask it, what if that man gained a little weight? What would he look like? And we have models then allow us to predict the effects of aging, variation in height or arm length, various circumferences, and so on. Here's one of my colleague, John Shepard. This is real John in the middle, BMI 28, and he asked, what happens if I lose 20 kilograms of fat or gain 20 kilograms of fat? And you can see this will predict reasonably accurately what he would look like. And this is the same John, again, losing or gaining 20 kilograms of lean mass. These are built using a technique called manifold regression analysis. And you can also predict what someone's DEXA image would look like. Here's the actual person, their optical scan, and this is the actual DEXA of that person. And this is what would be predicted just from the optical image alone. Very close, the difference being very small. And I only show these to show the power of these techniques in producing a remarkable variation in body images and body shape. So just to close then, I've gone through very quickly many techniques, but shows you the depth and breadth of how we can measure these multiple body compartments. And these are the techniques we have at our disposal: MRI, DEXA, ultrasound, optical imaging. And I didn't talk much about just simply calipers and bioimpedance analysis, but the power of this technology today to study the kinds of things Dr. Heber talked about is very, very important to us. Thanks very much.
Well, thank you, Steve. That was really a masterful review, and I know that there's no one anywhere in the world, really, that has your command of the area of body composition. Your whole career has been devoted to this. I just wonder, from your viewpoint, bioimpedance analysis has been used for a long time in weight loss clinics. Yeah. What do you see as the future of shape analysis? It seems very powerful and a way to personalize results, especially with about 40% of women not being satisfied with their so-called target weight once they get there. Do you think this has some potential benefit?
Yes, I think that we'll be able to get reasonable body composition estimates from it. That's one thing. But we'll also be able to get shape estimates, which are very unique. Body shape, as you know, for example, a classic apple and pear, which is related to your metabolic risk. That apple and pear is extremely crude sort of phenotype, body phenotype. I promise you in the coming years, we're going to see much more sophisticated body shape phenotyping with this technology. That's just a few of the things coming forward, David.
And I think there may be some genetic research as well that identifies how different fat depots are inherited perhaps in different ethnic groups around the world and how that relates to their predisposition to obesity.
Yes, that's absolutely right, is that with this kind of technology, we'll be able to get much more accurate phenotype. And I didn't speak directly on how that's done, but how do you quantify someone's shape? Well, you can look at them and think, oh, that's an apple person or a pear person, but there are mathematical approaches that can be used to give numerical values to your shape. And those numerical values then can be linked to genetic risk factors or genetic loci. And so before long, I promise you, we will be able to get the genetic associations with real shape, not just a waist circumference, but someone, I'll give you an example. People vary tremendously in the proportion of their body sizes, arm and leg length. Some people have very long legs, other people have very short legs. And those variations are related to metabolic risk factors and genetic propensities. So I think you're going to see much more of this going forward.
Well, thank you again, Steve, for a wonderful talk. And I now want to introduce Dr. Bill Evans of Duke University and University of California, Berkeley, who's going to talk to us some more about some modern methods for assessing muscle mass and the metabolic consequences. Thank you again, Don.
Thank you very much, David. It's a real pleasure to be a part of this symposium. And my talk today will discuss the importance of muscle mass and the accuracy of measuring muscle mass in helping to define sarcopenia, and to present some new data on how muscle mass may influence health-related outcomes in older people. These are my disclosures. And it's important to start off, and Dr. Heinzfield provided a really beautiful assessment of body composition methods, especially newer ones. But one of the fundamental mistakes that investigators often make is the assumption that lean body mass equals muscle mass. And we know, in fact, that it doesn't. Lean body mass, as you probably know, includes viscera, heart, bone, blood, and changes in hydration status may obscure important changes in muscle mass. Dual X-ray absorptiometry, or DEXA, which is very, very frequently used to assess lean mass, is precise, but it's inaccurate in the measurement of muscle mass. CT and MRI can provide muscle cross-sectional area of muscle volume, but with advancing age, muscle contains lipid and fibrotic tissue, and also changes in body water may reduce accuracy. So 26 years ago, I delivered the keynote address at the very first conference supported by the National Institute on Aging on sarcopenia. And at that time, my definition of sarcopenia was this, pretty simple. It is the age-related loss of skeletal muscle mass. Now, since that time, there have been a tremendous number of efforts in attempting to redefine sarcopenia. And fundamentally, the reason is that until recently, we really have been unable to measure the amount of muscle mass in a human being. And in fact, recently, the NIH put together a group called the National Institutes of Health Sarcopenia Project in an attempt to define what this thing is. And their conclusion after reviewing all of the literature was this: low lean mass by itself is a poor predictor of functional impairment compared with low strength. And that weakness increased the likelihood of mobility impairment regardless of low lean mass. And mortality risk patterns were inconsistent. So we have a definition of sarcopenia, which initially was simply the age-related loss of muscle, which seems to imply that muscle, as measured by lean mass, is relatively unimportant in predicting outcomes in old people. So my colleague and I, Dr. Mark Hellerstein, decided to take advantage of research that Dr. Heimsfield has published, some seminal work on the fact that you can measure muscle mass by collecting all of the creatinine that a person excretes in a 24-hour period of time. The data are pretty impressive, and a lot of people have used this, but there are some problems with using this method. So we use a so-called tracer dose. We put heavy hydrogen on creatine. So we use a single oral tracer dose of deuterated creatine, which, as you can see, we provide enterally. We have absorption, distribution, and active uptake of creatine. And creatine is not made in muscle. It's made in the kidneys and liver and actively transported into muscle, where about 98% of the entire creatine pool is found. Fortunately for this method, creatine is converted into creatinine in a non-enzymatic irreversible reaction, and creatinine is lost in urine. So a single urine sample at isotopic steady state, which is 2 to 4 days in most people, it can be collected, and we can measure the enrichment of creatinine and have a pretty accurate and direct measurement of muscle mass. And we do the analysis by LCMS. Interestingly and importantly, creatine is found in the contractile component of muscle. It is concentrated around the Z disc and the A band and the other components of muscle contraction. So it appears that this method actually measures functional muscle mass, and it's undiluted by lipid or fibrotic tissue. So simply put, if we use a person with a large muscle mass, as you can see here, we have a small amount of creatinine in the urine. Person with a small amount of muscle mass will have a larger concentration of creatinine. And so validated against whole body MRI that we actually performed at the Pennington Biomedical Research Center. And you can see that our method, creatinine dilution against whole body MRI, which, as Dr. Heimsfield said, is actually a pretty darn good estimate of whole body measurement of muscle. We have a strong relationship here, and importantly, the line goes right through the line of identity, which implies that these measurements are measuring the same thing. So let me turn to a series of studies that we have published from the so-called Mr. Oss trial. And this is a large NIH-funded cohort study of older men. And we received a grant from the NIH to introduce this method into this cohort. We had about 1,322 usable samples from the analysis, and we had some outliers and some men in the study who either didn't take it or took it at the wrong time. And interesting, this is the relationship we see between D3 creatine, our measurement of muscle mass, and DEXA, total lean mass. What you can see is just a moderate R value, but you can see that DEXA vastly overestimates the amount of muscle because it measures lean mass. Interestingly, when we compare our measurement against DEXA appendicular lean mass, which is most often used as an estimate of muscle mass, because this is the lean mass of the arms and legs, there is no correlation at all, not even the hint of a relationship. So DEXA appendicular lean mass is measuring something completely different. Now, in our population of older men, and when we compare our method to DEXA, we know that DEXA actually measures fat mass pretty well, and that's what you can see here in yellow. Bone is in black. The light blue plus dark blue is lean mass, lean body mass, and the dark blue is muscle mass, ranked from high to low. And what you can see is that this light blue, or what we call residual lean mass, is pretty unrelated to muscle mass. The yellow dots here are appendicular lean mass, which is unrelated to muscle mass. So in this population, we were able to see that, for example, a number of functional measurements like time to complete 5 chair stands is strongly related to muscle mass. Compared and using appendicular lean mass, there's no relationship at all. Peak lower extremity velocity at peak force, highly related to muscle mass, no relationship with DEXA appendicular lean mass. Time to complete a 400-meter walk, the short physical performance battery score, which is now a standard geriatric assessment, strongly related to muscle mass with no significant relationship with appendicular lean mass. And looking at the risk of mobility limitations, bathing and showering, difficulty getting in and out of bed, lifting at 10 pounds or performing heavy household work, we see a strong relationship with muscle mass, a strong risk of low muscle mass. And if you look at DEXA by appendicular lean mass, no relationship at all. More recently, we've been examining the effect of fat on functional status. This is a paper that is now in press in the Journal of Gerontology, titled The Importance of Muscle versus Fat Mass in Sarcopenic Obesity, a reevaluation using D3 creatine muscle mass versus DEXA lean mass. And our conclusion here is when an accurate assessment of muscle mass rather than lean mass is used, reduced muscle mass is highly associated with important outcomes and the negative effects of adiposity are minimal, suggesting that obesity has little relevance for the understanding of the important outcomes of sarcopenia. And we can see that here looking at a standard geriatric assessment, which is walking speed, habitual walking speed. You can see we look at fat quartiles versus quartiles of muscle mass. And what you can see in this measurement is that low muscle mass is highly related to walking speed, irrespective of how fat the subject was. And by appendicular lean mass, we see almost exactly the opposite relationship. You can see that a little bit more clearly here. This is looking at the likelihood of prevalent mobility limitation, DEXA versus percent fat, and you can see whether a person is higher or low fat. It's the low muscle mass that is predictive of mobility limitations, whereas with DEXA appendicular lean mass, it's really just the opposite. We also just published a paper looking at the dietary intake and D3 creatine muscle mass versus appendicular lean mass. And very interesting data here to go along with what Don was saying is that we're looking at here the relationship between total protein intake and muscle mass. And you can see that looking at non-dairy protein, animal protein intake in particular, was strongly related to the amount of muscle. And if we look down here at DEXA lean mass, there was, there was no relationship. And so our conclusion was adherence to a Western dietary pattern, which was rich in red meat, fried foods, and high fat, was associated with higher muscle mass in older men whereas adherence to a healthy dietary pattern, which was rich in vegetables, fruits, whole grains, and lean meats, was not associated with muscle mass or DEXA appendicular lean mass. Recently, we've looked at the risk of low muscle mass in looking at the prediction of fracture risk. And again, you can see that looking at hip fracture in this patient population, we can see a strong relationship between muscle mass and risk of a hip fracture or the amount of hip fractures, with no relationship with DEXA appendicular lean mass. And whether we adjust by bone mineral density or FRAX or chair stands or function, the relationship is still true. And recently we have a paper in publication looking at muscle mass and incident disability and mortality in older men. And again, just to remind everybody, activities of daily living are eating, toileting, transfer, bathing, and showering. And instrumental activities of daily living are more activities that, for example, balancing your checkbook, that involve cognitive function. And what we can see here is we look at low quartiles versus high quartiles of muscle mass, again, strongly related to things like gait speed, chair stand time, and importantly, incident ADL disability, a much higher risk with low muscle mass versus high muscle mass. And interestingly, even incidental activities of daily living and cognitive function are associated with muscle mass. And this is the IADL disability, and you can see the same sort of relationship here. So low muscle mass is a risk factor for ADL, IADL, and mobility disability, and low DEXA appendicular lean mass not significantly associated with disability. And in this paper, we also show that muscle mass in this patient population of older men is strongly related to risk of mortality, whether it's multivariate adjustment or adjusted multivariate plus performance and strength adjusted. We still see that muscle mass is a powerful predictor of mortality. And so I just want to also show you one other paper that was published recently looking at change. So we were able to measure a group of men before and after about a year and a half. So we have repeat assessments in a convenience sample of about 40 men before and after 1 year. And so let me just show you, over the course of a year and a half, there was no change in lean mass, no change in appendicular lean mass, but actually a pretty large change in muscle mass on the order of 4 to 5 kilograms of muscle, and no change in body weight. However, there was a significant reduction in grip strength and a significant slowing of walking speed. So if you didn't measure muscle mass in this study, your conclusion would be, and the conclusion of most of the field would be, that there's some intrinsic change in muscle, something about muscle quality is changing because muscle mass isn't. But if you measure it accurately, we see that the loss of muscle mass is significantly related to the reduction in function. And in an editorial accompanying our paper, Laura Schaap concluded that by isolating contractile muscle from non-contractile components, including fat, the D3 creatine assessment is not only an accurate method to assess muscle mass, but is less biased by obesity and aging than DEXA appendicular lean mass. And so what I call the streetlight effect has been operative, I think, for the last 20 years in the field of sarcopenia. Some scientific research is shaped by the need to perform replicable measurements, but these measurements do not always accurately reflect the phenomenon that is being investigated. The term streetlight effect is sometimes used to name this form of observational bias. And remember this old cartoon, this guy is saying, I'm looking for my quarter. I dropped it. Did you drop it here? No, I dropped it 2 blocks down the street, then why are you looking for it here? Because the light is better. The definition of sarcopenia it has inconsistency, and there's a lack of consensus, and based on a fundamental error. And that fundamental error is this, that lean body mass is the same as muscle mass. The measurement of lean body mass is an assessment of muscle mass is the street light. DEXA is precise but not accurate for the determination of muscle mass. We have sacrificed accuracy for precision and ease of measurement. The loss of muscle with advancing age is universal, complex, and multifactorial. Provides multiple targets for drug development, stimulate anabolism or inhibit catabolism, are a number of different targets, but the true extent of functional skeletal muscle loss and its effects have not yet been determined. Sarcopenia probably equals dynappenia, or the loss of force production. We have to take into account the role of muscle mass in the risk of disability and age-related risk of chronic diseases, and we must start with an accurate measurement of functional muscle mass that's undiluted by lipid, connective tissue, fibrotic tissue. In this way, the true effects of intrinsic age-associated effects on muscle contractile function can be determined. Thank you very much.
Bill, that was an outstanding talk. And I really want to salute you for doing something that really makes us look a little differently at this whole problem of sarcopenia. And you being one of the pioneers of sarcopenia, that's particularly important, especially with regard to sarcopenic obesity, which is so common and so underappreciated. What I didn't realize until today's talk is how the location of the creatine is in the contractile element of the muscle. So right. It's truly functional. And I'm wondering, in the future, how you might look at the activation of that creatine. Is it phosphorylated? Do you know what percentage is phosphorylated? And is it really kind of the stretched rubber band waiting for the muscle to activate?
Well, the creatine dilution actually measures both phosphocreatine and creatine. So it is truly just the total body creatine store, but it is functional muscle mass. And it's interesting, we see this strong relationship in very old men, perhaps because with advancing age, there is a great increase in both lipid and fibrotic tissue. So the men in our population are between the ages of 80 and 100. So these are men that have a large variability in functional muscle mass. And it's probably, and the fact that we see these powerful relationships is because we're measuring just that component of muscle that is the contractile component of muscle that's undiluted by all the other things. We see the same sort of relationship now in boys with muscular dystrophy, whose muscle is filling up with fibrotic tissue and fat. Same sort of relationship between this measurement and function.
So do you believe the phosphorylation of the creatine is a fairly constant element, or do you think that's also varied?
It's fairly constant, and it's related to how much creatine you have. So that's why creatine supplementation increases the amount of creatine in the muscle and therefore increases the amount of phosphocreatine in muscle. So that's the one caveat. If someone is taking creatine supplements, it'll look like they have more muscle mass than they actually do with this method.
Very interesting. Well, thank you very much again, Bill. And now it's my pleasure to introduce Dr. Don Layman, Professor Emeritus from the University of Illinois, who's going to give us a wonderful talk on protein metabolism. Don?
It's my pleasure to join the seminar today, and this is just a list of my disclosures. I retired from the university a few years ago and primarily doing consulting now. I think everyone recognizes that proteins provide amino acids, but we make a mistake in saying that we have a protein requirement when we actually have a requirement for individual amino acids. It's like saying we have a requirement for a vitamin pill when we don't really require the pill. What we require is the 12 amino acids in the pill. protein is simply a food source of amino acids, and specifically the 9 essential amino acids, which we can't make. When we begin to compare plant and animal proteins, it's important to focus on the essential amino acids, and especially the 4 amino acids, lysine, methionine, tryptophan, and leucine, and for us to understand that all plants proteins are low or limiting or even deficient in 1 or more of these essential amino acids. In freshman nutrition, everyone learns that amino acids are building blocks for proteins. This is the most fundamental of the requirements and represents charging of tRNAs. This requirement is met at minimum cell concentrations and with minimum dietary intakes. Beyond this basic role, virtually every amino acid also has metabolic roles. And for a few examples, leucine is a stimulation for mTOR for protein synthesis. Cysteine is a precursor for glutathione, an important antioxidant. Lysine is the precursor for carnitine for fatty acid oxidation and transport. What's important about all these metabolic roles is they're all dependent on intracellular concentration and ultimately on the amount of protein. So in essence, we can have a low protein diet and meet the basic function of building blocks, but we downregulate all of these metabolic roles. In the last 15 years, we've had a changing view of protein needs, and that's largely been driven by our understanding that with aging, we get a reduction in the efficiency of protein utilization. Our ability to replace and repair proteins tends to go down, and we refer to this as anabolic resistance. And the problem is keeping muscles healthy. We know that lifetime health relates to things like sarcopenia, osteoporosis, insulin sensitivity, fatty acid metabolism, and all of this leads us to sort of think about more of a muscle-centric view of protein needs. How do we keep muscles healthy as we age? As I mentioned, one of the key aspects of this is protein turnover. We have a continuous need to repair and remodel existing proteins. In fact, the body has to build approximately 250 to 300 grams of new protein each day. We always talk about growth being in place for protein, but growth is actually a pretty minor amount. Maximum rates of growth as far as accumulation of new protein is only about 5 to 10 grams per day. But repair and replacement requires us to make 250 to 300 grams. One of the important aspects to also remember is every tissue is different. There's a different priority. For example, the liver or the kidney or the heart have very high priorities for protein synthesis. Of the 250 grams of protein we make per day, 75% of it goes to the liver and other organs to replace it. So even during an overnight fast or even during a long-term fast, the liver continuously has to make proteins or we simply die. Skeletal muscle, on the other hand, makes up over 50% of body proteins and is only allocated about 25% of daily protein synthesis, and it's much more sensitive to diet. Optimal diet is very important in terms of optimizing muscle health. Regulation of protein turnover in each tissue is also different. I'm going to concentrate on muscle. One of the key regulatory sites in skeletal muscle is an enzyme complex known as mTOR or mTOR complex 1. And it's a primary regulatory site, and it integrates input from a variety of different signals. Across the top of the slide, I have 4 different keys. One is dietary protein, and it senses specifically the branched-chain amino acid leucine. It senses hormones, particularly insulin and IGF-1. It senses overall energy, particularly glucose, through the AMP kinase mechanism, and various kinds of stress, and in muscle, particularly resistance exercise. So mTOR is able to integrate all of these and decide that the environment is correct to trigger this very expensive process of protein synthesis. What's also important is this is different between tissues, and it also is different at different ages. So this is the full complex of it, and I just want to highlight sort of one comment. down at the lower, in the middle, just above protein synthesis, one of the initiation factors, EIF4F, there's about 12 initiation factors controlling protein synthesis. In muscle, EIF4 is a key, and for example, in liver is regulated primarily by EIF2, 2B, which are mostly energy sensitive. So we have tissue regulations and other things going on tissue to tissue. I want to simplify that just for a second to highlight one point. In skeletal muscle, two primary regulations are insulin and leucine. What we know is that in children and young adults, they are very responsive to small meals of protein. Breakfast meals, for example, of 8 or 10 or 12 grams of protein will stimulate muscle protein synthesis in a growing child. That's primarily because protein synthesis at that point is driven by hormones. Once we get beyond growth, the hormones no longer regulate this process, and we shift to diet quality, and the primary signal is leucine. And what we now know is the regulation requires approximately 30 grams of protein and primarily 2.5 to 3 grams of leucine to stimulate this mechanism. This overall role of leucine and the overall regulations are pretty well recognized now. And this was just a summary from an international study group called the PROTAGE study group. And they recommended that for older adults, we need to be focusing on meals that have at least 30 grams of protein, supplying at least 2.5 grams of leucine. So if leucine is an important criteria, then it's important to look at different foods. So as I've just mentioned, our requirement we're targeting is 2.5 grams per leucine per meal. If you look at the footnote, the current FAO requirement for leucine is 59 milligrams per gram of protein, which is 5.9%. And for the RDA and a 70 kilogram person, that translates into 2.7 grams per day. So 2.5 grams per meal is the minimum versus the 2.7 grams per day, pretty different. If you look at the black box then of the different proteins, you see that going from whey protein, which is particularly rich in leucine at 11%, down through various different proteins, down into the grains, which are fairly low, around 6%. If you look at quinoa, for example, at 6%, it meets the FAO requirement for protein of 5.9%. But what's important to recognize is if we're trying to meet these meal requirements for adults, you can meet it with a whey protein meal of 23 grams or a soy meal of 32 grams, but quinoa requires 40 grams and over 1,100 calories. So basically, you walk into a health food store and you'll see that quinoa is listed as a superfood with all the essential amino acids, which is true, but it requires a lot more protein and a lot more calories to get to the adequate amount. The hypothesis supporting the overall plant diet sort of takes two forms. One, it's better for health, and one, it's better for the environment. And those are subjects for other talks. But what I really want to do is just highlight the issue of what are unintended consequences of going this way? What are some of the things we're going to see, and really where are we currently at? So I think it's useful to look at our food distribution in the U.S. right now. So these are data from the NHANES data, and I've got 2010 here. 2016 is essentially the same. And what we see is that Americans already have a plant-based diet. We get over 70% of our calories from plants and only about 30% from animal-based foods. But if you look within that, in the large circle, we get 51% of our calories, our plant-based calories, come from added fats, oils, and sugars. And in the small circle, another 33% from refined grains. So of those plant-based calories, over 80% are coming from foods that have very low nutritional value. I would argue that we really don't need a more plant-based diet. We actually need a diet with better plants. One last thing is in those 30% of calories coming from the animal products, it's important to recognize that we get over 70% of our dietary protein, almost 100% of calcium, vitamin D, and B12, and over 60% of zinc, iron, selenium, B6, etc. So the question is, how much can we take out of this category and still maintain a nutrient-dense, healthy diet? Sort of related to that, Christian Peters at Tufts University looked at some of the plant-based environmental models, and he sort of looked at what would happen as we deplete protein out of the environment by changing agriculture. So this category, if you look at the black box on the left, he went from high omnivore diets down to vegan. And if you look at the yellow box, basically as you do that, you deplete protein out of the environment. This isn't just a dietary recommendation. This is changing agriculture. So we're actually depleting it from the food supply. And to replace that on the gray box on the far right, basically you see we're going to replace it with carbohydrates, primarily from grain products. And so one of the questions about unintended consequences is who's going to be affected by this? If we deplete protein from the grocery stores, who's going to be most affected? Is it going to be the individual having steaks at expensive restaurants on expense accounts, or is it going to be our most vulnerable and lower income populations? So we've talked about reducing protein quantity. Let's shift over to quality. As I mentioned before, two of the most critical amino acids in the food supply are lysine and methionine. So these are data from Luc van Loon in the Netherlands. And at the top, we have the lysine, and the bottom, we have methionine. In the middle of each screen, each table, what you see is a dashed line, and that's the requirement. So in the open bars on the left, you see all of the plant-based foods, and you can see they're lower in lysine and methionine. In the top bar, in the top panel, basically lysine, I've highlighted the blue. Cereal grains, oat, wheat, and corn are particularly low or deficient, versus in the gray bars, we have the animal proteins, which are all adequate. The bottom set of bars, we have methionine, and in that case, all of the legumes, soy and pea, are particularly low in the sulfur amino acids. If we look at protein quality, it basically has two components to it, trying to assess it. It's amino acid score and also the bioavailability or digestibility. The current measures are PD-Cas and DIAZ, digestible indispensable amino acid score. So we're going to look at those briefly in a table. This table obviously has lots of numbers in it, so let me sort it out. This is from Paul Mohan in New Zealand, who's one of the world's leaders in protein quality measures. In the if you look at the vertical bar, the blue bars I put in, the vertical one, to the left of that are all protein isolates and concentrates. These are all highly purified proteins. And to the right-hand side are all the whole foods. If you look across the yellow bar at the bottom, these are DIAZ scores. And what you see is basically a trend going from higher values down to lower values. All of the isolated purified proteins are 0.8 or higher, and everything in the natural proteins are 0.6 or lower. If you just look at the wheat bran column, for example, a 0.4, what that means is if you pick up a wheat cereal, for example, and it says it has 10 grams of protein in it, its bioavailability is actually only 40%. So it actually only has a true availability of 4 grams. Let's look at some of the DIAZ scores. What I've highlighted is whey protein isolate and soy protein isolate. And in the red triangles at the bottom, what you see is the WPI is 1.09, where the soy protein isolate is 0.906, not enormously different. If you look within the columns, though, what you see is that these scores are based on the lowest of the single essential amino acids. So for soy protein, it's based on methionine, which, as I highlighted earlier, is a critical amino acid. But for whey protein, it's actually based on histidine, which is probably the least essential of all the essential amino acids. In fact, it's been shown to be essential in infants, but to my knowledge, has never actually been shown to be essential in adults. So we're kind of comparing apples and oranges. If we compare apples and apples and look at methionine between whey and soy, what you see is the numbers almost twice as high. Or if we look at lysine, it's almost twice as high. So if we're really looking at the critical amino acids, there's enormous differences between the amino acids. So then just looking at lysine, again, the metabolic need for lysine is about 3.4 grams per day. And if you look at the black box, again, it shows sort of highlights different foods going from animal products that are fairly rich in lysine down to wheat, which, as I've already highlighted, is actually quite deficient in lysine. It's important to note that wheat provides over 50% of the plant-based protein in the American diet under the current situation, current eating patterns. So if we look at protein required to reach that lysine requirement, around 40 grams of animal proteins will reach the lysine requirement, where it takes 130 grams of wheat to do it. Also in freshman nutrition, we all hear about complementary proteins. So if we have a protein that's particularly low in one amino acid, we can balance it with a protein. And since we usually don't eat just single proteins, this is very important for mixed meals. So wheat, again, is a good example. We can take the wheat gluten, which I said already is deficient in lysine, and we can balance it with beef, make a roast beef sandwich, which is very rich in lysine. So 1 gram of wheat can be balanced with 1 gram of beef, and it's a complete protein. We can also do it with cow's milk and soy. We can take 1 gram of wheat, and it will be balanced with 1.5 grams of cow's milk, but it takes over 6 grams of soy. So once again, the take-home message is complementary proteins are important, but if you do it with plant proteins, it takes more proteins and more calories to be equal. So just to summarize that, what we now realize is that as we get older, we have anabolic resistance that reduces the efficiency of our protein use. Thinking about a muscle-centric type of health, we really need to increase our protein, and most of the recommendations now are protein intakes above 1.2. The concern with that is that all of the plant-based models that are being discussed all reduce the protein quantity, quality, and bioavailability. And so we're sort of left with worrying about the unintended consequences of this. We can argue about the importance of a high-quality plant-based protein or diet, high-quality plants such as vegetables and fruits, but we need to be careful about the protein content and really worry about the most vulnerable people in our population. So with that, I'll say thank you and turn it back over to our moderator.
Hi, Don, thank you very much for a wonderful talk. I think what you're moving us toward is optimum protein as opposed to minimum protein. And really, most government recommendations around the world are based on data from 1973 on the minimal protein to reach nitrogen balance, which is really a crude measure. So I really want to salute you for really getting us down to the micro level of how we can develop this. And I wanted to just ask you, what do you see as the future of protein blends that will take advantage of maybe allowing us to have our cake and eat it too, so to speak?
Yeah, I think you're right. I think it's important to recognize that the minimum requirement, the RDA, is very important. And worldwide, there's still many people who are struggling to get to that level. So malnutrition is an issue. But in more developed countries where obesity is a problem, optimizing protein for adult health is our key. And we're seeing that really requires a better protein. And as far as blends, I think that's going to be important. And right now it's kind of either or. It's like it has to be plant or it has to be animal. And I think my last slides highlight that the best is really a blend of both. The complementary concept is when we use both and emphasize the huge benefit of essential amino acids in the animal protein to complement the poorer quality of the plant proteins.
Thank you very much again, Don. What a wonderful, masterful talk. And it's really a pleasure to have you on our panel. And it's my pleasure to introduce Dr. Xiaoping Li, who is the director of the UCLA Center for Human Nutrition. Dr. Li is going to speak to us about branched-chain amino acids, which we've been talking about a little bit in Don's talk, and their effect on muscle and also glucose metabolism. Dr. Li.
Hello, everyone. This is Xiaoping Li. It's truly a pleasure to join a group of world-renowned specialists here to talk about protein and the assessment of muscle mass and muscle function. I really don't have any relevant disclosures. I'm from the UCLA Center for Human Nutrition, And our mission, it is to study food and study each individual and trying our best to figure it out what is the best food and for any given individual. And clinically, we're trying to practice we call individualized nutrition or personalized nutrition. Obviously, the goal for all of us in the near future would be precisional nutrition for everyone. And Dr. Lehman has talked about protein quality. And I'm going to be talking about specifically protein quality, particularly in the setting of branched chain amino acid. As we have reviewed with Dr. Lehman, there are 3 branched chain amino acids, and they are within the 9 essential amino acids. And here are the structures of those 3 branched chain amino acids. You can see the smallest molecule is valine, and there's leucine, isoleucine, with the carbon branch chain at different location. The reason they're important, particularly as a group, is that they are not just building blocks for proteins and other essential neurotransmitters and cholesterols and everything else. And there is a very special function this group of amino acids serves, and that is they are also very important for nutrient initiated signaling. That include what we have heard about mTOR activations, is also really regulate the branch point where the amino acid would go for oxidation, catabolic process versus even lipogenesis versus protein synthesis, as we have heard. And those functions together, that is serving as a basic nutrient, as a building block for protein, and can be oxidized to produce ATP, can be converted to glucose through gluconeogenesis, or lipids through lipogenesis, and also with what we're just talking about is a regulatory function. So that really set the stage for branching amino acid to be involved in many chronic diseases. And that varies from cell growth, that we talk particularly in growing kids and adolescents, and to gene regulations, organ functions, even to gut microbiota. So we are going to try to review globally and how branching amino acid it is involved in overall metabolism. So here is just a cartoon to refresh our memories how branching amino acid, particularly leucine, as a signaling and also regulator for protein synthesis, particularly in the muscle. As you have heard from Dr. Lehman, leucine in particular, out of the 3 branching amino acids, is a strong stimulator of mTOR, particularly complex 1. And that would really start a downstream protein synthesis, and also they have also inhibitory effect on protein breakdown. So as a net result, and leucine will stimulate muscle protein gain overall. So that is one of the key regulatory features of branched chain amino acid. And on the other end, for the catabolic metabolism, here's how the branched chain amino acid is being catabolized. So the first step is losing the amino group, and that is through the enzyme called branched chain aminotransferase. That is really a not highly regulated step, and is also reversible step. The next steps happening in the catabolism of branching amino acid actually is highly regulated. It is a irreversible step of branching amino acid metabolism. So that is really controlled by a enzyme complex that is located in the inner layer of the mitochondria. It is called branch chain ketoacid dehydrogenase, or BCKD as short. So as you can see in this cartoon, this enzyme complex is activated by an enzyme, the phosphatase called PP2CM, but is also inhibited by a kinase that is called that will cause one of the subunit of the enzyme to be phosphorylated. So that is indicated here in the red. After passing this step, and then the branch chain amino acid will be oxidized to produce the ATP, and as we can see in this cartoon, demonstrated TCA cycle. And next, I wanted to just go over how the BCAA overall homeostasis is regulated in our body. This is what we're very familiar with, glucose homeostasis. And what we're looking at here, it is a glucose tolerance test. And this, we know this so well, and even the two different phases of the blood glucose levels in the blood. And what we're not so familiar, it is after BCAA challenge, and our body also tightly regulate and the blood BCAA level. As you can see here, is almost mirror image of the glucose tolerance test. You see a first phase rise of BCAA, and that is quickly regulated down to the baseline level. So that really, make us to think, what are the steps involved in really tightly regulate the blood branching amino acid level? So here is another cartoon to remind everyone. And BCAA is also unique in such a way, it does not get metabolized in the liver. So there is no first pass effect. So BCAA got to the blood in its original form, and then get a chance to go to every single organ and the tissue. Only when the amino group was removed, as we talked about above, and then the ketoacid acids, and then back to the bloodstream, and then got reupdated in the liver hepatocytes, and then there will be more catabolic process start right there. So this is another unique feature of how BCAA may be different from all other amino acids or nutrients. So now we talked about all the basics, physiology and regulations of BCAA. So why we are really having so much research going on to study BCAA, and also why we have so much controversy or concerns related to BCAA intake and its blood levels. And this is really because the fact the association has been discovered between the blood levels of BCAA and the risk of diabetes. One of the studies is showing here, as you can see, in the different quartile of people with BCAA intake. The higher BCAA, not intake, blood levels, you can see in the higher circulating levels, like the upper quadrant of 4, you have much higher risk for diabetes and also that translated to less survival. So this is very striking, as we can see here. And then coupled with animal studies, when we try to figure out the causation or its association between the diabetes risk and BCAA, so we got to animal model. This is one of the hallmark studies done by the group in Duke, led by Dr. Newgard. In this particular study, for rat, when they are fed with the high-fat diet plus or minus BCAA, you can see from this slides, in spite of BCAA addition, you kind of prevented the weight gain, but you still see tremendous insulin resistance associated with additional addition of BCAA. And this is one of the studies make everyone really on edge as to, well, what is going on? Are we really getting the insulin resistance not just from glucose, but it may actually from our dietary intake of BCAA, or the another group was saying it is the BCAA catabolic process that is not working well, and that is the fundamentals or stepping stage of insulin resistance, and then later on diabetes. And this is another human study, so we're not rats and we're not mice. So in this study, it is the same group of people. Look at subject two cohort, one in Columbia, one in Duke, and they look at the BCAA response to the dietary intervention, either after bariatric surgery versus just starting a low to very low calorie diet, matched for weight loss. But you can see here, though, it doesn't matter is the bariatric surgery. Here is the gastric bandingplasty versus the diet. Even the match for weight loss, and you can see the BCAA level are significantly different. And this is another evidence people would quote, and BCAA really could be the causation for insulin resistance is not as a result of insulin resistance. So this is where all the controversies start. And there is another metabolomic association study and trying to also address the same question. That is, and this is what is the chicken, what is egg, what would come first? And in this study involving a couple of thousand people, they also see association of genetic variations that would really would result in high BCAA level, and those genetic variants of the individual with the genetic variant would also have also high risk for diabetes. This is a different methodology, kind of suggestive the same relationship. Again, all those studies have its limitations, and even the surgery, the weight loss match surgery versus medical weight loss study, and that the diet is not matched, and it is also small subject number study. And so the truth is we're still not convinced in what it is really going on here. And on the other hand, what we see on the market, it is all kind of products with BCAA, and they're not only muscle builders taking protein supplements, particularly here was we see BCAA, but there are plenty of patients, including my own, are taking those supplements. And I really have to say that nobody have, in our practice, we really have not seen, quote, negative impact and by patient taking those products. So that really makes us wonder what is really going on. Here's one study looking at obese women with metabolic syndrome. And in addition to their usual diet, all the women were provided additional branched-chain amino acid supplement for 8 weeks. What you can see here from those small figures is that the amino acid, each individual levels, or the total branched-chain amino acid level, including their first step metabolite, did not change in spite of additional supplementation of BCAA. So they are remaining same. You look at the gray is baseline versus the dark gray, that is after supplementation. In spite of the blood level are not changed, but if you look at the HOMA-IR relationship with the plasma BCAA level, and at baseline, they were highly correlated. But after 8 weeks of supplementation, however, the associations no longer existing. That really are indicative, and they're actually significant change. And that is not consistent with if the high BCAA is a causative reason for insulin resistance. On this background, our group decided to look at the issue as well, particularly in the context of glucose metabolism, insulin response. So we designed a study similar to the study we just talked about, and we recruited obese individual already with pre-diabetes, or we should say with glucose intolerance. So we would assume those people are already reached their, or about just about beyond their energy balance. So they are getting to the point, lose their metabolic control for glucose metabolism. So we randomize those individuals to a sequence of taking BCAA first, 20 grams, versus taking a low 20 gram amino acid control. So we're controlling how much amino acid load we're giving to the individual. And then we have a crossover with 2 weeks washout in between. And we did a glucose tolerance test at the baseline after each supplementation 4 weeks. And here's what we see. Oh, here's the composition of the product we used. The BCAA supplementation, you can see, is 20 grams. It is leucine is 10, and isoleucine valine is each about 5. And the rice protein powder has less than 5 grams of total BCAA, but it accounts for amino acid and protein load. And this is our key finding. We did not really see the 20 gram supplementation of BCAA had impact on the glucose response in a standard glucose tolerance test. However, given the study we only have a little bit more than a dozen of people, we do see, and there is decreased insulin response with a p-value of 0.08 and a strong a trend to see significance. So this really make us to think, and it is not as we think as straightforward. This association, it is definitely there, but it does not seem like we settled with the question if BCAA is really causative for insulin resistance, and more looks like it is a contrary, and the BCAA is likely a result of insulin resistance. And also the BCAA supplementation may through mechanism we're not quite understand. And from the study we know, likely by increased protein synthesis, particularly in muscle, and to really ameliorating the insulin resistance. So that is what we learned from our own experiment. In our studies, the same thing, the blood level of BCAA did not change in spite of supplementation, and we did not see metabolites change either. So the other aspect we have not been really fully taken into consideration or control is that giving individual, a patient, when they're taking whey protein, there could be a lot of other factors, and that is contribute to what we see. One thing is, like this study is showing, whey protein consumption could really have impact on the individual satiety. So that may then in turn to translate to decreased overall calorie intake. Those are all good questions to really look at in much more detail. In this study, they did see significant change in one of the satiety regular hormone, that is GLP-1, as we see right here. So truly, to summarize the state we're at as to BCAA and insulin resistance, and we really need to consider, and this is a very dynamic process, and it is really, interfaced with each individual's gene, environment, and also other life, physical activities, and concurrent nutrients intake, and overall energy balance of that individual. So the second level we need to really look at is that each different tissue may have a different way to be regulated for BCAA metabolism. In muscle, we talked about their protein synthesis and also their catabolic function. But in liver, like we just talked about earlier, it may actually be totally different than muscle, and depends on your overall body energy balance and other nutrient component. And each tissue can be regulated totally differently. Those are things really warrant further investigation and study. So that is pretty much, I'm planning to talk about today, and is listed here in the summary, as I have just gone over. And thank you all for your attention.
Dr. Lee, thank you so much for a wonderful presentation. I think your talk was really masterful in making us realize that when you look at leucine or branched chain amino acids, we're not looking at isolation and sort of a drug kind of approach, but we're looking at a metabolite that sits at the center of protein metabolism, carbohydrate metabolism, fatty acid metabolism, and the energetic center of the cells in the mitochondria. So I think this is really of interest, and I'm wondering where these studies are going to go in the future. If we're going to use branched chain amino acids and possibly creatine, as Dr. Evans had suggested, to overcome the weakness of sarcopenia with aging, what do you see as the role of branched chain amino acids now that you've shown that really, even in pre-diabetic people, it appears to be relatively safe?
Thank you, Dr. Heber. I would want to say, number one, BCAA supplementation for healthy people is definitely safe. And we have seen some benefit for our obese, insulin-resistant individuals. As to the population has been talked about by Dr. Evans with sarcopenia and elderly, I think there definitely a big role for BCAA to take as well. I think all our studies really need to look at different populations and look at different conditions of each individual. I think we probably need to look at male and female as well, and premenopause and postmenopause. All of those are important factors to consider to really answer the question, what is the role for BCAA in the overall effort to live well and live better.
Well, thank you very much again, Dr. Lee, for an excellent presentation. I want to thank all of the speakers, Dr. Steve Heimsfield, Dr. Don Lehman, Dr. Bill Evans, and Dr. Lee for their outstanding presentations. And of course, I want to thank our sponsors, the Herbalife Nutrition Institute and the American Society for Nutrition. And I want to thank all of the attendees who have come to this symposium, and I hope that you all enjoyed the science that was presented. Thank you very much.
Welcome. This is Sarah Olhurst, ASN's Chief Science Policy Officer. Thank you so much for joining us for this afternoon session, Strengthening Federal Nutrition Research, Gaps and Opportunities, including Lessons from COVID-19. As a global organization with more than 7,500 nutrition researcher members, opportunities to strengthen nutrition research are of utmost importance to the American Society for Nutrition. This session, organized by the Tufts University Friedman School of Nutrition Science and Policy, will build on ongoing work supported by the Rockefeller Foundation to identify the most promising approaches to strengthen federal food and nutrition research and policy, as well as highlight findings from a forthcoming white paper, also organized by the Friedman School, focused on strengthening federal nutrition research. This session and the white paper are in line with ASN's own efforts to strengthen nutrition research, such as through ASN's identification of the top nutrition research needs in the Working Group Report, Nutrition Research to Affect Food and a Healthy Lifespan. ASN regularly advocates for increased support for nutrition research and has for many years sought better coordination of nutrition research across all federal agencies. Last year, ASN requested member input regarding one concept covered in this white paper, a National Institute of Nutrition, and held a related panel session during Nutrition 2019, ASN's annual meeting. Today, we will hear from two panels with a diversity of experts who will discuss the federal government's coordination and support of food and nutrition research, outline various limitations of the current system, and potential paths forward to strengthen federal nutrition research and coordination at the National Institutes of Health, as well as across the more than 10 federal departments and agencies that currently conduct or rely on federally supported human nutrition research. Other topics presented in the white paper will be discussed, such as diet-related health burdens and their implications on many facets of American life, including healthcare spending and military readiness. We will hear more about food and nutrition challenges and concerns, particularly during the global COVID-19 pandemic, from Dr. Dariush Mozaffarian, Dean, and Jean Mayer, Professor of Nutrition at Tufts University Friedman School of Nutrition Science and Policy. Next, Dr. Sheila Fleischhacker, with Georgetown University Law Center, the paper's lead author, will review the white paper's findings, including opportunities for and the potential impact of new scientific discoveries related to nutrition. The comments of the speakers and panelists are their own and do not necessarily represent the positions of the Rockefeller Foundation or ASN. Following the panel discussions, there will be a live Q&A with the opportunity for members of the viewing audience to ask questions of today's panelists. At any time today, please feel free to submit your questions throughout the session using the questions box. The session will end with closing remarks and potential next steps from Dr. Paul Coates, ASN's Vice President-elect. I'd like to thank all of the speakers and panelists today for taking time to join this important discussion. And now I will turn the program over to Dr. Mozaffarian.
Hello and welcome to this ASN session discussing federal nutrition research. I'm going to talk about food and nutrition and particularly why research is so critical in the current era. The world is facing stark global nutrition challenges. Poor nutrition is the single leading cause of poor health around the world, largely due to chronic diseases like obesity and diabetes, cardiovascular diseases, many cancers, and also due to undernutrition, which is a huge problem for many low-income populations. This is also causing and worsening and exacerbating diet-related health disparities. In our country and around the world, those who are disadvantaged or marginalized often have the worst diets, and this leads to vicious cycles of poor health, worse productivity at work, challenges in learning at school, and continued poverty. This is a major, major issue in our country and really in every country around the world for healthcare costs. The spiraling healthcare costs in the United States in particular are largely due to chronic diseases. And this is really straining government budgets and the growth of private businesses and overall economic growth. There are also enormous issues in food and nutrition for sustainability and climate change. 70% of the world's water use, 90% of tropical deforestation, 25% of climate change gases, stress to the oceans, loss of topsoil. The resource challenges and opportunities are incredible. And this is also a major issue for national security. Almost 3 in 4 young Americans who want to enroll in the military can't qualify. And the #1 medical disqualifier is obesity. And that's why groups like Mission Readiness, nearly 800 retired admirals and generals, have said that poor nutrition and childhood obesity is a national security threat. And so nutrition touches almost every aspect of our lives. And this has been really, really laid out in dire terms for COVID-19. We know so little about how food and nutrition could influence immunity and help bend the curve for COVID-19. The challenges of COVID-19 are also causing incredible food insecurity in our country and around the world. We're learning that the top risks for poor outcomes from COVID-19 are diet-related illnesses like diabetes and obesity and hypertension, which I'm going to talk a little bit more about. This is a huge issue for seniors in whom COVID-19 is causing by far the largest number of deaths and in whom we know there's hidden hunger with significant prevalent nutrient deficiency. Disparities are also clear in our country and around the world. African Americans, for example, are suffering much higher rates of poor outcomes from COVID. COVID has disrupted supply chains, causing incredible food waste, causing uncertainty and food insecurity in our country and globally. Incredible losses to the economy, lots of confusion to the public, and really critically, insufficient science and nutrition to address these challenges. So, very briefly on COVID and immunity, there's actually very promising evidence that multiple natural compounds in foods could both improve the immune response, in particular the T-cell response, and also reduce the severity of excess inflammation from COVID-19 infection. There's a range of vitamins, other minerals, flavonols, food extracts that have really actually compelling evidence, at least as strong evidence as many of the drugs that are currently being tested against COVID. This includes evidence for specific activity against COVID-19 proteins from studies with similar coronaviruses like SARS, evidence for generally augmented cell-mediated immune function and resistance to many other viral respiratory infections. Importantly for COVID, many of these nutrients in multiple models of lung injury, including from viral infections, reduce reactive oxygen species, reduce inflammation, reduce cytokine storm, improve survival. And this is, again, crucial because in COVID-19, the major cause of critical illness, need for mechanical ventilation, and death is excess inflammation in the lung. And in many randomized controlled trials of other viral respiratory infections, many of these nutrients hold promise. And finally, they have very attractive safety profiles, again, much better than many of the drugs that are currently being tested. And so it's first, unfortunate that we don't have better evidence and we didn't have a stockpile of nutrition science to immediately implement when COVID-19 hit. And in addition, we absolutely need critical, urgent, more science and randomized controlled trials for these compounds in COVID. There's also incredible impacts on food security. More than 1 in 5 working adults in the United States are now unemployed. 55 million children are out of school, and many of them receive half of their calories at school. Restaurants are closed, work sites, school, university cafeterias are closed. Supply chains are disrupted. Food can't get to the table, and major products are being thrown away. And yet there's little real-time data on these issues. Most of these challenges are not well documented. We have little real-time data on food insecurity, nutrition, food waste, supply chains. This shows that there's tremendous research needs for monitoring and surveillance. And as mentioned, one of the most striking things about COVID is the very, very strong links between diet-related diseases like obesity, diabetes, heart failure, and hypertension, which individually give many, many higher fold risk of severe outcomes with COVID, and together, may explain the majority of hospitalizations in our country. And this has been seen not only in the United States, but in the United Kingdom, in Italy, and in China, very consistent that in particular diet-related diseases are linked to far higher risks. And while the mechanisms are not well understood, it seems likely from other lines of evidence that this could relate to both suppressed immune function with these conditions, increased background systemic inflammation, and so COVID is like pouring gasoline on a smoldering fire, decreased lung reserve, in particular with obesity, which has been documented with influenza. And then also, these diseases are also signs of underlying poor lifestyle, poor diet, and physical inactivity, which also can reduce immune function and increase inflammation and increase susceptibility to disease. So we absolutely have to be determining how these diseases impact COVID, and how fairly rapid interventions in nutrition, which we know rapid interventions in nutrition can improve diabetes and hypertension within 4 to 6 weeks, how those might help bend the curve. And this is not small change. Americans are incredibly sick. It's fair to say that many more Americans are actually sick than are healthy. Half of American adults have diabetes or pre-diabetes. More than half have cardiovascular disease, causing over 2,000 deaths a day. 3 in 4 adults are overweight or obese. And the economic costs are incredible. Healthcare costs have risen from 5 to 28% of the federal budget, from 5 to 29% of total state budgets, and from $79 billion to $1.18 trillion for U.S. businesses, all inflation adjusted. The annual direct and indirect costs of diabetes and cardiovascular disease are in the hundreds of billions of dollars, and obesity in the trillions. There are critical research questions and opportunities that can help us address all of these challenges, and these are summarized in a forthcoming white paper. For time, I'm not going to be able to go through all of these, but there's incredibly important opportunities for cross-governmental planning and prioritization of nutrition research, to advance the science for dietary recommendations to the public, to leverage new technologies in data science, and to accelerate foundational basic science research. As just one example, the molecular basis of nutritional programming in the first 1,000 days of life. Or another, the molecular and health effects of flavanols and trace bioactives, additives, FODMAPs, food processing. There are so many questions that we need to address. Additional opportunities, crucial opportunities, are to understand and address diet-related health disparities, the multi-level determinants, the influence of individual and sociocultural industry marketing and food environment factors, the roles of other discriminatory policies and practices, and the corresponding translational solutions. There's also, beyond diet-related health disparities, many opportunities for translational and implementation science. We need to understand, for example, how many of the current federal investments, the Dietary Guidelines for Americans, food labeling, health claims, restaurant menu labeling, SNAP-Ed, these are enormous investments, how these effectively improve public choice. We also need to perform interagency research on food as medicine, how medically tailored meals, produce prescriptions, nutrition, medical education, or medical nutrition therapy can be better integrated into healthcare. And then finally, there are critical cross-agency research priorities. USAID, $27 billion per year. The USDA Nutrition Assistance Programs, $100 billion per year. Research questions that are critical to the DoD on optimized performance, recovery from injuries, critical to the VA, critical to NASA, food safety research for FDA and USDA, and then all of the research that underpins FDA regulatory decisions on nutrition facts, dietary supplements, health claims, food additives, standards of identity. There's critical research priorities that need to be expanded, strengthened, and coordinated. The intersections with supply chain, sustainability, food production, COVID-19 has really called out the limited strengths of this system and the frailties of this system, and there's so much to study, both in supply chains, novel regenerative agriculture, and more. And then, as mentioned at the beginning, we need much more monitoring and surveillance, which both informs research and then research informs what we need to monitor and understand in the U.S. food supply. The return on investment will be enormous. We know from prior studies that just general NIH funding provides a manyfold return on investment. As mentioned, USDA and USAID have over $127 billion in food-related investments themselves. We need to reduce diet-related diseases and the hundreds of billions of dollars in spiraling healthcare costs from these conditions, in particular obesity-related and diabetes-related diseases. To catalyze and unlock economic growth through new public-private partnerships, capital investment, small business creation, job creation, to advance the economic vibrancy of U.S. farms and rural communities, and to improve our resilience against acute threats like COVID-19. In 2019, the Director of National Intelligence reported to Congress that our national disinvestment in science and technology is one of the 10 global threats to our country because we're weakening our economic competitiveness. And in 2018, the FDA commissioner said that improvements in diet and nutrition offer us one of our greatest opportunities to have a profound and generational impact on human health. The public health gains of such efforts would almost certainly dwarf any single medical innovation or intervention we could discover. And so I think the evidence is clear that it's time for a national moonshot to strengthen and fix our food system. We have to leverage food as medicine, build a strong public health and food systems infrastructure, and rapidly expand critical nutrition research. As was said by a prominent United Kingdom physician, a massive campaign on diet would save lives and change the course of our nation's health forever. And that is absolutely true for the United States.
Thank you, Dr. Mozaffarian. Now we will hear from Dr. Sheila Fleischacker, founder and president of Fly Health and an adjunct professor of law at the Georgetown University Law Center. She'll be providing an overview of the forthcoming white paper on strengthening federal nutrition research.
Hello. Building on Dr. Mozaffarian's presentation, I will provide a brief overview of the forthcoming white paper entitled Strengthening National Nutrition Research, Rationale and Options for a New Coordinated Federal Research Effort and Authority. Today, I will focus on the findings from our Aims 2 and 4, what we refer to shortly as the Landscape and Options sections. The Landscape section provides an overview of the rich history and contemporary efforts of the more than 10 federal departments and agencies that currently invest in critical nutrition research. A new federal research investment and coordination structure must leverage, harmonize, and catalyze these efforts. Given no current or complete accounting of all federal research funding, nutrition research funding is available, we obtained and collated information on the largest departments and agencies focused on nutrition research and their current estimated spending on nutrition research. We estimated the annual federal expenditures on nutrition research were just over $2 billion. The NIH is the largest funder, with nutrition research investments estimated at $1.9 billion annually. The NIH nutrition research funding is about 5% of total NIH funding. About 1.3% of NIH funding goes towards disease prevention research regarding nutrition research. This white paper provides an extensive history of nutrition coordination at the NIH, including updates on their long-awaited nutrition strategic planning efforts and recent release of a request for information on precision nutrition research. We also provide a rich history of USDA nutrition, which is the second largest funder of U.S. nutrition research, with an estimated annual budget of about $0.17 billion across several institutes and services. A 2015 USDA report illustrated how federal expenditures for nutrition research increased since the 1980s, but not at the USDA. In 2020, the USDA put forth a new science blueprint for the next 5 years to help promote synergy across the department for prioritized objectives and strategies. This blueprint includes specific objectives in nutrition and health promotion. But how new funding streams, leadership, and coordination structures may help achieve these laudable goals were not addressed in the blueprint. In addition to the NIH and USDA, multiple other federal departments and agencies invest in or rely on nutrition research, including the Departments of Commerce, Defense, and Veterans Affairs. In addition to the Environmental Protection Service, the Federal Trade Commission, the Agency for International Development, the National Aeronautics and Space Administration, and the National Science Foundation. Multiple major reports over the last 50 years have called for greater coordination of federal nutrition research. As one example, the 1994 Institute of Medicine report on nutrition and food sciences, pictured here, called for a second White House conference on food, nutrition, and health, and also the establishment of a presidential initiative for nutrition and food sciences within the Executive Office of the President. Specific to the NIH, this committee encouraged nutrition be represented more strongly within the agency beyond the NIH Division of Nutrition Research Coordination, now disbanded. This included establishing an external advisory group on nutrition. Another option put forth was to establish a division of nutrition within an institute, likely the NIDDK, to initiate calls for specific research, oversee program and center grants, nurture training programs, and act as an advocate for young investigator awards, and develop clinical trial initiatives. This report was a very robust assessment of the state of nutrition and food sciences research, but many of these recommendations were not acted upon. At the time of this 1994 ION report was written, the Interagency Committee on Human Nutrition Research, or the ICH&R, was the major entity charged with improving coordination among federal departments and agencies engaged in nutrition research. And almost 25 years later, with little authority or funding changes, the ICH&R continues this leadership role. Meeting twice a year, the ICH&R works to improve coordination on the following activities: the National Food and Nutrition Monitoring and Surveillance, the Dietary Guidelines for Americans, the Dietary Reference Intakes, food and nutrition regulatory activities, federal nutrition education and promotion, and a national nutrition research database, among others. Several structural challenges have limited the ICH&R's impact. These include lack of any strong or consistent connection to the White House, no specific budget appropriations, no mechanism for reporting to Congress, and absence of any well-supported infrastructure for external advisory input on cross-governmental strategies for nutrition research. Overall, this forthcoming white paper and several prior reports found the ICH&R efforts to be important, but insufficient to address the current and rising diet-related disease burdens, food security, health disparities, healthcare costs, challenges to military readiness, and intersections with food and agriculture production, supply chains, and sustainability. Based on the documented burdens, current landscape of research and coordination efforts, and identified opportunities, 2 priority strategies to strengthen federal nutrition research were defined and reviewed and detailed in the forthcoming white paper. The first was a new authority for robust cross-governmental coordination of nutrition research and other nutrition-related policies. And the second was to strengthen authority and investment for nutrition research within the NIH. These 2 strategies were identified as complementary, with benefits accruing independently and further synergies to be gained by joint implementation. A third identified complementary priority was the need for strengthened investment and coordination in food and agriculture research, education, extension, and economics. Given the scope and focus of the present white paper on federal nutrition research, specific options for this third important priority were not considered in further detail and should be further developed and expanded in future assessments. Here lists the identified cross-governmental strategies for strengthening national nutrition research: a new Office of the National Director of Food and Nutrition, a new U.S. Global Nutrition Research Program, a new Associate Director for Nutrition Science within the White House Office of Science and Technology Policy, a new U.S. Task Force on Federal Nutrition Research, among other options. For each of these options, potential advantages, disadvantages, executive and legislative considerations, and path forwards are discussed in the forthcoming white paper. As one example, the new Office of the National Director of Food and Nutrition would be a President-appointed, Senate-confirmed director serving as the principal nutrition advisor to the White House, heads of executive branches, departments and agencies, senior military, and Congress. This office is modeled after the Office of the Director of National Intelligence It would be charged with coordinating and harmonizing the work of 10 plus U.S. departments and agencies that comprise the federal nutrition research community, and would ensure that timely and objective national nutrition information is provided to key federal leaders. Key advantages of this new office include it's a tested and effective model with dedicated leadership, staff, and funding. A few key disadvantages include the focus on multiple nutrition issues could dilute relative focus on research and innovation, and this office may be too high level to address the on-the-ground infrastructure and investment of key research agencies. The path forward for establishing this new office would include congressional authorization and appropriations and presidential appointment of the director with Senate confirmation. As the nation's largest funder of nutrition research, the NIH is one essential, although not exclusive, home for increased authority, coordination, and funding in nutrition science. Any new NIH strategy must leverage and amplify, not replace and compete with, existing extramural and intramural nutrition research across the 27 current NIH institutes, centers, or offices, or with existing nutrition research across any federal departments and agencies. Key identified strategies were a new National Institute of Nutrition, a new National Center for Nutrition Research, a new NIH Office for Nutrition Research within the NIH Office of the Director, and a new Trans-NIH Initiative in Nutrition Research. In brief, the new National Institute of Nutrition would lead research, coordination, training, and outreach on fundamental and foundational topics in nutrition and health, and would have additive focus areas and funding to existing NIH and other federal nutrition research efforts. Key advantages of a new National Institute of Nutrition include strong leadership, robust infrastructure and investment, and could better address nutrition science that is cross-cutting rather than disease-specific. Disadvantages of a new National Institute of Nutrition include that it requires a new additive appropriation to prevent reductions in any ongoing NIH or other federal nutrition research, and could increase siloing of nutrition research. The path forward requires Congress to establish a new National Institute of Nutrition by statute with dedicated appropriations and by updating the current cap on the number of NIH institutes and centers. A more immediate step could start with congressional inquiry and/or appropriations could fund research to further assess the current status of federal nutrition research and potential options, including a National Institute of Nutrition. In conclusion, we hope this forthcoming white paper provides an objective, informative summary of the burdens, current nutrition research landscape, opportunities, and options for strengthening national nutrition research. Ultimately, we hope it helps lay the foundation for accelerated advances in nutrition research to help improve and sustain the health of all children, adults, families, and children. Now I turn it over to Sarah to kick off the esteemed panel discussion.
Now we will be moving on to our panel discussion. Our first panel is on opportunities for federal coordination of food and nutrition research and policy.
I'm Sylvia Rowe, and I have the honor of moderating this session. So to address these exciting coordination strategies are three experts with highly significant government experience and external perspectives. They are former Secretary of Agriculture, Dan Glickman, who also serves as Vice President of the Aspen Institute, Executive Director of this congressional program, the Bipartisan Policy Center, the former Secretary of, and the Bipartisan Policy Center. Dr. Kathy Witecki is also, she's a former Undersecretary and Chief Scientist at USDA, and today she's a professor at Food Science and Human Nutrition at Iowa State University. Dr. David Kessler, formerly Commissioner of the Food and Drug Administration, is also now the Director and Board Chair of the Center for Science and the Public Interest. I'd like to welcome you all and throw out the first question here so we can get right into a very in-depth discussion on this. So let's start with Secretary Glickman and just ask you, what are the ways in which you think, based on your own experience and what you've observed since you've been external to the agency, in which greater cross-governmental coordination and investment in nutrition could be game-changing for specific agencies?
Well, it's a very good question. I'm delighted to participate. I think a lot of it has to do with priority setting. So in our national government, food, health, agriculture, medicine are stovepiped. And so you have different agencies doing different things, and many of them are doing very great work. But we don't see this issue as a holistic part of both agriculture and medicine. So that's why coordinating between the primary agencies, which is, of course, the Department of Agriculture, the Food and Drug Administration, the National Institutes of Health, so the Center for Disease Control, and many other public and private agencies are stovepiped. So, for example, in the medical profession, most physicians aren't really trained in the area of food and nutrition. In the Agriculture Department, while we did a lot and still do a lot in the food safety and research area, most of the efforts are devoted to production of agriculture. In the CDC, it's more epidemiology and warning of people of, about disease. The NIH does research, but doesn't do a lot of research at all in the area of nutrition. So there really is, and there are more than that, than the four that I just mentioned. There's the Pentagon that does a lot of work in this area, National Science Foundation, others as well. So because of that, we're not able to really come together and collaborate like we should to deal with this question. And certainly the COVID-19 issue has brought that to the fore. That would be my initial answer.
Great. Dr. Witecki, why don't you weigh in at this point?
Sure. I think Dan made some very good points. And I would add that at this point, the epidemic is showing us that there are a lot of failures and weaknesses in our overall food system, and that advice based on science, whether it's to the food assistance programs or whether it's to the medical establishment that's treating patients, needs to be science-based. So a better coordinated nutrition research program as it relates to these issues of the role of nutrition and the role of food in promoting health, preventing diseases, and particularly preventing and now treating a viral disease is very important. Also in the pandemic, we need to be thinking about what the data is that's being developed right now, but is not being collected in any kind of systematic way that would give us insights, both from the clinical nutrition perspective all the way through to the food systems perspective. So what is that essential data that needs to be collected? And finally, we also need to be developing an overall strategy for research about nutrition as it relates to immunity and for the future, for prevention and being in a better situation to respond. And I don't see within our current organizational structures as they relate to nutrition research or to managing response for the food system being coordinated and aided in any kind of systematic way. So the kind of recommendations that we're talking about and that we heard about from a director of nutrition intelligence based on the director of national intelligence through to other structures are things that really I think are worth thinking about at this point in time.
Dr. Witecki, do we have ample surveillance and data collection? Even if it's fragmented, is it sufficient?
I think a big question is whether any of the surveys that are so important for understanding food security are in the field right now. Are they capturing information that we really need to better understand the situation? And if not, are they capable of putting them into the field right away? That's from a domestic perspective. From a global perspective, there's also a threat of famine associated with the pandemic as it reaches developing countries.
Right.
And the surveillance systems aren't in place at all.
Right. Thank you. So Dr. Kessler, you're coming from a slightly different perspective in the government at FDA, but nonetheless highly relevant position. Your thoughts on the need for the game-changing ways that this coordination might help you within, or would have helped you within FDA, will help for future FDA commissioner or other agencies.
I really think this could greatly inform and strengthen FDA's regulatory, food safety, food labeling, and public communication efforts. And the FDA depends very heavily on federal nutrition research from other departments and agencies. People don't realize that certainly CIFSAN and the agency does some research, but it's relatively limited. Even if you go back to the 1990s and the first modern nutrition facts label, you see that the principles with that label was based on recommendations from AG, from HHS. So FDA is very heavily dependent on research, but rarely conducts it itself. And I think when you look at whether it's food safety, nutrition facts labeling, front of pack labeling, restaurant labeling, health claims, standards of identity, food additives, I think the agency could benefit from a more coordinated research effort. And the fact is, and I think Kathy, Dan, and I, recognize that, most of these decisions are very, how do I say, they're person dependent, the coordination. Good relationships between AG, HHS, FDA, CDC. When we work together, the three of us, I mean, it's much easier when everyone really respects and gets along. But the reality is that sometimes, at certain points in the history of our agencies and departments, there's been tension. I mean, there was tension on the first food label. And I think coordination is going to be key. Again, the personal relationships, the data will speak for the data. But I think some coordination could be very helpful.
A champion would help.
Kathy, do you think hunger rather than obesity can bring people together in insecurity, or is it as partisan as everything else?
Well, I do think that everybody is being really shaken by these pictures of, miles-long lines of people queuing up to get food from food pantries. And it was that kind of shocking image back in the 1960s that led to the White House Conference on Food, Nutrition, and Health. So, yeah, I think hunger issues can really bring people who are deeply divided over other aspects of policy together.
It was also,
and I would just
Sinclair.
Add your question. There are some champions on Capitol Hill. I mean, Jim McGovern, Rosa DeLauro, there's Jerry Moran, the senator from Kansas, Pat Roberts. These are people who have tried to strengthen our SNAP program over the years. Some of them are conservative, some of them are liberals. And so I think I think the anti-hunger movement is real, and as Kathy said, these lines are bringing it home. The one thing that's been missing is that we talk, and when we talk about hunger, we often talk about quantities of food and calories more than what's in the food and the nutritional value of the food. And that's true both in the global and the domestic sense. And that's one of the areas that, as Kathy talked about, and David too, why we need much more research, because food is not just food. Some food is different than food. Some food has higher nutritional values and deals with food deficiencies. So you need to talk about both hunger and then what is actually going into people's mouths.
So I'm going to jump in here again and say you all have eloquently delivered the clarion call here for better coordination, better cooperation, and better understanding among the cross agency. And I want to sort of pivot now based on the fact that the white paper has laid out a number of approaches that may be very good options for accomplishing this. And I'm interested in talking to each of you, finding out, do you have a preference in terms of which of the options you think would be most effective, but then what are the strengths potentially of other options that you may want to raise as well from, again, you're very familiar with the options, so who would like to jump in and start?
Well, I guess I can jump in. When we first started working on the white paper, our focus was on nutrition research. And at that point, I was thinking that the model of the Global Change Research Program, which would require legislation to establish an office that coordinates the research agenda, would be the way to go. And it was based off of the Global Change Research Program. So it would be a global nutrition research program. But the pandemic has really showed up the weaknesses not only in research, but the implementation across programs and our responsiveness. So I am now of the mind that, to your provocation to us earlier, do a thought experiment. With the pandemic, really what we need is a high-level director who can not only coordinate the research, but also use that to inform policy and actions that need to be coordinated across the federal establishment. So under these circumstances, I'm favoring the Director of National Intelligence model.
Right. Which, by the way, you could argue that all of the issues that you're addressing are as fundamental for national security as the National Intelligence Office. So, very, very. David, Dan, your thoughts on this?
I would say a couple things. One is, an honest answer is, I don't know what the best model is. I do know that what we need is leadership, and in this area, there's historically, I mean, David and Kathy are the examples of two very excellent leaders who were able to move their agenda even with the difficulties in our political system, because they were committed and their goals were to really serve the public interest. So however we organize this thing, you've got to have people of leadership who give priority to health, nutrition, medicine, and agriculture in a coordinated way. Now, the second thing is, I've been involved in government for so many years that I've seen boxes move around in government all the time, and sometimes it makes a difference and sometimes it doesn't, and sometimes, you move an agency here or an agency there, and for a few years it works out okay, and then a crisis hits and it doesn't work out okay. I still like looking at the NIH as kind of as a lead model as a place that would kind of help coordinate the research on the nutrition side, because it's gotten to be a very low priority there. Recently, in the NIH's annual report, and this may be a silly anecdote, but the word food was only mentioned once in their last annual report. And what it was somebody mentioned the words Food and Drug Administration. So it was just the agency that was mentioned, not the word food. And so I don't want to make too much out of it, but it does indicate to me that in all the various institutes there, they all have to relate to food in terms of how they examine and do scientific research on disease and those kinds of things. So whether it's a National Institute of Nutrition or some dramatically elevated position within the NIH that's charged to work with USDA, which has a lot of great research going on, but not as much focus as I would like, and the FDA and the CDC, which I think could stand to have more research going on. You need some leadership in this organization. And the problem in Washington often is when you get into the battle of which agency is going to be the leader, you end up with a leaderless type of organization. So that's why I'm now thinking you need to one of the existing organizations that is given the imprimatur to take the lead. That would be an answer right now.
Well, Dan, certainly your anecdote illustrates the point you raised earlier about the siloization, if you will, of food and nutrition within the government. David, your thoughts on this? Your favorite?
Yeah. I think Kathy and Dan summed it up pretty well. I would just add that, at the end of the day, the Secretary at HHS, the Secretary of Ag, the respective committees, the Commissioner of FDA, none are going to easily cede jurisdiction. That's the reality. So the goal here is to find that both the individual and the place where those individuals, those principals, see that they alone can't accomplish certain goals. And in some ways, if you get them to help set the agenda collectively, and this function fills that gap, then it would be successful and could help move, improvement in food and nutrition.
So, David, you've raised the question of the path forward. How do we move from what you all have so beautifully laid out and certainly articulated the challenges as such? How do we move to positive action, basically to address one or more of these potential options? Because they will be different in strategy and tactics, both. Dan, let's start with you.
Well, again, because of my experience in the Congress, I would hope that the Congress would hold hearings on this specific subject, how to better elevate and coordinate nutrition research in our national system. I recall in my days at USDA, and maybe Kathy, you can recall as well. I recall a few hearings on the issue of SNAP and school meals, but they were not very intensive type of thing. I think this needs the kind of national legislative attention that our national security agenda gets. And again, it's got to relate to other issues such as public health as well. But we've got a lot of people at Tufts University and other schools of nutrition around the United States that are finally recognizing the need for this, particularly because of the comorbidities associated with COVID-19. But I'm hopeful with our papers and the intellectual work, which Kathy has been so much involved with herself, that we can raise the issue to get congressional attention.
Great. David, your take on the path forward.
I think these things take persistence and patience. I think issues like this, ideas like this, you stick with. They don't happen overnight. But if you keep this discussion going, at the right time, there will be individuals in positions move this forward, and who will take this issue on and move it the next step. Not going to happen overnight, not going to happen immediately, but it's our job to continue to push this discussion.
So Kathy and Dan, how can we add that urgency that David said we desperately need now, rather and take advantage of where we are today and the issues which have been so dramatically illustrated by the COVID-19 crisis?
Well, I think we need to do two things, and I agree with Dan on oversight hearings and certainly working with those members of Congress that have had an interest in nutrition and who are on committees with jurisdiction to encourage that kind of hearing would be one action item. And the second is we are in a presidential election year, and we should be working to inform the candidates about the importance of nutrition and work with the campaigns to make sure that nutrition is part of their thinking. So I think there's two action items.
Excellent. Dan?
I think Kathy's hit the nail on the head. The political process is in play. At the same time, we have this public health crisis right now, and sometimes out of crisis comes the opportunity that's so desperately needed. And with a couple thousand people a day dying, in many cases because of in this country, because of comorbidities issues, in part caused by poor diet, poor health, poor prevention. Let's hope that we can make an opportunity out of this.
Excellent. Well, I want to thank all of our panelists, and I want to say that your inspirational remarks, I hope, have created many champions out there that are listening to you, understanding, and see the potential for the future. Thank you very much, Secretary Glickman, Dr. Witecki, and Dr. Kessler.
Thank you.
Thank you.
Our second panel of the day is on opportunities for NIH nutrition research.
Hello, everyone. I'm Dr. William Lee from the Angiogenesis Foundation, and today I'm delighted to be moderating this panel, Opportunities for NIH Nutrition. We've got a truly distinguished group of panelists. Senator Tom Harkin, former U.S. Senator from Iowa, Dr. Patrick Stover, Vice Chancellor from Texas A&M University, and Dr. Van Hubbard, Rear Admiral, Uniformed Services University. Thanks everyone for joining. And as you've heard earlier today, there's a white paper being developed focusing on federal nutrition research. And today, we want to speak more specifically about that topic. Building off the earlier panel and diving a bit deeper into NIH, the largest funder of nutrition research, we want to have a conversation, an open conversation about how NIH can play a greater role in strengthening federal nutrition research and what are some of the issues that have arisen, especially with a heightened awareness of health in the COVID-19 era. With that, let's dive in. Patrick, let's start with you. From your perspective, what are some of the research opportunities that can be transformative for nutrition and public health?
Certainly, and thank you. I think if you look at this report and the timing of this report, we see how critical right now nutrition is in the lives of every single American, both in terms of the health effects for those who have diet-related disease, but also on the greater impacts for the overall food supply. And so the time is now really to make nutrition research a priority. And there's other factors that make the timing right. Not only do we see that nutrition is a major driver of public health and public health related healthcare costs, again, due to that diet disease relationship, but at the same time, we're also seeing that we have tools and technologies available to us to address nutrition research that have never existed before. In the past, nutrition research really focused on understanding nutrients and pathways and physiology, but in the context of avoiding nutritional deficiencies. But now, as we know, with the increased awareness of that connection between food and health, now we're beginning to look at the role of nutrition in chronic disease prevention and chronic disease management. And this has really moved us into the area of systems biology and understanding complex systems, which the types of research and questions we didn't have the tools and technologies. But now with the advent of systems biology and more computation and engineering coming into physiology, coming into medicine, the time is right to begin to address these complex relationships between diet and disease in a way that'll benefit public health.
Van, you know, you're having some experience with NIH. What are some of the infrastructure characteristics that would be needed to be able to take full advantage of the modern tools that are available?
Okay, thank you for the question on that topic. I think everybody is quite aware that nutrition is a multifaceted area of science as it relates to biomedical health. And there are multiple institutes and centers and other funding components of the National Institutes of Health that are involved in supporting, to some level, nutrition research. It is quite important to try to also achieve some coordination across all of the funding components of NIH, as well as attempting to coordinate efforts with other agencies and departments. But within the NIH, what is really something that would be of value is to have a strong advocate for nutrition taking part in the senior leadership interactions where all representatives of the NIH composition are involved and can discuss the needs and the areas that would warrant further investigation with regard to the nutritional sciences. At the same time, within NIH, a lot of people have also considered the review, should it be more concentrated or what? And from my experience, I think that it is a benefit to have the broad area of nutritional sciences go to as many funding pools as possible, because that maximizes the probability of increasing the level of support when you're tapping multiple funding pools.
Great. Senator Harkin, you know, the NIH is the world's leading research institute, and its leadership is really made possible by the guidance and structure that Congress has provided for it. Can you give us some perspective of how we arrived from a congressional perspective to the NIH of today, and what would be needed to actually, from Congress's perspective, to help guide NIH into a nutrition-oriented group for tomorrow?
Well, thank you. I think that some of my comments will overlap a little bit with what I heard on the first panel this morning, both from Sylvia and also from Dan Glickman. Let's recognize one thing. There is a general recognition among members of Congress that bad diets and bad eating habits are leading to more diabetes, earlier onset of diabetes, heart disease, obesity. This affects productivity among workers, and it has a huge impact on our healthcare spending, both in Medicare and Medicaid, but also in the private insurance sector. We've had a lot of studies from the RAND Corporation, for example, on the impact of chronic diseases in America. But the problem is that while individual members of Congress recognize this, and if you speak to them personally, they know that we need to have better diets and better nutrition information. But what happens is that collides with, the RAND studies and everything else collides with the food companies, food processors, fast food chains, marketers, and with the structure of American agriculture. The structure being that we have, since World War II, basically subsidized fats, sugars, starches, red meat, poultry, and pork. But we have not subsidized very much fruits and vegetables and other nutritious type crops. And we have never really, USDA has never really had any kind of a support column or direction towards nutritious crops at all. So again, while the individual member might say, yes, we'd like to do this, but they're confronted with constituents and everything else. This is where an NIH institute would be so helpful. As a truly independent and respected entity, that's what NIH is, that could do nutrition research unimpeded by the influence of market forces or food companies or big ag, etc. That would give members of Congress the backstop needed to move forward on better food policies leading to better nutrition. I think this entity from NIH, because of the independence of NIH, because it's removed from those lobbyists and other forces, I don't mean to use that as a pejorative term, but from those influences that hit USDA and other entities, and they do their research on sound scientific basis, then I think this would be information to the general public that would lead to better nutrition and better health. I think it would also lead to more research in the private sector, actually develop more tasteful and ready-to-eat nutritious foods. I also think an entity like NIH, because of its strict independence, would also lead the USDA to have a better nutrition-based policy. Lastly, I think it could lead to major changes in the Farm Bill legislation that comes up every 4 or 5 years to provide, again, more support for nutrient-heavy crops that are nutritious and that are cheaper because they're more subsidized by Congress than what they've ever been subsidized before. Keep in mind, it wasn't until 2008 and I'm trying to remember the Farm Bill, 2006, that we ever provided any support to fruits and vegetables at all in America. So NIH, in short, that independence, sound scientific research can have all kinds of residual benefits in changing policies at the federal level.
Just to follow up, Senator Harkin, what do you think the likelihood would be that Congress would pass the legislation needed to establish such an institute at NIH dedicated to nutrition?
Well, some things that have to fall in place. And again, I don't mean to sound political, but what the hell? With the present administration, you're not going to get anything done. It's just not going to happen. If there is a change in the administration for next year, I would be one who would be advocating that all of us engaged in this effort should begin to have a master plan for how the new administration would take the lead, have a plan for developing, as Sylvia said this morning, the champions, and Dan Glickman pointed out 2 of them, Rosa DeLauro and Jim McGovern, to have them have early hearings next year on this, start getting the scientific community behind it. Former directors of CDC, for example, almost all of them I know, I think would be very supportive of something like this. And again, to try to enlist the at least tacit support, but at least, if nothing else, not an adverse reaction from Francis Collins, the head of NIH. He's very well respected on both sides of the aisle. And his opinion would go a long way. It would be great if we could get him to support such an institute. But having funded NIH for so many years in the Senate, I can tell you there is a kind of a feeling, I think, among Francis and others that we have enough. We don't need to keep expanding the institutes. Well, every time they say that, we always get a new institute, one way or the other. So I think with a new administration, with all the study, with everything sort of that's kind of coming to a head now, I think Pat Stover mentioned that it's sort of the time is kind of right for something like this.
And Patrick, clearly, as Senator Harkin has mentioned, NIH is one of multiple players that all can have skin in the game to help improve our nation's health through nutrition research. What are some of the other federal agencies that you think could be positioned or optimally positioned to really help be transformative with a new Institute of Nutrition?
Absolutely. Thank you for the question. Nutrition should never be disconnected from food, should never be disconnected from agriculture. And certainly Senator Harkin just talked about that linkage across the whole food supply chain. So the science really can't be separated either. It has to be coordinated. So where you have the National Institutes of Health that primarily really focuses again on issues of human health and human disease, the United States Department of Agriculture, both through its in-house research as well as through NEFA, the Competitive Grants Program, really looks at that connection between agriculture and food. And I think there would be complete consensus across this committee that drafted this report, as well as I think all scientists, that we could do better in having a more seamless connection between agriculture, food, nutrition, and health. And that's where this option report, where it talks about various ways that we can make greater investments in nutrition research, but also how we can have better coordination across, not only within NIH, but across federal agencies that fund research to make sure that we're fully leveraging all of the opportunities and all of the good research that needs to be done, such that we have a more seamless transition between agriculture, food, nutrition, and health. And so the report, I think, does a very nice job of looking at historically how nutrition research has been conducted within the different federal agencies, primarily focusing on NIH and USDA, how there have been structures put in place to coordinate that research across the two agencies that have those two very different missions. Yet again, nutrition is cross-cutting, so how that research has been coordinated. And the report does a very good job, in my view, of looking at both of the historically, the strengths and weaknesses of various attempts to coordinate. And that's where I think the report is also going to be valuable in really calling for better coordination and dialogue across federal agencies that fund nutrition research, not only to increase the level and commitment to nutrition, but also to make sure that we don't silo the various segments of the food system in that connection between food and health.
So looking at prior reports for the DRIs and DGAs, the Dietary Guidelines for America, clearly research gaps still exist in our understanding of human nutrition and how food can influence the body's capacity to maintain health or to resist disease. The research gaps are able to be addressed, shouldn't be on the shoulders of just one agency, but really could be addressed by collaboration across different agencies. How might you see, Van, how might these gaps be coordinated, given the fact that there are different agencies that each might have their own contributions to be made? What might a future look like where research gaps actually involve the coordinated approach across different agencies?
Well, to build upon some of the comments that Patrick has just made, I think it's very important that there be dialogue among the various funding agencies, especially within the federal government, but not limited to the federal government. There are foundations and even some industry supported research that could be of value as we move forward. I think it's very important that we must be aware that there are many research issues that will benefit from advances in basic science, and basic science can be carried out largely, with NIH support, but also some of the other agencies. I think we're talking about NIH a lot here just because up to now, NIH has been the largest funder of the nutritional sciences. But as we move forward, we also are moving into an area where there's a need for more interaction and bringing together the expertise that the different agencies and departments may offer. And so we have to have that dialogue among the federal agencies and departments. Now, one of the issues is each of the agencies have their own methodologies or procedures to follow in order to fund research. They have different timelines. And so you need to have some of the senior people involved at the different agencies and departments to have this dialogue to see how best things can be worked out to have that coordination. One of the other things I wanted to mention is as we move forward to the public health and dealing with chronic disease prevention and management, we have to remember, unlike historically where we're talking about nutrition deficiencies, it isn't a simple matter of just supplying one type of nutrient to cure the problem. We now have a situation where it's a complex interaction among nutrients that are found in various foods, interacting with the total caloric intake, interacting with other lifestyle parameters, especially physical activity. And all of these make the research much more complex. And it also requires some longer duration type of studies in order to come up with results that will bear the test of time. And so the more we can have the interaction and tap the expertise of the different agencies, the better off we will be.
I think the importance of basic fundamental research across agencies is so important to coordinate and to look for advances that can emerge from those, much in the same way that multiple agencies, led by the NIH primarily, has led to fundamental discoveries leading to better pharmaceutical development for the treatment of disease. Senator Harkin, you actually mentioned something really interesting, which is moving beyond the past era of just supporting nutritional deficiencies to really using food in a more proactive way to manage disease or potentially even treat disease. This is sort of the, more popularly viewed as the food as medicine movement, but it's actually much more than that. And as it can be grounded in science. One of the things that would be important to look at, and I'm sure you've had a lot of insights into this, would be the role of the FDA, which has primarily been focused on food safety, which is something that it rightfully should do to help protect Americans. But how would you see a transformation within FDA to be able to support the use of food in a proactive way to manage disease?
Mute off. That is an excellent point. How do you get FDA to move on that? Well, again, I go back to my square one. I said I think that having an institute like that at NIH that is independent, that can then give out their grants to do other basic research, as most of the research is not done in-house at NIH, it's done in universities across the nation. That again would be a kind of a backstop for FDA to start changing their viewpoint on nutrition. and food as you said, food as medicine, but food as nutrition, and getting them just not only to look at food safety, but to start looking upon an expanded role, which many of us have talked about for years, that FDA should have an expanded role of providing the general populace and members of Congress with information as to the choices in food and what that food means for their health, their wellness, their well-being, their lack of need of medical care. But I still think that FDA is not going to do it without better scientific independent research that would be done by NIH. I also want to just say that all nutrition foods, when you think about the largest federal program that we have for the purveyance of food, it's the school lunch program and school breakfast, but mostly school lunch program. Under Tom Vilsack and others, we started making some changes in that, and as you know, it was pretty tough. Although I think we've survived most of it, but the FDA was sort of a bystander to all of this, and I'm hopeful that in the future the FDA would be more proactive. But I go back to what I said earlier: having an independent research agency like the National Institute of Nutrition at NIH would give the FDA the kind of backstop that they need to do that.
Well, this brings us to the end of this session. I'd like to thank our panelists, Senator Tom Harkin, Patrick Stover, and Dan Hubbard, for their terrific and insightful conversation that's related to the white paper about the opportunities to advance nutrition research in America. Thank you to all.
I would like to thank all of our excellent speakers and panelists for this afternoon's session. This has been a very informative and engaging session. And now I will turn the program over to Dr. Paul Coates, ASN's Vice President-elect, with closing remarks.
Chris, are we live?
There he is.
Chris, are we live?
Thank you again to all of our panel moderators and speakers. I appreciated today's discussion as well as your valuable insights on the need to strengthen federal nutrition research as well as some potential options to help achieve that goal. Before we turn the program over to Dr. Coates, we have time for live Q&A now, and we are lucky to have a number of our panelists joining us in order to answer your questions. If you have questions, please feel free to enter them into the questions box at this time. To get us started, we do have a few questions. Where will the white paper be available to view, and when might it be available?
This is Paul Coates here. I think I'll take the job of directing questions to people, and I'm going to stick Sheila Fleischhacker with this one.
I think that would be. Yeah, we were originally planning for the conference, but as COVID has adjusted many timelines, we adjusted ours. We're aiming for June 23rd, and it will be within the American Journal for Clinical Nutrition, so we'll disseminate accordingly through their networks. Thank you.
Okay.
Great. Thank you, Sheila. Regarding collecting data from participants to improve nutrition databases, which can be costly as well as time-consuming, how can we use mobile data collection, link nutrition data collection with other data collection efforts, or improve tools to reduce the time burden for our participants?
Although this question probably has more to do with some specific kinds of research issues and not necessarily with the issue of how do we strengthen overall federal nutrition research, I'm going to see if Kathy Wotecki might have an interest in answering that question about using mobile data.
Sure. Well, I think one thing to point out is, with respect to this question, is that between the time that we filmed our conversation that you viewed and this session, the NIH came out with its strategic plan on nutrition. And within that, there's an emphasis on all different kinds of data. And we're in this era of big data, data analytics, and how can we make better use of data? So, and which comes through loud and clear as a priority within the NIH strategic plan. And within this year also, the Department of Agriculture issued a strategic plan for agricultural research for 2020 to 2025. And it has data also as a primary area for research across the entire food spectrum. So with the opportunities that we have with big data and data analytics, I think we're going to see more and more with these of the federal agencies putting priority on then what are the technologies that we need to put in place that can inform different types of research questions related to health across the board for NIH, for food across the board for USDA, and for us in nutrition science as well. So I think that we'll see this question of what technologies, mobile technologies, as well as other kinds of data that are being generated within the food system, within the health system, and how can we access them to shed light on questions that are of importance to our different research agendas within nutrition. So that's how I take a first cut at that question.
Yeah, no, I think it's a good start, and it actually reflects on something that Patrick mentioned in his presentation about really innovative new strategies and tools. Patrick, did you want to comment on this at all?
Again, if you look, there is so much innovation right now in the private sector, looking at various, wearables, other types of omics data that's really giving us so much information about that variability in response to diet. But it's driven right now, at least from my perch, by the private sector. We need more research along those lines, again, that better connects food and health for the purpose of lowering healthcare costs, and do that in a way that is coordinated and efficient, and allows innovation that everybody is so excited about right now to really be married with these national priorities we have to address public health through nutrition.
Could I see if anybody else on the in this group would like to weigh in? This is provocative and a good case study for one of the things that really is important about this overall effort of coordinating research across the agencies. Would anybody else like to weigh in?
Hey, Paul, I will just remind everybody during Francis Collins' remarks yesterday, he actually mentioned the issue of food intake and improved technology for the use in that type of methodology, and specifically new ways of doing data analysis with all big data systems and computers, so that we can look at better ways of analyzing that data.
Thank you. Dari?
And Paul, I'll add, I think, as you'll see when the white paper comes out, we didn't aim to identify or specify exactly what research priorities would be. Our white paper is more about structure and process. And so we identified a range of major issues. And so this issue of technology, data science, omics is huge. The issue of the gut microbiome is huge. The molecular basis of nutrition in the first 1,000 days of life, or healthy aging for seniors, is incredibly big. The intersections between food security and social determinants of health and health-related disparities. there are so many big questions that we've identified. And so I think what we hope and what we aim is to strengthen the process and structure to address all of these questions. And I think what comes out from our review, we really tried to have an extensive, deep review, is that what's most important is not even the next few years of priorities, but what's going to happen 10 years down the line, 20 years down the line, because nutrition is one of the major 21st century challenges. And the COVID-19 pandemic is showing us how important it is to have a resilient population, and food is one of the major routes to get there. So I think that, my response would be, this is absolutely important, precision nutrition, big technology monitoring, but more important than identifying which priorities, is the structure. And in that way, our report is very complementary to the USDA Science Blueprint that just came out this year, and now the NIH Strategic Blueprint for Research that just came out this year, both of which have excellent goals. And so we are really providing an operational platform of options goals in the next 10 years instead of in the next 50 years.
This is such an interesting topic area because, as Patrick pointed out, the notion of the private sector being the sort of the hotbed of innovation that may be very valuable for a lot of the aims that we see need taken care of going forward. And so I'm wondering whether I could put Senator Harkin and or Secretary Glickman on the spot to just comment on how do we make that marriage happen with industry, academia, the federal government in both the legislative and executive branches, because it's a kind of an interesting nexus. Do you have any insights into that, either of you?
I'll go to the senior senator first. He's there.
Any thoughts, Senator Harkin?
Well, I guess I keep going back to my 40 years being on the agriculture committees of the House and the Senate, chairing the Senate Agriculture Committee for 2 farm bills, and then wearing another hat as chairman of the Health Committee after Senator Kennedy died, and funding NIH through all these years through the Appropriations Committee. So I've seen this over a long period of time. And you talk about harmonizing and bringing these together. There's so much political and industry influence that comes into the Department of Agriculture. And I would say even FDA too, and others, that it's hard to get that kind of really sound science and research done that is really cordoned off from those kind of influences. That's why I keep coming back, that you need, and I think Congress needs, policymakers need that kind of an entity at NIH that is once removed from politics and industry influences and lobbyists and all that kind of stuff, to sort of set the table for what we're talking about in terms of good nutrition, nutritious foods, and then from that, it would inform, I think, the Department of Agriculture and others in their policies. But to try to put them all together in the same corral right now and to come out with some kind of a coherent policy is impossible. It's impossible.
That's a very straight up answer, and I appreciate it. We could probably take a lot of time to discuss the implications of this and the implementation of identifying the areas of need and potentially developing, for example, public-private partnerships for the implementation of some of the.
Paul, could I just add to that answer, Senator Harkin? I'd make a couple of points. Number one is lots of industry now is recognizing that the health and productivity of their employees are impacted by, how people eat, whether they're obese, and the general prevention. And so you're finding just an abundance of American companies, larger ones usually, getting into this and working with in the insurance industry in terms of how they rate their employees on and costs of insurance. So I do think there's a lot of work being done in the private sector and especially with the insurance industry in trying to get their handle on this problem. The second thing I would say is we haven't really talked about the consumer today. The consumer desperately needs good information about nutrition. And the consumer is bombarded by inconsistent anomalies about what to eat and what is good for you. And those anomalies are accentuated by marketing and advertising of foods, which by and large are high caloric and not good for you. You rarely see strawberries and blueberries and related products advertised on television or in social media. So when you get all these things together, you have a consuming public that operates with really some degree of ignorance as to what is right, what choices are available to them. And the USDA has done a good job in school meals. I applaud them for that. But that's why your community, the community of nutrition, needs to highlight this subject because there are so many countervailing influences. I don't know how much television you watch. I watch a lot of it. I'm not proud of it, but I watch a lot of television. And when you watch what food is being advertised on television, most of it is high caloric, high sodium, high sugared up food that is just not good for you. And that has as much to do with consumption. And part of that has to do with the information about what one should eat is not presented in a way that's very, very carefully targeted either the medical community or the consumers at large. That's a whole other side of this picture. But what you do in the nutrition side should be able to influence those messages to consumers. And right now, there really is a disconnect there, I must tell you.
Good, thanks. Senator Harkin, you had something else you wanted to say?
Well, to buttress what Dan just said, the National Bureau of Economic Research up in Cambridge just came out with a study in March looking at the impact over 10 years of the menu labeling in the chain restaurants. That was part of the Affordable Care Act that mandated that chain restaurants had to put out on their menu the caloric information for everything that they serve. Well, there was a lot of pushback on that, but anyway, I won't go into the story how we got it done, but we got it done. Consumers are using that. And the Bureau of Economic Research report in March shows, as they said, a small but statistically significant reduction in body mass index and among people over the last 10 years who have been reading these menus. I think to follow what Dan said, what that's doing now is it's pushing the industry to provide different kinds of foods. I know that Subway, I don't mean to pat anyone on the back, but Subway has changed a lot of their menus because people come in, they read the caloric intake, they order something different than what they've been ordering before. So the consumer really does want that information, and they will use it if it's provided.
Thank you. Sarah, are there any other questions that have come up?
Yes, we did have another question sort of along these lines of how can federal nutrition programs and policies be better coordinated? Are there any ongoing efforts at this time that focus on that type of alignment?
So I think because of the time I spent with Kathy Wotecki on the Interagency Committee on Human Research, I'll ask you to comment since it's sort of a structural piece that's already in place, but
yes.
I all agreed that there needs to be much more. What do you think?
Yeah, well, you're absolutely right. On the research side, there is the in existence an Interagency Committee on Human Nutrition Research. It was established a good 30 years ago or so, and is co-chaired by the Assistant Secretary for Health at HHS, and the Undersecretary for Research, Education, and Economics at the USDA, with membership from all of the 10 agencies that and departments that Sheila Fleischacker had on one of her slides. It went out of business during the George W. Bush administration and was revived actually at the request of scientists within the federal agencies working on nutrition problems, research problems in particular. So it is in existence and provides a coordination focus. But it's been very helpful in coordinating the work on the dietary guidelines as well as on the DRI process. But it is shared, as I said, by two people who are in their jobs for a relatively short period of time, political appointees. It doesn't have behind it the weight of law. So what the agencies bring are voluntary things. And like Dr. Kessler was talking about, if there's good rapport within leadership among agencies with an intention, then something like that interagency committee can be very effective. And if there's not that kind of top-down interest in nutrition as a part of an administration's agenda, it's going to have a much harder time keeping the agencies focused in all of their work coordinated moving forward. So yeah, that's one way of coordination. Another is on different topics. The advisor to the president on science and technology, the head of the Office of Science and Technology Policy, through the National Science and Technology Council, can convene committees to focus on a specific topic and to report out. And then over time, to continue the coordination activities that might go on within that specific topic. In recent years, there have not been that many topics, not any that I can think of right off the top of my head that NSTC has been involved with, but that's another mechanism that exists and has been used. But on the other questions we were talking about and about the food assistance programs, there are, under the emergency response protocols that the federal government has, ways that the federal agencies coordinate on that food response. But again, that's something that's brought in under a special kind of emergency situation and not from the longer term perspective of how do you develop programs and policies based on the scientific evidence.
Good. Thank you. Dari, you had something you wanted to comment on in this?
Yeah, I think that one thing that came out of our investigation, which is summarized in our white paper and Sheila Fleischacker summarized, is this incredible need for coordination. And we actually found over 50 years, major government reports, congressional committee reports, Institute of Medicine reports stating that the food and nutrition research and the food and nutrition programmatic and policy activities of the federal government are not sufficiently coordinated. And yet, out of all those reports, sufficient authority and structural change was never really initiated to address these things. And so this is one of the major findings of our analysis. And we, again, Sheila highlighted some of the core recommendations to address that. And as one of them, you know, after September 11th, the Office of the Director of National Intelligence was created because it was recognized that we were investing tens of billions of dollars in national intelligence, but that was fragmented and not coordinated. And so the Office of the Director of National Intelligence was created, a cabinet level position that coordinates and harmonizes national intelligence, reports to Congress, reports to the White House, reports to the Joint Chiefs of Staff, and it's been very successful. And I think COVID-19 has highlighted that food is a national security issue just as well. And we have, more than 100,000 Americans dead, and incredible fragmented supply chains, and a shutdown economy in many ways exacerbated by poor metabolic health. And so this is also a national security threat. And I think we have a moment to think about one of the options, which is to create a new Office of a National Director of Food and Nutrition, who would coordinate the over $150 billion of federal investment in food and nutrition programming beyond research, USAID, the domestic food nutrition programs, the research, and much more. And so we really do think that the coordination at the highest levels is important and would help national security, and that this is the right time.
Thank you very much. I'm going to need to end the question and answer component of this now. We've left, I think there are a lot of things on the table that are going to want discussing. And I'd like to just make some closing comments, and then you will be able to move on to your next step, folks. I wanted to thank all of the speakers, the moderators, and the panelists in today's session. On behalf of the American Society for Nutrition, I want to end the session by making a few closing comments. First, it's evident that we live in an era when the role of food and nutrition in health and disease has become foremost in the minds of people of all stripes. It's also evident that we face challenges as nutrition scientists and professionals in helping our citizens to make the right food choices. For one thing, the science is always evolving. On top of that, conflicting reports that are often based on a slice of the science rather than on the totality of the evidence makes our job of effectively informing the public all the more difficult. And to add further complexity, the pandemic of COVID-19 has exposed the risks to and the shortcomings in our food supply chain and has upended what we think of as normal. The white paper that frames the discussion at today's session, along with the thoughtful remarks of our speakers and panelists, point to a range of options for enhancing the nutrition research effort in the U.S., most of which is funded and coordinated by agencies of the federal government. They outline a series of options, including some all-of-government approaches and some NIH-specific approaches for enhancing the scientific effort and the translation of those research results into policy, information, and action. From my perspective and that of the ASN, all of the options that are put forth in that paper should be considered and thoughtfully discussed in advancing this initiative. It should be noted that there's a big role for the private sector, which would include industry, academia, and healthcare in this job, and all can have a say in the final outcome. The release last week by Dr. Collins of the NIH Nutrition Research Strategic Plan for 2020 to 2030 is a welcome addition to this conversation. It puts some very important issues on the table, outlining 4 strategic goals for the NIH with a wide range of objectives. They include innovative research to address ingestive behavior, the microbiome, precision nutrition, and approaches to capturing dietary data. Number 2 is to explore dietary behaviors that optimize health. Number 3 is to understand better the role of nutrition across the lifespan. Number 4, the use of food in reducing the burden of disease in clinical settings. This array of objectives underscores the very important ways in which nutrition science interacts with other biological and medical disciplines. The white paper, as you just learned, will be published later this month. June the 23rd is the anticipated pub date. And there'll be plenty of opportunity for you to comment on the options that are raised therein for enhancing the federal nutrition research effort. The ASN appreciates the opportunity to sponsor this session, and we hope that it will help us all to focus on the next steps in enhancing the U.S. federal effort to strengthen nutrition research. Simply put, we need the data, and we need strategies in place to translate the data into informed public policy. As the premier professional society dedicated to nutrition science, ASN has long advocated for expanded government funding for research, and we look forward to continuing to play an important role in defining the future of nutrition science in this country and around the world. In closing, thank you for being part of this session. We look forward to your feedback on the options presented in the white paper. And that's it. That's a wrap.
you